We're in for an exciting day of insightful presentations and discussions. Before we start, I request yesterday's poster presenters to take down their posters to prepare for the next session. To begin with, we have the pleasure of hosting a plenary lecture by Professor Vijay Kuchru, a renowned professor of neurology at Harvard Medical School and senior scientist at Brigham and Women's Hospital, Boston, USA. I'm delighted to introduce the chairpersons for this session, Professor Sunil Arora from PGI, Chandigarh, and Dr. Amit Awasthi from THSTI, Faridabad, who will introduce the speaker and the title of the talk. Please welcome to the stage.
Good morning, everyone, and hope you all are doing good. Yesterday was a great day, and I think we all have to give a big round of applause for Dr. Dipankar Nandi for doing such a great job and arranging this meeting in such a nice manner. So it's a profound privilege for me to introduce the speaker today, Professor Vijay Kuchru. I don't think I need to read it here because I know him so well, and I am sure most of you also know him equally well. He's a very well-known, name in Indian immunology and world over. He keep coming to India on many meetings and including our immunology conferences. And I think we all should be grateful to him because he and some of his close colleagues have been conducting some very good immunology courses for the young children, students and young scientists. For many years now. So many people have learned a lot of immunology from him, including myself. Not only immunology, but how to teach immunology. That's the tact he has been telling us. And it's very nice to introduce him. And I will not stand between you and Vijay. Please, Vijay, take the stage and show us today. You would be amazed that his lab has really, discovered Th17 cells, Th9 cells, and now TIM3. And my co-chair has been trained in his lab in Harvard Medical School when he was doing his postdoc. So a lot of our association. Thank you very much, and stage is all yours.
Thanks very much. Can you hear me at the back? So Sunil, you took 10 minutes off. It's 9:10 now. So just reminding you, when you stop me, so give me 10 extra minutes. So I'm going to talk about checkpoint molecules. And we discovered TIM3 about 20 years ago. And then we, in fact, discovered TIGIT. We didn't call it TIGIT. It was called VSTEM3. So today, actually, I'm going to talk about how there is a misnomer that everybody thinks that the checkpoint molecules only work on CD8 T cells or CD4 T cells. I often seem to rush at the end, so I really am grateful to a number of people who did the work in my lab: Karen Dixon, Lloyd Bode, Amir Sohail, Yuhao, Norio Asaf, Nicole. And Amit actually did a lot of work on IL-27 when he was in my lab. A lot of T-cell exhaustion work was done with Anna Anderson. And I also am grateful to Rafi, who is sitting in the audience here, who got me into the way of working on TIM3 and T-cell exhaustion. His initial paper was done with him on LCMV. A lot of genomics work was done with Aviv Rego, and Arlene Sharp made most of the mice that I'm going to talk about. Rick Blumberg and Amit Gandhi for the crystallizing TIM3. And David Hafler and Tomo for most of the human work that I'm going to describe today, and Ray Sobel for pathology. So my talk, just to take you along, this is the outline for the talk. My talk has 3 parts. And the first one is actually how we discovered TIM3. As you all know, T cells, when they come in contact with antigen, they activate, expand, and differentiate. They acquire different effector phenotypes. Tim Mossman and Bob Kaufman first described Th1 and Th2 cells. And Th1 cells are the ones that produce interferon gamma and lymphotoxin, IL-2. And they are critical for clearing intracellular pathogens. And we also thought they were the critical cell size for autoimmunity. And Th2 cells make IL-4, IL-5, and IL-13. And they are critical for clearing extracellular pathogens like parasites. And they also mediate allergy, asthma, atopy. Since we were working in autoimmunity, and we wanted to actually identify a cell surface molecule by which we can pluck these cells from the tissues and identify their functions. And in fact, that's exactly what we did. We took Th1 cells, the Th1 clones. We immunized rats and generated 20,000 monoclonal different antibodies. And for 6 months, a student and a postdoc, not the robots, worked on this and identified 4 different monoclonal antibodies. And 2 of these monoclonal antibodies were specially staining cell surface on Th1 cells and CD8 T cells and did not stain Th2 cells. And these are the antibodies. You can see they're staining all the Th1 cells, CD8 T cells, but not Th2 cells. And we went ahead together with Gordon Freeman and cloned the gene. It was a new and novel gene. It had a signal peptide, IgV domain, mucin domain, and a long tail. And long tail has 6 tyrosines in it. There are actually 69% homology between mouse and human gene. And it's actually at a very critical place in the genome. It's in 5q33 in humans. And it's on the centromeric end of chromosome 11. There's a whole family of TIM family of genes and that we have identified, and they've been associated with many immune-mediated diseases. This is actually showing the structure is that it has an IgV domain, there's a mucin stalk, and the tyrosine tail, which has got 6 tyrosines in it. So TIM3 got a second lease of life, and when Anna Anderson was working in the lab, and in fact, we had identified TIM3 is an inhibitory molecule. Its ligand is galectin-9. When galectin-9 binds TIM3, it clumps the cells together, and the cells eventually die. And if you're looking under a confocal microscope and looking at these clumps, in fact, it's almost like 4th of July or Diwali for here, is that you can see that these cells just bursting. At the end of 4 hours, you have these clutter of dead and debris sitting on the microscope. So it is an inhibitory molecule. It is expressed on interferon gamma producing T cells, and its ligand is galectin-9, but there are other ligands that have been identified as well. So when Anna Anderson was trying to be independent in the lab, and I asked her actually, why don't you work on TIM3? It may be the CTLA-4 of Th1 cells or TC1 cells. And as you know, when you transplant a tumor progressively grows, it gets infiltrated with CD4 and CD8 T cells, but they can't get rid of the tumor. And if you look at what happens to CD8 T cells, the CD8 T cells inside the tumor will progressively acquire different checkpoint molecules or inhibitory receptors. And shown here is that you can have some cells that have neither TIM3 or PD1 on the cell surface. And if they acquire PD1, they will lose some of the functions. And then when you acquire both TIM3 and PD1 together, they become dysfunctional and terminally exhausted. And in fact, we used to call them co-inhibitory molecules. And in people in the field, in cancer, call them checkpoint molecules. And in fact, there is not one checkpoint molecule that has been identified. Now there's a plethora of at least 10 different checkpoint molecules that have been identified. T cells, in order to get activated, they need 2 different signals. Signal 1 by T cell receptor, and signal 2 is provided by all the co-stimulatory molecules, and CD28 is the classical molecule. As the cells get activated, they will upregulate all these inhibitory molecules. CTLA-4 was the first one to be discovered. And then you can see all of these molecules come up in order to shrink the expanded T cell repertoire, and T cells become dysfunctional. And one of the classical experiments that Anna did was that when you transplant a tumor, and in fact, the syngeneic tumors will progressively grow. And if you do anti-PD1 or PD-L1, and sometimes it works in this tumor, this is a melanoma tumor, sometimes it doesn't work, but if you co-block the 2 checkpoint molecules, PD1 with TIM3 giving anti-PD1 and anti-TIM3, in fact, tumors melt away and develop anti-tumor immunity. In fact, there are 11 different clinical trials right now for solid tumors with PD1, and anti-TIM3, and you can go to NIH Gao, and there is all the clinical trials that are there on solid tumors. But there's something interesting that I want to introduce, and I think Rafi Ahmed is going to talk about this tomorrow a little more. And one of the interesting features of this checkpoint molecule, TIM3, is that there's an antagonism between TIM3 expression and TCF1. And TCF1 is one of the molecules that Rafi identified, and it is a molecule that forms the stem-like T cells. These are the cells that respond to checkpoint blockade anti-PD-1 therapy. And there is a perfect antagonism by expression of TIM3, and whenever TIM3 comes up and the TCF7 is extinguished. And in fact, I'm sure Rafi is going to talk about this in a little more detail tomorrow. And I'm just showing you one slide here, is that when the cells do not have any checkpoint molecules or have a little bit of PD-1 expression, they express TCF1. They have stem-like cells. They are stem-like cells. They are the resource cells that respond to anti-PD-1 blockade. But when they upregulate TIM3, they get terminally exhausted and become dysfunctional. And this way of actively working up how this antagonism happens in immune cells. And this antagonism is not only in T cells, but also in DCs. So once the TIM3 comes up, these TIM3 expressing T cells do not respond to anti-PD-1 blockade. So as we were working on this, then we got together with Anna Anderson and Aviv Regev, and we started asking a series of questions. And the questions were very simple, and we wanted to know on which cells are these checkpoint molecules expressed, and which molecules, if any, induce the expression of these checkpoint molecules in tumors. And once they interact, who are the partners? What are the cell types in the tumor microenvironment that are talking to each other? And once they interact with each other, what is the effect of this interaction on both the T cell and on the responding ligand-expressing cells? And we wanted to actually build this whole pathway by doing single-cell RNA-seq, and we wanted to build this whole pathway by doing spatial transcriptomics. Let me quickly go through the first one. The question is, what molecules are actually involved, especially in tumor microenvironment, in upregulating the expression of these checkpoint molecule, PD-1, TIM3, LAG3, TIGIT. And we identified by single-cell RNA-seq, there was one molecule, a cytokine called IL-27. It's a suppressive cytokine that's induced during resolution of inflammation. And we found out that this molecule, IL-27, is really very active in inducing TIM3 expression. And we did a whole series of experiments, and we built these whole networks and chain networks to identify IL-27 doesn't only induce TIM3, but induces PD-1, CTLA-4, TIM3, LAG3, and TIGIT. In fact, PD-1, TIM3, LAG3, and TIGIT, they all come together as a module, and the correlation coefficient of these four molecules is 10 to minus 23. And that basically tells you that it doesn't happen by chance. If you block one molecule, that's not going to be sufficient to induce anti-tumor immunity. We also build these networks, the transcriptional networks, to identify the transcription factors. We identify two transcription factors, is CMAF and PRDM1 are the two transcription factors that induce these whole module of checkpoint molecules. Let me finish the first part of the talk. What I told you is that we identified TIM3 as a molecule that's differentially expressed on Th1 and TC1 cells. And it led to identification of this whole family of genes called TIM family of genes. There are 8 members in mice, there are 3 in humans, and they have been genetically linked to many of the immune-mediated diseases and allergic diseases. And in fact, I showed you some of the data. The upregulation of TIM3 makes T cells terminally exhausted, and PD-1 TIM3 together will synergize to induce T cell exhaustion. And anti-PD-1 and anti-TIM3, in fact, synergize to induce anti-tumor immunity. And there's this whole module of co-inhibitory receptors that's expressed on exhausted T cells. As they acquire more of the inhibitory receptors, more exhausted cells get. And in fact, at the end, there's an expression of TIM3, which antagonize expression of TCF7. And I showed you some of the data that the PRDM1 and CMAF are the 2 transcription factors that induce the expression of this whole module of checkpoint molecules, because PRDM1 and CMAF will bind to both promoter and enhancer elements in the checkpoint molecule. PD1, TIM3, LAG3, TIGIT, all of them come together. I'm going to go to the second part of the talk very quickly. And in fact, I'm going to make a point that I told you that the checkpoint molecules come up on CD8 T cells. Their major job is to shrink the repertoire, and that's how they work. And that is a lie. And I'm going to tell you is that if you detail analysis, is that we will identify what cell types they really work on. So Karen Dixon in the lab, in fact, did a single-cell RNA-seq to identify where TIM3 is expressed, and she took out these single cells from draining lymph node, non-draining lymph nodes, and from the tumor. She annotated them. These are B cells. These are DC2s and DC1s, the myeloid cells, and these are your T cells, or TILs, T-cell infiltrating lymphocytes. And if you really look at TIM3 expression, it's largely restricted in tumor microenvironment to the DC1s and to myeloid cells. And this is the expression you see in T cells. So I've been talking all along about T cells, but in the tumor microenvironment, it's largely expressed on the DCs and myeloid cells. And this is the protein expression of it. And we made a floxed allele of TIM3 to see which cell type does TIM3 really work on. And we crossed it onto all different Cree drivers. In fact, there was a one flox allele being crossed to all these Cree drivers, and a whole mouse house was breeding for just one flox allele. And in fact, the flox allele is faithful that if you go CD11C Cree, you delete it in DCs, not on T cells. If you delete it in CD4 Cree, they delete on CD4 T cells and CD8 T cells and not on DCs. So now we had the mice to ask the question, which cell type is TIM3 as a checkpoint molecule most important? And if you cross it to CD4 Cree, which deletes it on CD4 T cells, CD8 T cells, and Tregs, and if you really look at the inhibition of tumor growth inhibition, there is not much of a tumor growth inhibition. In fact, no journal will take your paper if you show that data. If you delete it from CD8 T cells, this is where the surprise was, absolutely nothing happened. If you delete it in FOXP3 ART2 Cree, in Tregs, absolutely nothing happens. This is one of those situations when your postdoc comes to your office, is about to commit harakiri. And your job is to see how you can get the postdoc out of your office and can do further experiments. And then I asked Karen, I said, didn't you say that it was largely expressed on myeloid cell and DCs? And she crossed it onto the CD11C Cree, transplanted the tumor, and you can see massive growth inhibition, tumor growth inhibition. And in fact, I said CD11C is also expressed on some T cells. Maybe it's a specialized T cells. She used ZBGB46 Cree, which is a much more specialized DC Cree, and you can see the massive tumor growth inhibition. And if you really look at it, when you did, and since the data is published, I'm not going to go into the details of it, and you do single-cell RNA-seq, you have deleted TIM3 on DCs, you get massive tumor growth inhibition. There was no effect when you delete it on CD8 T cells. There was no effect when you delete it on Tregs. There was a little bit of effect when you deleted all T cells, but the massive effects was on DCs. And when you delete it on DCs, and one of the major things that happens to DC is that if you look at this single-cell RNA-seq data sets, there is a massive activation of all the components of inflammasome pathway. I'm not going to go to the details of it. And TIM3 is an inhibitory molecule, also on DCs, where it inhibits the activation of inflammasome. And in fact, here's a score. You can see inside the tumor, when you delete TIM3 from DC, there's a massive increase in the inflammasome activation. And if you look at the protein expression, is the TIM3 deletion on DCs will upregulate IL-1, IL-18, but there is no difference in the TLR stimulation, IL-12, and other cytokines induced by TLR. So in the second part of the talk, I started out by saying the checkpoint molecules came, we identified them on T cells. They inhibit T cell activation, expansion, differentiation, and they are there to shrink the repertoire after it has expanded. But do all checkpoint molecules only work on T cells? And I also showed you the IL-27 is a cytokine that induces in T cells. But in second part of talk, when you delete these checkpoint molecule systematically in different cell types, and what I gave you data is that it's expressed. In fact, the DCs has the best function for TIM3 at least. And the reason it works is that the TIM3 on DCs is really curbing or inhibiting the DCs from getting activated, and activation of inflammasome is inhibited by TIM3. So let me conclude the second part of the talk. TIM3 is prominently expressed on myeloid cells and DCs in tumor microenvironment, and conditional deletion of DCs gives you most effective anti-tumor immunity. And in fact, TIM3 inhibits the inflammasome activation on DCs. And if you give anti-IL-1 and IL-18 antibody, you can abrogate the protective effect that you observe in TIM3 knockout mouse. Let me go to the last part of the talk, which is actually a little different. I'm going to talk about that we always think of the checkpoint molecules on CD8 T cells, and they are communicating with antigen-presenting cells and sometimes on tumor cells. But which cell is the most important and where the effects are most? So the next part of talk is on TIGIT. And when the interaction happens, what are the functional consequences when the cells come in contact with each other? And I showed you this slide. For a T cell to get activated, you need two signals. Signal 1 is given by T cell receptor, gives a specificity. Signal 2 is by these activating co-stimulatory signals, and then they upregulate these inhibitory receptors, and these whole inhibitory receptors come together to give you T cell dysfunction or T cell exhaustion, a term coined in chronic viral infections. What I'm going to talk about to make a case for a checkpoint molecule called TIGIT, which we were working in the lab already. It was an orphan gene called VSTIM3, and we had already knocked it out when people in Genentech actually called it TIGIT. So just for the students in the audience, is that you know that antigen-presenting cells have B7-1 and B7-2, and the receptors on T cells, CD28, the co-stimulatory signal, which cross-links these B7-1 and B7-2 to activate T cells. Then the inhibitory receptor comes up, and they have 10 times higher affinity for the inhibitory receptors, and they take and co-opt the inhibitories, the ligands, and inhibit the T cell activation. In fact, TIGIT pathway is something very similar. And there are two ligands for TIGIT pathway, CD155 and CD112, and they activate T cell by CD226. When the T cell is activated, they upregulate TIGIT, and it co-opts all these inhibitory ligands from antigen-presenting cells and turn the T cell off. It's a system very similar to CD28 CTLA-4 system that has evolved. And TIGIT is a bona fide inhibitory receptor. It has got two ITIM motifs. And we made a complete knockout mouse of TIGIT. And we transplanted the tumor on it. And you can immediately see that when there's no cell that has TIGIT on it, you can see there's a massive tumor growth inhibition. In fact, TIGIT, if anything, is inhibiting immune responses in the mouse. The question is, where does it act the most? And having learned from the TIM3 debacle, so I asked Lloyd Board, just go ahead and do a single cell RNA-seq to identify where is TIGIT expressed in tumor microenvironment. And he did both in mouse, and this is the data. In fact, I will not go to the details of it. Safe to say that TIGIT is expressed on CD8 T cells, NK cells, but the most expression is between NK cells and FOXP3 positive regulatory T cells. It's not only in mouse. If you look at and mine the human data from our data sets at the Broad, and immediately same thing is true. TIGIT is expressed in tumor microenvironment on CD4 T cells, CD8 T cells, NK cells. The most expression is on Tregs. We went ahead and have made a floxed allele of TIGIT, and again, cross it onto every Cree driver that you could think of. And we know that if you cross it onto E8I Cree, and it can faithfully delete in CD8 T cells and does not delete in CD4 T cells. And if you do CD11C Cree, it would delete on DCs, but not on CD4 T cells. So we went ahead and started doing the experiment. And in fact, when you delete it on myeloid cells using lysum-Cre and transplant the tumor, and absolutely nothing happens. In fact, they look identical. If you delete it on DCs using CD11C Cre, which worked in the TIM3, it does not work for TIGIT. If you delete it on B cells, little something happens, nothing you can write home about. Then you delete it on all T cells by CD4 Cre, there is slight tumor growth inhibition, and we thought it was because of CD8 T cells. You delete TIGIT on CD8 T cells, absolutely nothing happens. And it's the FOXP3 positive T cells where TIGIT plays the most important role. In fact, this is shown here, is that if you took the wild type versus FOXP3 knock-in, FOXP3 deleted TIGIT mice, and you get, in fact, massive tumor growth inhibition. I told you its ligand is CD155. It's a high affinity ligand. And we went ahead and tried to look at where is the ligand expressed in tumor microenvironment, and the majority of the ligand is expressed on tumor cells themselves. And these are all the tumor lines we have in our facility, and you can see MC38, B16, F10, all of these mice, all of these cell lines will express very high levels of the ligand for TIGIT. And this is what is expressed on antigen-presenting cells, the myeloid cells and DCs, and this is the non-hematopoietic cells. In fact, if you look at mouse or human tumor cells, they heavily express the ligand for TIGIT. We have not seen a single cell line or a normal tumor where TIGIT ligand CD155 is not expressed. We made a deletion mutant of TIGIT. We did two of those. We either took CD155 off from the tumor cell, or we deleted the tail of CD155. And this is shown here that we can, in fact, delete it completely from the tumor cells, both in vitro and in ex vivo. And now we have perfect tools where we have TIGIT expressed on Tregs or CD155 expressed on tumor cells. And we can delete it either on tumor cells or we can delete it on Tregs. Shown here is that we can, this is the wild type mouse, that the receptor is on Tregs and the ligand is on tumor cells. You have receptor deleted, TIGIT is deleted, but the tumor cells express the ligand. Here, the receptor is expressed on the T cells, Tregs, the ligand is missing, or you have both of the ligand and receptor missing. So it gives you a perfect scenario is to see what does this interaction between TIGIT expressed on Tregs doing to CD155 expressed on tumor cells. We transplanted tumor, did a massive single cell RNA-seq, something like 84,000 single cells where both the tumor cells were analyzed and the T cells were analyzed. And I'm going to cut this long story short. And then we looked at the expression of what are the genes that are being regulated by this interaction of TIGIT on Tregs and CD155, a high affinity ligand on tumor cells. What is the outcome? And this is the scenario. This is why I'm only giving you data of the tumor cells. And you can see that if you have both receptor and ligand present, there's a whole set of genes that are upregulated, and there are a whole set of genes downregulated. And if you look at the genes that are upregulated by the interaction of TIGIT with its ligand expressed on tumor cells, we were first surprised and shocked that this is a fruitful interaction, that the Tregs have co-opted the pathway, that by interacting directly with CD155, it upregulates oncogenes, and it upregulates HES1, PARP10, BRAF, and NRAS, the oncogenes that are functionally making the tumor cells grow, proliferate, and evade immune system. And in fact, what you have is a unique, innate function.
Vijay, your time is up.
2 minutes. All right. So what I told you is the interaction between, you did take my 10 minutes. I started at 9:10. All right. So what did I tell you is that the TIGIT expressed on Tregs is the most functional molecule, not on CD8 T cells, not on DCs. It interacts with CD155 on tumor cells, and it upregulates these oncogenes, And this is an innate function of regulatory T cells to keep the tumor alive, proliferating, and evading the immune system. It has nothing to do with inhibiting the immune system. What is the outcome? Is this interaction promotes the Treg stability, and on the other side, it actually promotes the tumor growth and evasion. So let me sum up just for you. Okay, TIGIT is a part of checkpoint module expressed on exhausted T cells, but it's the most functional is on FOXP3 positive Tregs. When you delete it on Tregs, you see that you can induce tumor growth inhibition. CD155 is maximally expressed not on engine-presenting cells, it's there, but it's maximally expressed on tumor cells themselves. And this interaction between the TIGIT on Tregs and CD155 on tumor cells makes a direct interaction. And we have a whole series of experiments that will come out in the paper that you can overexpress, in fact, these oncogenes in these CD155 knockout tumor cells, and they will again grow. And just what did I tell you? Just to get this cartoon together, is that I told you that there are not only CD4 or CD8 T cells that express checkpoint molecules, there are a multitude of T cells in tumor microenvironment that express these checkpoint molecules. I gave you an example of IL-27, one of the cytokines, a suppressive cytokines, that comes during resolution of inflammation that's inducing a whole lot of checkpoint molecules in mice. In humans, is type 1 interferons. And this interaction is, in fact, affecting both ways, a bidirectional interaction. I gave you an example of what TIGIT expression on Tregs induces and enhances the function of the tumor cells, inducing oncogenes, and they forms a nidus for tumor growth. This is my lab. Our lab, we call ourselves Kuchru Lions. We have a soccer team, and we have lost every game we have played. And finally, before COVID, we did win one game, and this is the losing captain. If you are a soccer player or interested in do good science, please come and visit my lab. I thank you for your attention. I'm happy to take any questions you may have.
Thank you very much. Wonderful. I'm sure there are many hands which are being raised. We can allow you 3 questions because you already showed up. So maybe 4.
You have to be fair.
But
look at the time. All right, guys. Okay, let's go.
So we go ahead with yes. You.
Oh, you.
Yeah. Can somebody give the marks? And Dr. Mehra then, and Javed, and maybe Samishna. There are many hands. All of you must be ready to ask questions, but he'll be around, so you should interact with them.
Thank you very much for this very interesting talk. I'm just wondering about the CD155 molecule. In what stage of the tumor is this expressed? Is this from the beginning there, or is this some sort of evasion of the tumor developing this so that the tumor can escape?
So is the question at what stage CD155 is functional?
Yeah, expressed and functional. So that this.
So CD155 is constitutively expressed on all tumor cells. It goes up a little more with interferon gamma, and in engine-presenting cells, it's all entirely dependent on TNF and interferon gamma. So this is a tissue evasion mechanism. It may be a normal mechanism for maintaining homeostasis, and during development between Tregs, how they maintain the stem-like cells in the system.
So my question is, since there are array of inhibitory molecules expressed on the cells as well as secreted out, but it's quite intriguing that blocking TIM3, it brings about radical changes in the function of T cells or dendritic cells.
Yeah.
So how come other inhibitory molecules, they stop functioning? Or is it that if you block interaction with TIM3, these molecules get neutralized?
Is the question that you have a series of checkpoint molecules, what is unique about TIM3 or other checkpoint molecules? See, I mean, I think this is what we have done. I think we have glossed over a lot of detail. And we think, yeah, checkpoint molecules, if you grossly look at them, their main job is to contract the effector T cell pool. Once they come up, but that's not the only function they have. So this is the reason I actually was trying to make an example that each of these checkpoint molecules have specialized function, not only on T cells, but antigen-presenting cells in tumor growth. And maybe we're missing a whole lot of biology, but entirely fixating on one cell type. And each one of them, like I gave you example, TIM3 and TCF7, this antagonism, is so important that you can't, the TIM3 comes up, the cell cannot respond to PD-1 blockade. So it is doing something specialized in every cell type. Okay, that's we can talk a little more.
Yeah,
about
Dr. Mehra, please.
Vijay, thank you very much for this wonderful talk. You have convinced us that in addition to those noble two checkpoint inhibitors, the TIM3 is also very important. But you also showed that the TIM3 is expressed not only on the T cells, but other cell types, the myeloid cells and the dendritic cells. Has any studies been done to look at the quantitative expression? Is the quantitative expression more on T cells rather than the myeloid cells?
Yeah. So in fact, if you quantitate the expression of TIM3, the maximal expression in the body is in the microglial cells, not on T cells. T cells are the weakest. And then the next one is the myeloid cells. The next one is DCs. And the maximal expression is microglial cells. And each cell type, they have an important function. And we have actually a paper coming out in how it regulates microglial cells phagocytosis and in fact, Alzheimer's pathology. And it's a GWAS gene. In Alzheimer's disease, in the recent GWAS study, is one of the genes that are genetically linked to development of late onset Alzheimer's disease. So by looking at the expression of different cell types, you can actually begin to uncover novel biology that we have missed.
This could be remarkable for dementia. I think this has to be followed, the microglial cells.
Yeah. Vijay, following up a little bit on that concept here.
Yeah, I see it.
So
I see it. Thank you.
TIM3, you showed beautiful induction in a particular subset of T cells, right? From the early studies. You showed wonderful expression on dendritic cells. Is there a congruency in terms of the signaling pathways that induce the expression on the dendritic cells? And is there a function more in a type 1 setting on dendritic cells versus a type 2 setting? Because that might allow us to start to categorize these inhibitory receptors in different types of immune responses.
So I told you, and actually this is a beautiful question. And in fact, I wasn't ready to give that data. So TIM3 on T cells inhibits expression of TCF7 and inhibits the generation of the stem-like T cells. TIM3 expression on DCs inhibits the factors that induce stem-like T cells. So you can see the whole biology is so beautiful. The whole biology is going the same direction. That if on T cells, it's inhibiting the induction of stem-like T cells, on DCs, it is inhibiting DCs to produce the factors that are promoting the stem-like T cells. In fact, I showed some of the data to Rafi the other day, is that the DCs produce these factors and form these niches, separate from the effector T cells, where all the stem-like T cells proliferate and grow. And so everything's going in the same direction. I'm going to stop. I think, thank you very much. Thank you for
we have one more, Soman.
Yeah, fantastic talk. I just have one quick question. This is regarding IL-27 galactin-9. And did you find their expression levels or abundance in changing in the tumor or in the draining nodes?
With IL-27?
IL-27 and
so IL-27 is a double.
Ligands of TIM3, I mean, basically.
So IL-27 is a double-edged sword. On one hand, it is inducing the expression of all these checkpoint molecules, right, together with type 1 interferons. On the other hand, it's inducing IL-21 to promote CD8 T cell activation and expansion. And there was a drug company that actually was going to target IL-27 for anti-tumor immunity. And in fact, I told them this is not a great idea. And they threw me off the board. So it is, I'm not sure whether IL-27 would be the best molecule to inhibit. Okay, thank you very much.
So sorry to stop you here because I hate to. Such kind of lectures should have more discussion time than their speaking time. So anyways, it was great to listen to you, and maybe my co-chair will like to finalize.
Thank you, Vijay, for a wonderful talk. In half an hour, you discussed 4 Nature paper, and the 5th one perhaps going to come soon, I guess. Thank you so much.
Thank you, Dipankar, for giving me opportunity to chair this session. Thank you, everyone.
I would like to invite the chairpersons and the speaker to get on the stage for a picture. Moving ahead, we have another fantastic plenary lecture by Professor Wilfried Ellmeyer, a professor of immunobiology and head of Institute of Immunology at the Medical University of Vienna, Austria. I'd like to invite Professor Javed Agarwala from IIT Ropa to share the session and introduce our next speaker.
So we welcome Professor Wilfried Ellmeyer. He works at the Department of Immunobiology and head the Institute of Immunology at Medical University of Vienna. He is the chair of a scientific program committee for IUIS 2025 Congress in Vienna, and he will serve as the president of Austrian Society of Allergy and Immunology for a period of 2025-2026. So I invite you, Professor Ellmeyer, for your talk.
So, good morning. Thank you very much. I would like to thank really Dipankar and his whole team for the great and honorable invitation to come to India. It's my first time here. It's a great country, great people, great science, great food. So I'm really enjoying my stay. It's wonderful. And this morning I would like to tell you a little bit about histone deacetylases and how they control CD4 T cell mediated immunity. Just looking for the pointer. Just 1 second. Okay. So my laboratory actually comes from the field of T cell development, and for a long time we were interested to understand how CD4 and CD8 T cells develop. But I think I don't have to convince the audience here that T cells come in much more flavors, both CD4 lineages and CD8 lineages. There are a variety, as you also heard in the previous presentation, there are a variety of different T cell lineages, and my laboratory is interested to understand how these various subsets are regulated. So how are cell fate decisions and lineage decisions controlled during effector differentiation, and also actually how this knowledge can then be used for translational approaches to develop novel therapeutic strategies. We have interest in understanding the function of certain molecules. Some of them are shown here. And with respect to technology, we are embedded in a strong collaborative environment, and we are working both on mouse and more recently also on human cells. We're trying to use a variety of different mouse models for autoimmune diseases, but also for infection. I think you're all aware that, and know for sure that cell fate decision and differentiation processes are linked with lineage-specific gene expression programs, as kind of indicated here. And these lineage-specific gene expression programs are regulated on one hand by master transcription factors, but on top of it, they're also regulated by epigenetic processes. That means via chromatin regulation. So, and during these differentiation processes, you have lineage specification, commitment, as well as regulation of lineage plasticity. And You all know DNA is not a naked molecule. It's wrapped around histone octamers. They can form higher structures. And depending on accessibility of DNA, it's decided whether a gene is expressed or gene programs are expressed or not. And this is epigenetic gene regulation. And among the epigenetic regulatory processes, reversible histone acetylation of lysine residues on histones is arguably one of the best characterized modifications. They are mediated by two opposing gene families, the histone acetyltransferases, which are considered as transcriptional co-activators, and they are part of chromatin remodeling complexes, while histone deacetylases, they're considered as erasers and associated with transcriptional co-repression. They're part of co-repressor complexes. So regulation, epigenetic regulation via transcriptional changes is regulated via chromatin-mediated effects. However, if one works about histone deacetylases or thinks about reversible acetylation, one should not forget that actually there's much more than regulation of chromatin accessibility. And in the last decade, more than 20,000 acetylation sites on more than 8,000 proteins among different species have been identified. And lysine acetylation of non-histone targets affect proteins in multiple ways. They can regulate the activity, the intracellular localization, the ability to interact with other proteins. So lysine acetylation is likely to be comparable to other post-translational modifications, for instance, protein phosphorylation. So one always has to keep in mind if one studies HDACs or reversible lysine acetylation in general. So the mammalian HDAC family is kind of a complex family. It's made up of 18 different members. They can be subdivided in 4 classes, also dependent on the homology to yeast enzymes. And my laboratory, for several reasons, I will touch them briefly later on, is particularly interested in class 1 HDACs, HDAC1 and HDAC2. What's also very important related to HDAC function is that there are already HDAC inhibitors in the clinic. So pan-HDAC inhibitors, so which target HDACs in general, are used for certain type of cancer, cutaneous T-cell lymphoma, peripheral T-cell lymphoma, multiple myeloma, and also for other disorders such as bipolar disorders or epilepsy. It has also been shown in many preclinical mouse models that pan-HDAC inhibitors affect T cells and T cell mediated diseases in many ways. First of all, they can induce regulatory T cells. This has been shown. And in addition, what has also been shown, they can attenuate many disease scores in a variety of different preclinical mouse models, indicating a high potential of targeting HDACs for T cell mediated diseases. However, HDAC inhibitors, pan-HDAC inhibitors have side effects, and therefore potential therapeutic benefits may not outweigh the risk of using these inhibitors. So therefore, kind of the hypothesis in the field is that selective HDAC inhibition or targeting HDAC-specific pathways and targets might be a promising treatment strategy for T-cell mediated diseases. And that HDAC function in T cells is important. Many studies by us, but also by many other people, have really shown or have been performed, in particular with genetic mouse models, that really have been shown that HDACs play a variety of different roles, both during T-cell development, but also in the differentiation of peripheral function of peripheral effector T cells. My laboratory contributed some of these studies with respect to HDAC1, and today actually I would like to introduce you a little bit into the function of HDAC1 with respect to autoimmune diseases. So actually, this really indicates that HDACs are key regulators of T helper cells. So we are interested in HDAC1 and also in HDAC2, coming actually from the T-cell developmental field, because we have characterized some molecules which play important roles during T-cell development, and we found that they are interacted with HDAC1. So therefore we thought, well, probably HDAC1 is an important target regulating T-cell development. So we continued our studies. We started to collaborate with Christian Seiser's group in Vienna because he had generated conditional HDAC1, HDAC2 alleles, and therefore we tried to explore the function of HDAC1 and also in part HDAC2 in more detail. So they're highly homologous, they can interact with each other, and interestingly, HDAC1 is upregulated in the absence of HDAC2 and vice versa, so there are some compensatory effects. And questions that we have been addressing during the last couple of years is, does HDAC1 regulate T-cell development? What is the impact actually if you delete HDAC1 on the variety of different T helper subsets? Does it control autoimmunity? Has it a positive or negative effect on disease score? On disease scores? Are there unit functions for HDAC1 in comparison to HDAC2, because why do we have actually so many different HDAC members expressed in T cells? And all the 18 members I showed you before are expressed in T cells. And of course, it's also an important question, why is this happening? And also, reveal non-histone targets in T cells. So in order to address this question, as I mentioned already, we are using conditional mice. So we have T cell specific knockouts of HDAC1 or HDAC2. So it's novel mouse models. And we also work on human CD4 T cells and apply HDAC inhibitors and ask various questions. Okay, but today I'm going to tell you about the role of HDAC1 in T cell mediated autoimmunity. So I think I don't have to really introduce probably in this audience, and also Vijay Kutru is here. He developed this model and showed it with further detail. So actually we have been addressing the role of HDAC1 in experimental autoimmune encephalomyelitis. So why did we do that? Because first of all, it's a T-cell mediated inflammatory disease of the CNS. It's used as a model of multiple sclerosis. Also, I think there are really also important differences between. So it's also kind of a neuroinflammatory diseases. And what's important, it's driven by Th1 and Th17 cells, in particular by certain pathogenic subsets. So indicating that this is an ideal model system to address the role of certain molecules in T helper subsets. I think this is just a scheme where actually if you induce the disease, I'll show you briefly in a second how this is done, then actually you get activation of CD4 T cells, you have a leakage in the blood-brain barrier, then you have entry of T cells and other cells into the CNS, and then actually you get demyelation in mice. And it has been shown that HDAC inhibitors ameliorate clinical scores of EAE. So we raised the hypothesis that HDAC inhibitors in the EME model also target HDAC1 function in T cells. In order to test that, actually a talented PhD student, she's long time finished now, actually Lisa Göschel started to address this question by inducing EAE in the conditional knockouts we'd had. So this is kind of the scheme what you're doing. So we have either wild type or mutant mice, you inject MOC peptide, so myelin oligodendrocyte glycoprotein, you're in CFA, pertussis toxic, and then actually after a certain time you can actually measure a variety of disease scores with respect to how the mouse behaves. There's some tail limbing, and also whether it's the hind leg or forelegs are paralyzed. So if you do that in wild type mice, actually what you can see upon day 10, actually clinical course start, then you had diseases peaks and then it goes slowly down. And this is also accompanied by weight loss. So this is the situation in the wild type mice. But if you do that in the knockout mice, I hope you can appreciate the red line. So the mice don't get sick, they stay healthy. And this is also shown with the weight loss. And if you look in the CNS and do some Luxol blue stainings or pus stainings, then you can see actually in comparison to wild type mice where you see a strong destruction of the myelin sheets, actually the knockout mice look very healthy in this situation. And this is also shown when you look for infiltrating CD3 positive T cells, which can be easily found in the wild type situation, but none in the knockout. Interestingly, what we also did, we did many experiments to analyze this further. I just would like to, for time reason, also show you one. Actually, what we have, even if you deplete regulatory T cells, which in the wild type situation lead to a more severe clinical score, there was no effect. So HDAC1 knockout mice, so HDAC1 missing in T cells, the mice didn't care whether regulatory T cells are there or not. They were still protected against EAE. So kind of the first conclusion of this presentation. So HDAC1 seems to have a novel pathophysiological role in EAE. T-cell specific loss protects mice from EAE. And in addition, we also observe differences in certain type of chemokine receptors in activated HDAC1 deficient T cells. So of course, this kind of is a very intriguing finding, but of course we raise the question then why are the mice with the T-cell specific deletion of HDAC1 are protected from EAE? So what are kind of the cellular and also molecular mechanisms? And then another PhD student joined the laboratory, Patricia Hamminger, and she tried to address that in more detail. And just if you think conceptually, so during the initiation of an immune response and also the development of autoimmune disease, what are the events that are happening? I mean, first of all, in the lymph node, you have CD4 T cell activation. So essentially T cell repertoire, APC, T cell interaction could be altered in the absence of HDAC1. Then of course you have cell proliferation. Then there's T cell migration into the tissue. The cells have to infiltrate into the CNS, and also they have to fulfill the effector function. And essentially a combination a single defect or a combination of all the defects can occur. So we started to initially address this by looking in terms of these early steps, and actually for all of that was normal. So there was no difference in the early activation stage of T cells lacking HDAC1. And therefore we decided, before we go really stepwise to test all these effects, maybe we should do a kind of a sequence on RNA sequencing experiments to really see what is actually happening to the T cells during the activation of the T cells. So what Patricia did, so she used a 2D2 transgenic mouse model. So the 2D2 is a T cell receptor transgenic, which recognizes specifically the MOC peptide. So essentially, she isolated wild type and naive T cells, transferred into syngeneic CD45.1 mice, immunized the mice, and then actually on day 4, so before the cells actually start to leave the lymph node, she isolated the cells and did the bulk RNA sequencing at the time experiments to really see what are the difference between activated wild type and HDAC1 deficient CD4 T cells. Of course, you get many genes up and down regulated, so that is not surprising if you do that. But when she did the pathway analysis, interestingly, what she observed, actually, she saw that genes associated with leukocyte extravasation signaling, so migration of T cells, are downregulated in the absence of HDAC1, as indicated here. I think I don't have to show you this classical, or really explain you in detail this classical textbook style. So what's happening actually when immune cells are migrating, so they migrate in the vessels, and then actually in order to transmigrate into the tissues, they have to slow down, so to roll, then they get activated, they rest, there's some kind of crawling of the cells, and then they do diapedesis into the tissue. And all these different steps are regulated by adhesion molecule, by integrin molecules, by chemokine molecules. And actually, when we looked in the RNA sequencing data, many of these pathways which are involved at various steps were downregulated in the absence of HDAC1. In addition, we also saw differences in enzymes which are important for glycosylation. So also an adhesion is regulated by modifying glycosylation patterns on the surface of molecules, so this was also impaired, indicating that in the absence of HDAC1, there might be changes in the migration behavior of T cells. So initially, what we did, we started that in vitro, so you can do transfile assays, as indicated here. So you put the cells on top, so we have wild type and knockout together, they were labeled in different colors, and then you put them on top, and then you just count the cells which migrate through. However, surprisingly, at the beginning, what we observed is actually that the HDAC1 deficient cells had even a higher migratory capacity towards the lower chamber. Of course, this is an unnatural situation because in vivo cells don't adhere to plastic. So therefore, we thought, well, maybe we should try to, and then we also teamed up with the group of Britta Engelhardt at the Theodor Koch Institute in Bern, who is an expert in analyzing T cell migration. And actually, what we did then, we repeated the migration assays, however, covered then the transfers with either brain endothelial cells, a cell line, or also with primary mouse brain microvascular endothelial cells. And if you do that, then actually one suddenly observes that HDAC1 deficient cells have a disadvantage and they migrate less through endothelial to brain endothelial cells. And in order to study that in more detail, Patricia went to Brigitte Engelhardt's laboratory in Switzerland and performed some kind of in fluidics or under flow, performed some migration experiments under flow conditions. So the system is shown here. So at the end, in principle, you have wild type or knockout cells. In that case, we differentiated the cells into Th17, and then you have a flow chamber connected to a microscope, and then you can actually apply the cells in the flow, and then you can measure how many cells adhere and how many cells migrate. And by doing that, so this is just a movie showing how this is functioning. What you can see is the cells migrate and then they adhere. And then actually at some point you stop. Then you can count the number of different cells. And I think it's already very visible that there are much more cells in the wild type and in the knockout situation. And then Patricia spent hours and days in front of the microscope and counting all these different fields. And what you can see when you do that is actually that the number of cells that are rested is reduced in the absence of HDAC1. And also the cells that are tried to probe, so they try to find and migrate through the endothelial cells, is reduced. However, those cells that manage to adhere, there's no difference in the crawling and also in the diapetesis towards the subsequent through the endothelial cell layer. So indicating that there's reduced adhesion of HDAC1 knockout CD4 T cells in vitro, both under static condition, but also under flow conditions. And it's a kind of a confirmation that there's indeed a kind of a migration defect. We also generated mixed bone marrow chimeric mice. So essentially what you do here, you have more or less wild type, CD45.1, and then wild type or mutant CD45+2 cells. You mix the bone marrow, transfer the cells into irradiated hosts, then you wait 8 weeks, and then you induce ERE. And interestingly, under these conditions, there was a similar disease incidence in wild type knockout chimeras versus wild type chimeras. There was also a similar clinical score. And when you analyze now which cells are in the CNS, so it's a comparison in the blood, there's a kind of an equal representation. If you have wild type chimeras, there's also equal numbers of cells. However, in the wild type knockout chimeras, you see that actually the majority of cells in the CNS are wild type cells, indicating that also in the competitive setting, so knockout cells cannot go into CNS, but there's no other dominant effect on wild type cells which would actually lead to an impairment of ERE. Indicated indeed that one of the cellular reasons why T cells or why mice with the T cell specific deletion of HDAC1 don't develop disease is because of impaired migration of these cells. Okay, so kind of a second conclusion is that T cell specific loss, I mentioned it already before, protects against ERE. HDAC1 regulates CD4 T cell adhesion to brain endothelial cells, and a defect in T cell migration is one of the underlying mechanisms resulting in ERE protection. For time reasons, I cannot show you the data. However, we also observed that mice are protected against adoptive transfer colitis. So this is a model where you transfer CD4 T cells into mice that lack T or B cells. If you do it with wild type cells, mice develop colitis. If you do it with HDAC1 knockout cells, they don't develop colitis. And in collaboration with the group of Michael Bonelli, we have also shown that this T cell specific loss of HDAC1 attenuates collagen-induced arthritis. So this really indicates actually that HDAC1 might be a great target for T cell mediated or to target T cell mediated autoimmunity. But how can we really test this in more detail? Because if you really think about, so ideally you would think about maybe one can apply an HDAC inhibitor, but think how this is actually happening at the molecular level. So in a wild type situation, an HDAC1 is a molecule or HDACs, they cannot bind to DNA itself, so they bind to transcription factors, and by this they're recruited to DNA. So in a wild type situation, you have properly formed complexes. In the knockout situation, several things can happen because HDAC1 protein is not there. So either it's displaced by other members of the HDAC family, or maybe complexes are dissociated. And actually, this might result in a completely different phenotype in comparison if you would apply an HDAC inhibitor. Because if you apply an HDAC inhibitor, it would still be present in the complex. And in order to test that, we teamed up with the group of Christian Seiser again, because so he generated a novel mouse strain where actually he generated mice that express a catalytically inactive HDAC1 molecule. So these are just the details of how I generated the mice. For time reasons, I will not go, but essentially it's a very cool system. But because what he did is he made a knock-in of either a wild type or a catalytically inactive HDAC1 molecule in the ROSA26 locus, combined with a stop cassette. So essentially, we have two strains of mice now. One, it's called HDAC1-on, which has a catalytically wild-type form of HDAC1 in the ROSA26 locus, and an endogenous deletion of HDAC1, or an HDAC1-off mouse strain, which expresses a catalytically inactive HDAC1 from the ROSA26 locus, and also has a deletion of endogenous. So essentially, it's 5 or 6 genetic alteration in one mouse strain, so kind of a complex model. But HDAC-on is the wild-type form, HDAC-off is the form which is catalytically inactive. And then Valentina Stolz and she, Ashley Shu, two postdoctoral fellows, one in Christian's lab and one in my lab, actually tried to analyze this mouse in more detail. So first of all, what you do, of course, you check that the mouse is really working as it should, that the system is working. So HDAC1 is expressed, both the on and the off form at normal levels. So it's also flag tagged and iris GFP. So you can also see that actually all the cells express HDAC1 on and off. And you can also see the stability is not changed, and actually also and it's
5
also
minutes left.
Yes, and also the confirmation that you have a kind of a inactive version of HDAC1. So you really reduce the acetylation activity. And then actually, so expression is normal, also normal T cell homeostasis. And then actually we also could show that there's a normal interaction with different complexes. So we made and more or less have determined the interactome and by mass spectroscopy. So that's all normal. So the question is now, what's the phenotype? So what's the impact of a catalytically inactive version? Then we induce disease again, and interestingly, or not interestingly, excitingly, maybe I should better say, that actually in contrast, while wild type mice got sick again, however, mice expressing a catalytically inactive version, so the HDAC1 mice, they stayed healthy, really indicating that targeting the catalytic activity is sufficient to protect mice from the development of EAE. Of course, this has a big translational implication because HDAC1 is an interesting target for the treatment of autoimmune diseases. There are certain challenges because HDAC1 is the target. At the moment, there are no isoform selective HDAC1 inhibitors available, so they don't exist. It also might be difficult to generate because there's a high homology between HDAC1 and HDAC2. An alternative approach would be maybe to develop or to deliver HDAC inhibitors selectively in T cells. That's also not something which is existing at the moment, really. So the hypothesis to overcome this limitation would be to target HDAC1 targets. And in order to do that, and I would like to show you only one data slide, what we have tried to do. So actually this brings to the more general question, how do HDACs function? So is this more chromatin mediated, or do they really regulate it also beyond chromatin and regulate many, many different non-histone protein? So essentially within a special research program in Vienna, which we have set up, we are trying to address this, and we are mapping currently the acetyl proteome of T helper subsets. So we have set up a workflow where we differentiate T cells in a variety of different T helper lineages, we isolate protein, RNA, and also do attack sequencing from the same samples. And with respect to the proteome, we are determining the proteome, the phosphoproteome, and the acetyl proteome in order to really get a kind of a first impression of establishing an atlas of the acetyl proteome workflow. And I would just like to show you one data slide, kind of exciting. So we have identified more than 2,500 acetylation and phosphorylation sites on proteome wine level. So some of them overlap, some not. We have identified T helper subset specific acetylation patterns as indicated here. And if you start to do network analysis, you see that actually some pathways are regulated both by phosphorylation and some only by acetylation or phosphorylation. So I think with this, we are really heading towards the identification of HDAC1 regulated pathways, and hopefully we will then be able to target HDAC selective pathways, which might be T cell specific, and this might be a better approach to develop novel therapeutic approaches. I would like to thank my team. I mentioned already some of the members who did the work. I would also really like to mention the great collaboration with the whole SFB network. Without them, it would not be possible to perform this kind of comprehensive analysis. And finally, I think you're already very familiar with this slide. I would like to remind you again, but probably it's the last time we show this slide. I hope I can really, together with Rudi, Barbara, and also Ursula, next year we can welcome you in Vienna, and that we can meet again, and that we see many of you in Vienna, and we try to have such a great meeting and a lot of hospitality we are also receiving here. So thank you very much for your attention, and I'm happy to take questions.
Thank you very much on being on time. So it's really interesting and intriguing talk. So it's open for now question answers. Okay, Dipankar.
So, okay. Can I start? Yeah. Very nice talk, sir. Just I wanted to know, so instead of HDAC inhibition, if you use acetate to increase the acetylation level, so do you think you'd get the similar kind of result here?
No, that's an important question, because what I also didn't tell you It depends, because you can also say the other thing, right? I mean, you increase the acetylation. I mean, we didn't do the experiments. To have a short answer, we don't know. We didn't do the experiments, but we're currently doing some experiments also where we have HET activators. So we will try to do any kind of, following your question, the kind of experiments. But we haven't done the experiments, so maybe I have to give a short answer. Yes.
Okay. Thank you.
Yeah, very nice talk. I was just wondering what happens to the HDAC knockout mice if there's an infection?
That's also a very good question, and that we are actually currently doing. And we have done some experiments in collaboration with another group in Vienna, doing LCMV infection, particularly focusing on the CD8 lineage. And there we actually see changes in the subsets related. So we did chronic LCMV infection, and there actually we see changes related to the development of these various exhausted T cell subsets.
Right. No, so actually what I was trying to lead on to was in case of translation, then if you're inhibiting HDAC and you're inhibiting T cell responses, but they might be compromised during with some other infection or so, is that a concern?
No, I think that's a concern with every type of immunotherapy you have. I think it really depends on the type of disease, and again, what we do not know whether the same. I mean, we are just at the beginning. We would like to really also understand the acetyl proteome, finding HDAC1 targets, and then to really also study whether during an autoimmune disease, whether there are similar targets activated or regulated by HDAC, maybe in comparison to an infection. And maybe a dream result would be that you find that certain targets are regulated during infection, certain different targets are regulated during autoimmune disease, and perhaps then one can maybe fine-tune the targets. That would be kind of a dream result. But of
One
course,
short and sweet question from Patil.
Nice presentation, Dr. Wilfred. I'm from Neurosciences Division. So when you spoke about MS, though the condition is 1 in a million cases, 1 in a million individuals, so, but still we do get 2 to 3 cases every day because we have a lot of inflow of patients. My question is, as everybody knows the condition of MS, I will not talk on that. But if you can treat with CCR6 and CCR4 to these individuals, we may expect a good result because as of now, there is, just immunosuppressants are being given. Thank you.
Okay. Yeah, because CCR6, as you probably referred to, I mean, has been shown to also regulate the infiltration of T cells during the initial phase. But in our system, we didn't really follow up whether CCR6 is also important in that respect. We just observed that it's differentially expressed, but we didn't really try to target it or to have antibodies to block it, whether this has then an impact.
HDAC1 cells. So does it affect the migration of antigen specific? Like have you checked these are the MOG specific, or in general there's a migration of TS17 cells? It is an inflammatory response driven, homeostasis driven migration, or antigen specific?
It's a good question. I would assume it's antigen specific in that respect, because you need activation of T cells, but all the migration assays we did with the polyclonal repertoire, just regular T cells isolated from wild type or knockout mice. So whether the T cells have migrate to the CNS because they recognize a specific peptide there, I don't know whether you're referring to this. I don't know, but it's both in a polyclonal setting and in 2D2 transgenic mice. The mice are protected in the absence of HDAC1. So it could still be in the polyclonal repertoire that it's maybe a specificity recognizing MOG peptide, because that's how you induce the disease. But whether other autoantigens would be required, this we do not know because we haven't studied it. Okay.
Professor Amir, for your nice talk. Let us give him a big applause.
Thank you, Professor Elmeyer and Professor Agrawala. Next, we have a 10-minute technical presentation from Premas Life Sciences. Sorry, the presentation is going to be by Olink Proteomics.
Good morning. So first of all, thank you very much for the IMMUNOCON organizers for giving me an opportunity to present about the next generation proteome solution from Olink. Today, I'm going to give you an overview about this technology and also will take you through some of the possibilities to utilize this technology in your research. And finally, we'll give you a classic use case from multiple sclerosis. Olink Proteomics, part of Thermo Fisher Scientific. It started with a humble beginning from Uppsala, Sweden. And in short span of 8 years, Olink has become a global organization serving researchers worldwide. And now we are proud to work with more than 800 prestigious organizations around the world. In India, we have collaborated with our distributor, Premas Life Sciences, to help the researchers with our proteomic solutions. So we have more than 170 labs around the world providing services for our technology. And as of today, we have more than 2,000 publications, which itself speaks to the external scientific validity of our technology. So in this slide, I would like to give you a short overview about the technology and the workflow. The Olink technology, so-called proximity extension assay, it's based upon the dual recognition immunoassay. So the matched pair of antibodies labeled with DNA oligos, when they bind to a protein in a target in a solution, these DNA oligos come to proximity and they are hybridized and extended to form a double-stranded DNA. Following an amplification step, these DNA amplicons are quantified either through microfluidic qPCR instrument called Signature Q100 or through NGS platform, depending upon your research focus and the type of product used. So this innovative technology, coupled with robust internal and external controls, ensure high quality of the assays and able to offer unique features such as high sensitivity, exceptional specificity, broad dynamic range, and all of this with very minimal volume, sample volume. So based upon this technology, we have carefully designed our products to address various research stages, be it full-scale discovery or validation or down to the clinical implementations. For example, Explore products on the left-hand side, they are run using NGS platform. They help with high throughput solution. And target products and Flex and Focus are would help in mid to low plex solutions. So one of the important thing in healthcare research for a platform is to ensure scalability. So if you have a scalable platform, you can save time, resource, and money in conducting your research. So this is where you can see how the Olink adds value in helping, starting from discovery and also helping you with validation and also towards clinical implementations. So each of the products have unique role at several stages of the research. In the next two slides, I want to feature two of our products which are highly relevant for immunological research. That are Olink target immunosurveillance and targets 48 cytokine. So both of each of these target 48 panels have unique value in immunological research. So since yesterday, a lot of presentations, speakers talked about cytokines, chemokines, and interleukins as well. So all of these are available in these panels. So as this target 48 cytokine name says, it contains a variety of cytokines, and the immune surveillance also has many other immune research relevant markers. So essentially, if you combine both of these panels, it can give deeper immunological insights and help discover multiplex protein biomarkers across broad therapeutic areas. Finally, I want to discuss about a classic use case from multiple sclerosis. This is from Octave Biosciences from the US. So they are running a multiple sclerosis precision care program in the US, utilizing clinical parameters, imaging parameter, that's MRI, and 18-plex biomarker panel for assessing the disease severity and staging. So what they did was that they approached us with this request, and they started a discovery study using our earlier version of the Explore product at that point of time. So once they identified the protein signature, so they developed a focus panel with us, which is a custom panel for a clinical implementation. They went ahead with analytical characterization and also finally clinical validation using various cohorts. So they are finally developed 18-plex MSDA test is now available across many clinics and hospitals in the US. So this was my last slide, and just wanted to give you a brief overview, a flavor, how the technology works, and what are the various possibilities to utilize this technology. And we have many success stories to share with you. And we do have a booth outside. Please do come and visit us. And also there is some possibility to have some fun games as well at the booth outside. So thank you very much for listening. Thank you. I'm happy to take any questions in case there are.
Thank you, Team Oling, for the very informative presentation. Next, we would like to invite the Indian Immunology Society President, Dr. Amit Avasthi, and the Secretary, Dr. Kaustav Nayak, to lead the much anticipated award session. The Senior Scientist Award this year has been awarded to Professor Alpana Sharma from AIIMS, New Delhi. I would like to invite the awardee and the chairs of this session to come up on the dais for a group picture.
Can you put the on the red one? The So a very good morning to everybody. First of all, my heartfelt thanks and gratitude to Indian Immunology Society, my seniors who are present here or not present here, my colleagues, friends, and the organizers of IMMUNOCON 51 for bestowing upon me the IIS Senior Scientist Audition Award. So today I'm going to talk about reprogramming immunity, autophagy, and iron flux as novel therapeutic targets in urological cancers. Although my journey in immunology started with autoimmune skin diseases like pemphigus and vitiligo, and which I have presented in many immunocons. So today I'm going to talk about tumor immunology. So this work has been done in the Department of Biochemistry. So we all know that urinary bladder cancer originates from the urothelium and has potential to spread to other parts of the body. In India, the incidence is 0.8 to 7.7 per 1 lakh. It has many histological variants, but transitional cell carcinoma of bladder constitute maximum, that is 90% cases. When we talk about the invasion, it has non-muscle invasive bladder cancer as well as the muscle invasive cancer, depending upon the extent of invasion to the muscles. So MIBC are 25%, where non-muscle invasive bladder cancer is 75% cases. So initial work, we have worked on Th2 cytokines, Th1 cytokines, then T helper 17, T regulatory cells, then also focused on upregulation of autophagy-associated molecules and reported in UBC tissues, and clinical significance of blocking novel checkpoint B7H4 in UBC also reported. So today I'm going to talk about autophagy, what happens in tumor-associated macrophages. So first of all, we have seen the increased infiltration of TAMs in the tumor tissue as compared to non-tumor tissue. Also, we have seen the activation status that was also higher. Then we have seen what is the phenotype, whether it is M1 type or M2 type. And to our surprise, we have found that there was a mixed phenotype. M1 is also increased as well as M2 is also increased and is correlated with the disease severity. When we compared the both, then we found that yes, M2 phenotype was much higher than M1.
Then we have seen the localization of M2 phenotype in TAMS. And there again, we have found that yes, the total TAMS, this is increased, as well as the M2 and M1 phenotype in the tissues was also higher. So then also then IL-10 secreting TAMS and IL-12 secreting TAMS. So again, it is differentiating the phenotype for M2 and M1, and again, both are increased. Also seen the immune checkpoints first on the tumor tissue and non-tumor tissue at microRNA, at mRNA level, as well as at the protein level in the ISC. And there also it has shown that yes, muscle invasive tumors have much higher this B7H4 and PDL1. We also block these immune checkpoints, and then yes, the T cell activity, the CD8 T cell activity, we first blocked single, and then we blocked the in combination by using these antibodies, and then we found that yes, when we blocked in combination, certainly the CD8 T cells which are secreting granzyme were higher, as well as the CD8 T cells secreting interferon gamma was also higher. CD4 T cells, which are also secreting interferon gamma, was also higher in blocking these checkpoints. Then so this was seen in the tumors, then we thought that what is the status in TAMS of these checkpoint inhibitors? And then we have seen that again, the same picture have come, that yes, in TAMS, the PD1 was higher, the PDL1 was higher, and B7H4 TAM was also higher in the tissue-associated macrophages. Tissue localization in checkpoints was again the same picture as we have found in the tumors. So then we have made these TAMs in vitro. We have done from the monocytes, the stimulations were given for M1 interferon gamma and LPS, for M2 IL-4 and IL-10, and then for TAMs we have given the tumor, this conditioned media from the bladder tumor tissue, and then treated with the autophagy marker, these inducer as well as the inhibitors. And then flow cytometry was done for these panels, autophagy assessment was done, and then functional assays were done, like phagocytosis assay, the ROS production, and the arginase assay. So we have seen that yes, in TAMS, the Becklin was higher, the LC3 was higher, and P622 was lowered at the mRNA level. And when we have done the protein expression at the Western blotting, we have found that yes, LC3 is increased, but P62 has shown non-significant difference. So significant upregulation of autophagy in vitro TAMs were observed. Then we have thought of modulating this autophagy by using rapamycin, which is an autophagy inducer, and MHY1458 is an inhibitor of autophagy, and MTT assay was done, and the concentration was calculated. So in the untreated cells, this autophagy modulation has shown that yes, this M1, M2, this autophagy was higher, but when we have used the rapamycin, again the LC3 and P62 was increased, but MH1Y1458 has shown decrease in the LC3 and no significant difference in the P62. So rapamycin treatment resulted in significant elevation in LC3 in TAMs, whereas MHY1458 treatment decreased LC3 expression in TAMs. Then we have seen the effect of these pharmacological modulation, that autophagy modulation, in the M1, M2 phenotype in vitro derived TAMs from UBC patient. And then we have seen that yet in TAMs, when we have used MHY1458, that is the inhibitor, the M1 is increased, whereas M2 phenotype is decreased. Same with the stimulation. So elevating autophagy by rapamycin treatment in TAMs resulted in further increase in M2 phenotype, but inhibiting autophagy decreased M2 phenotype and increased M1 phenotype, that is the immunosuppressive from this anti-inflammatory to inflammatory phenotype now. Then in the tumor tissue also, what is the status in the bladder tumor tissue? That was in vitro. This is bladder tumor tissue. Again, we have found that M1 phenotype was increasing and now M2 phenotype is decreasing after using the autophagy inhibitor MHY1458. Then also we have seen what happens to the pro-inflammatory and anti-inflammatory cytokines. Yes, we have seen that IL-10 and TNF-alpha is increased, whereas this IL-10 and TGF-beta is decreased after using the inhibitor. The immune checkpoints PD-1, BDL1, and B7H4 all have shown decrease in the autophagy after using the MHY-1458 in the M1. So, and then chemokines, the CXCR1 and CCL2, we know that it is increased in this tumor. So when we have used the autophagy modulation by using the inhibitor, the M1 phenotype, these are decreased significantly. INOS increased, whereas arginase 1 and VEGF is decreased after using the modulation. Then what happens to the phagocytosis? Then we have, for the phagocytosis, we have used the T24, that's the high-grade tumor cell line. Then we have co-cultured this, and then we have seen that, yes, phagocytosis is increased, the ROS production is increased, where the arginase activity is decreasing. So after doing this, we have again done some T24 bladder cancer cells. What happened to T24 bladder cancer cells? For the proliferation and migration, there we have used the conditioned media to these cells and then done the co-culture studies. So proliferation in normally, the M2 and TAMS, they shown the increased proliferation and the migration. But when we have used the modulation or when we have used the inhibitors in the M1 phenotype, this proliferation is decreased as well as migration of T24 or bladder cancer cells were decreased. It means functionality is restored. So again, one more experiment we have done to see whether what happens to the T cells, where the immune response is yes, it is effective, or the immune response, whether they are able to mount the good response or not, where we have stimulated the bladder tumor tissue, the lysate from the cell culture, and then we have used the T cells from with the T cells we cultured this and seen the expression of interferon gamma and the proliferation. So in this, we have found that the proliferation of T cells is increased in M1, not in the M2, but interferon gamma again increased. So all these experiments, what do they say? That yes, in bladder tumor environment, microenvironment, the phenotype is higher of the activation status, the M1, M2 phenotype, the immune checkpoint inhibitors, and these migratory potential of the CCL2 and CXR1. The functionality is compromised, as we have seen the phagocytosis, INOS, ROS decreased, and the arginase and angiogenesis is increasing. And then the interaction with other cells, with the bladder tumor cells, we have seen the proliferation and migration is increasing. But the T cells proliferation in interferon gamma is decreasing. After using an increased autophagy in TAMS. So after using the inhibitor, what happens? The phenotype is reversed. TAMS switched to pro-inflammatory phenotype from the anti-inflammatory phenotype. The functionality is restored. And then the interaction with other cell, with the tumor cell, the proliferation and migration is decreased, whereas T cells activity is increased. That is why proliferation and interferon gamma, the T cell functionality is improved in tumor microenvironment. Hence, we can say we have not proved it. The autophagy inhibition in TAMS can be a potential therapeutic target to restore the anti-tumor immunity in bladder cancer after validating in more samples in future. So this was one part, and this work has been done by my PhD student Aishwarya, and we have published few papers related to this work. And the second part is that urological cancer, we have worked on renal cell carcinoma also, and there, since my lab is working on autophagy, so autophagy and ion channels in renal cell carcinoma was explored. So what is the limitation of therapies in RCC? We all know that chemotherapy and radiation therapy is not effective in renal cell carcinoma. We have radical and partial nephrectomy, that is the mainstay of treatment, and where we have metastasis and other chronic kidney disease problems. And the immunotherapies have shown just the overall response rate is 20 to 40%. So what can be the better modality. So we know that to increase the efficacy of immunotherapy, can we have the memory T cells which are long-lived? If their function is compromised, is there any way where the memory T cell can work actively, or it can mount the effective immune response? So we all know that in circulation we have memory T cells, and these memory T cells have different phenotype, different subsets. It can be central memory cells, the effector memory cells, but the T memory cells which are present in the tissues are called tissue resident memory T cells. So we have focused on tissue resident memory T cells and seen the status of autophagy and ion channels. So first of all, we have seen what is the status of TRMs in these RCC tumors. There we have found that as compared to non-tumor tissue, in the tumor tissue, this frequency was lowered. Frequency of CD8 as well as CD4 T cells were lowered. And then we have seen the activation status, that activation status was also low or the decreased. If the level is low, frequency is low, and the activation status low, is there these TRMs are acquiring some other phenotype? For that, we have seen that yes, they are going towards a regulatory phenotype, FOXP3 positive. And then we have seen the literature that KCMF1 is a potassium channel modulatory protein which regulate ubiquitination through intrinsic E3 ubiquitin ligase activity, and it has a role in ion channel activity. And this E2 E3 complex, that is RED6, KCMF1, and UBR4, it is a complex which is actually functioning as E2 E3 complex, which regulate lysosomal mediated degradation and autophagy. It means there is a link between ion channels and the autophagy. So first of all, we have seen in the tumor and non-tumor, what is the status of KCMF1? It was lowered in the tumor tissue as compared to non-tumor. The UBR4 was low as well as the RED6 was low by confocal microscopy. So by quantitation, we have seen that yes, all three KCMF1, RED6 and UBR4 are having low expression at protein level. Then we have seen the co-localization of this complex, and there we have found that yes, KCMF1, UBR4, this co-localization was low, as well as with RED6 it was low. It means this complex is not functional well. And then we have seen this HIF1 alpha, actually the substrate for this ubiquitin ligase. If this is not functional well, it is not able to degrade it, hence HIF1 alpha concentration was higher. Then again, we have seen what is the status of autophagy. In first, we have seen in the RCC tumors. Then we have focused on TRMs. So there we have seen that yes, LC3 as well as P62 in TRMs were lowered. Although in bladder cancer, we found the high. Here it both was lowered. And then we have seen that if autophagy is low, what is happening to these cells? So there we have seen that by JC1 staining, we have seen that mitochondrial membrane potential was high, and the cells are depolarized, these mitochondria, and they are going in apoptosis that we have seen by annexin PI assay. So with this, we can just say that this work I have just taken the important slides, not whole of the work here due to time constraint. So we can say that ubiquitin ligase was not effective. If it is not effective, it is not ubiquitination of proteins and organelles. Autophagy is low, and this autophagy, lowering autophagy, increasing the mitochondrial depolarization, and cells are going into apoptosis. Then what is the fate of ion channels if it is regulating both this KCMF1 and these ligases? So what happens in homeostasis? We have different channels, the calcium channel, the voltage dependent calcium channel, and the calcium-dependent potassium channel. So all the channels are working, the potassium is going out and calcium is coming in. But what is happening in tumor microenvironment, various events are going together. Like there is a hypoxia, there is PD-1, PD-L1 signaling, there are adenosine, and all three, they are inhibiting this potassium channel. So what is happening in TRMs then? First, we have seen the cation concentration by ICP-MS. This work has been done in DRDO. So the potassium concentration was higher, as well as the sodium concentration was higher in the tumor tissue. Then we have seen the cation levels in the tissue-infiltrated TRMs. Potassium was higher in TRMs. The sodium was higher, the calcium was higher, as well as the zinc was higher. But when we have seen the channels at the mRNA level, the Kv1.3, that is the voltage-dependent potassium channel, it was higher. The KCCN4 was higher, but the calcium channel OREC was low, as well as the zinc channel was low. Then we have seen the protein expression of these channels using confocal microscopy. There we have found that yes, now at protein level, the potassium, the voltage-dependent potassium channel is low, as well as the KCNN4 is low, and OREC was higher and JRT was low. So these channels are at protein level are different. So expression of voltage-dependent Kv1.3 is low, hence decrease the efflux of potassium in TRMs. Potassium is much higher inside the cell, hence it is hyperkalemic in nature. Then calcium-dependent KCNN4 and calcium OREC1 channel increases, resulting increase influx of calcium in the TRMs. Then we have seen the expression of transcription factor associated with functionality of tissue-infiltrated TRMs. The exhaustion marker was increased. Memory T cell differentiation marker was low. The memory T cell survival was low. Memory T cell terminal differentiation JAP2 was low, whereas the TRM development, this TBET and these are higher because they are the negative regulator. So in summary, what is happening in RCC, we are having different. In the tumor microenvironment, different events are going on. Hypoxia, necrosis, potassium is high, and then PD-L1 signaling. So all these are increasing the potassium influx in memory T cells, suppress anti-tumor activity, inhibit activation and cytokine production, and increases expression of exhaustion marker. It means that TRMs are going into exhaustion. And also the discrepancy in regulation of conscious flux within TRMs causes impairment in mitochondrial potential and trigger the apoptosis of tumor TRMs. Additionally, excess zinc inhibited the activation and proliferation and antigenic stimulation of memory T cells. So then we have seen the literature and we have found that riluzole. Riluzole is a FDA-approved sodium channel blocker and has been reported yet, yes, it decreases the progress of some of the cancers, like there is one report in prostate cancer, but their role in immune cells was not known. So we thought of exploring it in the TRMs and what is it doing in the TRMs? Is there any effect in TRMs? For that, we have optimized the riluzole in sorted memory T cells by magnetic sorting, and then we have calculated the MTTSA, and then we have seen that yes, CD45 RO positive cells, when we have cultured for 14 days with this drug, it is showing higher activation, higher number or frequency is higher for a longer time. And then we have also seen these functional assays like granzyme B release as well as interferon gamma, they were also higher. And we have also seen some transcription factors, which has shown the mixed picture, not very clear picture about it. But yes, it is showing that these memory T cells, tissue-resident memory T cells are higher for these surviving for a longer time. Some more work is going on, and so I cannot conclude this final one. So here I can say that defect in E2, E3, this is a complex picture. But yes, there are many events which we have addressed at few points. So defect in E2, E3 ligase causes increased HIF1 alpha, leading to hypoxia and angiogenesis. Autophagy impairment leads to altered mitochondrial fitness. Altered ion channel and flux causing mitochondrial depolarization, thus induces apoptosis of TRMs. And ion channels blocking with riluzole increases TRM survival and functionality and can be a novel therapeutic target in future by selectively blocking some of the ion channels, although we cannot block all the ion channels, but some of the ion channels selectively in TRMs, that it may be a good success. So with this, I have completed these two. Times is up, but
the time is up.
I
So
will just two slides.
Sure.
I will skip all these slides. So okay, I will just skip these slides. Recently, we have started working on atherosclerosis, but I will not speak about it. I'll just show two slides. So societal contribution. We have written immunology awareness program. We have done immunology awareness program for school children and written these two books, Your Amazing Immune System, How It Protects Your Body, in English and Hindi. And we have conducted many seminars, many lectures in schools for the school children, because after COVID, the immunology program and the awareness was very important for the school children. So we are doing this, and World Immunology Day, every year we call the students and we have good lectures for them. Many of the speakers are sitting in the audience, and also the public awareness of cancer. In English, we can find much more literature on Google, but in Hindi belt, in North, it's very difficult. Patients are not very educated. So for them, we have done it. I published one immunology book, the Anand Narayan Panikar's microbiology book, which all the UG students in India, they refer this book. So all 14 chapters on immunology I have written. These are my students, my collaborators. I thank them all and the financial support from different agencies. Thank you very much for patiently.
thank you for giving a wonderful talk, and actually your contribution to the IAES is a reflection that you have really contributed so much during your time in AIMS, and therefore the society has recognized you for Senior Oration Award. And as a customary that we don't have any question answer session in this award, so therefore we will have a medal and the certificate for you and the check. Thank you.
Thank you, Chairs, and congrats to Professor Sharma for the award. We'll now be taking a short break for tea. I request everyone to reconvene by 11:10 a.m. so we can continue promptly. Enjoy your break, and I'll see you shortly. I hope you're all refreshed and ready to continue for the next session. We now have a special lecture from Dr. Narendra Mehra, Honorary Emeritus Scientist at ICMR and former Dean at AIIMS New Delhi. I'd like to invite Professor Surolia from IISc to chair this session.
Okay. I'd like to welcome all of you to this session. I think could you call people to come in?
She's already some army she's sent outside
Yeah.
to get people in.
This is the usual. Troop in, I take this. I'm indeed privileged to chair this session. I'd like to congratulate Professor Deepankar Nandi for this splendid conference. And I'm sure all of you are enjoying the scientific lectures and deliberations here. I'm indeed honored to introduce Professor N.K. Mehra, though he needs no introduction to this particular audience. His work is internationally acclaimed in the area of transplantation, immunology, haplotypes, and in immunogenetics. He has made outstanding contributions, especially in relation to the HLA-DQ haplotypes associated, HLA-DR haplotypes, DR2 haplotypes associated with susceptibility to tuberculosis and leprosy during his early career. Subsequently, he made a remarkable observation about the distinct haplotypes in relation to HLA-DR and HLA-DQ antigens in relation to diabetes and arthritic conditions in the Indian population, which mark them distinct from the population elsewhere. For his remarkable contributions, he has been recognized by many prestigious awards, such as Shanti Sarup Bhatnagar Prize, the most prestigious scientific award in the country, and has had the privilege of founding FEMSA, that is the Federation of Immunological Societies of Asiatic and Oceanic regions. And he wears many hats. Apart from being head of transplantation immunology and immunogenetics at SRL Hospital at Gurgaon, he is also a national professor, ICMRCG Pandit professor at All India Institute of Medical Sciences, and he is also the foreign secretary or the vice president of Foreign Affairs at Indian National Science Academy. Apart from his scientific and personal accomplishments, I like to highlight two unique things about him. One, he is one of the wittiest scientists I have come across in my life. He has as many jokes as the repertoire of genes or the mutations available in the area of, immunology, genes like IgG, IgM, all that. Apart from that, poor Americans every 4 years can vote only after his birthday is celebrated in India. And folks, Trump doesn't like that. So if he becomes the next president, on the first day itself, he'll make sure that Americans no longer vote after the celebration of Rinder.
Thank you very much, Avdesh. Thank you very much for those very, 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 very kind words. And Avdesh Saruli is a good friend of mine for many, many years, and I've seen him after a long time. And thank you very much for those very kind words. I thought I'll speak to you xenotransplantation, because this is a subject that has been in news for the last few months or a few years. And I will start with this slide, which shows the transplant scenario in the country. The solid organ transplantation, living, the diseased, or what we called the other types of, all kinds of transplants are being done in India. What is more remarkable is that India performs nearly 17 to 18,000 transplants every year, the most in the world after US and China, but remains way behind in terms of the number of transplants, way behind many high-income countries in terms of the number per million population. So we only do about 0.65 because our population is very large compared to the European countries, which do about 6 to 8 transplants every million population. Notably, nearly 15% of kidney and 20% of the liver transplants in India involve the diseased donors, the donors who actually die, the young people who actually unfortunately die through the road traffic accident, and we can take their organs for transplantation. According to 2022 stats from the Global Observatory, India amounts to 25% of the LRD, the living related donor kidney transplants worldwide, but only 3% of the diseased donors. This is something that we need to really improve. And this slide shows the global scene where both in the living field, the living donor field, as well as the diseased donor field, the number of transplants that other countries are doing much more. When we look at the human-to-human organ transplantation, there are two challenges for us. One is the matching between the donor and the recipient. We want to look at the algorithm because the HLA genes are very polymorphic. The chances that my HLA genes will be similar to that of yours is actually a 1 in a billion chance in a random population. So therefore, most of the living-related solid organ transplants are being done with parents as the donors or your siblings as the donors. And there have been a lot of advancements in the next generation sequencing, because our ability to define HLA alleles at a single nucleotide level has really advanced a lot. And this actually has been a boon, because that actually takes away the ambiguities. And the slide shows the number of alleles that we know till date in HLA, A, B, C, D, or DDQ and all. The second challenge is even more important, and that is the presence of donor-specific antibodies. Antibody characterization is the most important clinical need. An introduction of the Luminance platform, the solid phase assay, has been a very singular achievement in the last few years, and this technology provides for us a good clinical assessment of the underlying immune response. But there is a growing gap between the number of solid organ transplants performed annually and the actual need. As I said, we do about 17 to 18,000 kidney transplants each year now in India. This is much more than we were doing about 3 to 4 years ago. We were only doing about 8 to 9,000 transplants. That shows the number of transplant centers are really increasing exponentially. But our need is of 200,000 transplants a year. So you see the big gap. And there is a critical shortage of HLA well-matched to donor organs, and the need for a sustainable and ethically viable alternative. And there are possible options for the alternative. You could use stem cell-based technology and lab-grown organs, 3D bioprinting, organ-on-a-chip technologies. All of these are on an investigational stage. But what we need to do is upscale the disease donor transplant program in the country. We are looking also at non-human primates as possible donors, because you can see the pig. The donor pig could be a sort of donor for us, for all kinds of organs, for skin, even for the islets of pancreas, for the treatment of diabetes. What is the definition of xenotransplantation? Definition is transplantation, implantation, or infusion of cells, tissue, or an organ across species. And when it occurs among closely related species, for example, mouse and rat, we call it as concordant. And we call it a discordant if it is between unrelated species, for example, pig to humans. Now, given the anatomical and the physiological similarities between pig and the human, the pig could solve the donor organ shortage. The main hurdle is that the humans and the non-human primates possess antibodies directed against carbohydrate targets on porcine cells. In the last few years, molecular engineering of the pig is likely to bypass this initial hurdle, such that we can now focus on early or late post-transplant immunological challenges. And one of the biggest challenges that we have is the hyperacute rejection there, HAR. Destruction of the vascular endothelium of the donor organ within minutes of exposure to human blood. This occurs right on the operation table. And there are complement-dependent mechanisms involved there. The main xenoantigen is actually called XGAL, of course, or the alpha-gal, galactose alpha-1 to the alpha-gal. Pigs and most mammals express this particular molecule, which places alpha-gal on their glycoconjugates. Humans are a natural knockout of this gene, actually. And this leads to the presence of what we call preformed anti-pig antibodies in humans, which are primarily directed against 3 glycan targets. And those targets are shown right over there: GGTA, which is actually the alpha-gal that we are talking about, CMAH, which is the new 5GC, and the 4 beta-4 gal-NT2, also called as the gal-NSC. These are carbohydrate antigens targeted by pre-existing antibodies in humans and the non-human primates. And then there is the issue of the SLA, the swine leukocyte antigens. The ability of SLA to sensitize human HLA is under debate, actually. But what we've seen is, thanks to the genetic engineering, currently the biggest challenge is the occurrence of preformed donor-specific antibodies. Molecular engineering of the donor pigs is largely able to bypass this initial hurdle. The gene-edited pigs undergo the following molecular engineering abilities. We need a 10 gene edited pig, and that 10 gene edited pig is the key to transplantation. We need 4 genes have to be knocked out, all those 3 that I showed you. And another one is the pig growth hormone. The growth hormone also has to be knocked out. Otherwise, the pig organ will keep growing inside the human host. And 6 genes have to be knocked in. So 4 knocked out, 6 knocked in. And the knocked ins are that the 2 genes for the complement inhibition, 2 for the immunomodulation, and 2 for the anticoagulation. This is the way it is done. Humanized pig organs, which genes to knock out and knock in. You have the fertilized egg, which is sort of implanted into the pig and the oocyte. And people have now started looking into many more genes as well. In addition to those 10, they've also looked into transgenics or knockouts of several more actually genes. Let's compare the HLA from the SLA. That's the most important issue because we're putting pig kidney into the humans. So we need to really compare the swine leukocyte antigen system for that of the human leukocyte antigen system. Fortunately, they look to be very similar. Their biochemical structure is very similar of class 1 and class 2 molecules. Like in the humans, we have the HLA1 and the HLA2. In swine also, we have that. And like actually, the bulk of the polymorphism is focused around the peptide binding groove. As you can see in the case of the SLA1, alpha-1, and the alpha-2 domain of the class 1. And in case of the class 2, it is only the beta domain over there. And that red bars, of course, show the sites at which the variability occurs. And if you compare the MSC class 1 genes between SLA and the HLA, the first thing that comes to mind is whether the HLA is very polymorphic. You can see that the number of alleles that we have identified so far through NGS-based tools for HLA A, B, C are extremely high. We have more than about 27,000 alleles known now. And in swine system, it's much lower. So SLA class 1 proteins are split into, like humans, into classical and non-classical groups with loci homologous to that of the HLA1. MSC class 1 chain-related proteins, what we call MYC, whereas in the human system, we have got several MYC. Here is only encoded by a single locus there. These similarities are observed despite differences in the genomic arrangement of SLA and the HLA. Class 2, similarly, the number of alleles that we find in SLA at this point of time are much lower than that of the human system. Human system, as you can see, is very polymorphic. DR, beta 1, and then associates with B3, B4, B5, DQA1, DQB1. We need to match for all of these genes if we are trying to select a suitable donor for organ transplantation. So the SLA class 2 system is not as complicated as the human HLA2. Only the DR and the DQ proteins are expressed. DP antigens are not there. DR antigens are restricted to DR alpha and the DRB1, and there is no association with DRB3, B4, and B5 like we have in the human system. The issue is of antibody testing for xenotransplantation. Whereas in the human system, we have the solid phase assay to sort of look for the antibodies. That system is not yet identified in the SLA system. So in contrast to the multiple tools available to us for HLA antibody testing in the laboratory, only cell-based assays are available for SLA. And cross-matches using porcine lymphocytes are further complicated by the constitutive expression of SLA class 2 genes on the T lymphocytes. So cell-based assays are there, but very difficult to be done. This means that the current gold standard for clinical HLA antibody assessment is missing from xenotransplantation. Yet, I think that for the longest time, you've seen that the xenotransplant studies were limited to porcine transplants into non-human primate transplantations. Although closely related to human, the NHPs have obvious limitations. More recently, a few very successful human trials have been done. So in the second part of my talk, I'm going to talk to you about those these three centers doing human transplants. New York University doing the decedent recipient transplant. Decedent means you're putting the porcine kidney into a brain-dead individual, trying to learn the biology. Compassionate use of pig to human heart transplants done at the University of Maryland. And then, of course, the pig to human kidney transplant done at the Brigham Hospital, Boston. This is the study where the 2 cases of pig to human kidney transplants were done by Bob Montgomery's group. Those of us on the field of transplantation would really know Bob Montgomery and his group. They have done extensive studies. So the bulk of the mechanistic insights have come from the decedent recipient model. In this model, the brain-dead recipient became the recipient of the porcine kidney transplant. Pigs were like, as I said, genetically engineered to be ex-gal knockout. And 2 xenografts were done in 2021, one in September and the other in November. And these both organs survived for 54 hours. And that was a great sign that something can be done. They also looked at the comprehensive immune phenotype studies of the pig to human kidney transplants. And these actually give you insights into the underlying biology, because at 54 hours post-transplant, the xenografts show a histological picture that resembles the classical ABMR that we see in the human system. In these particular pictures over here, you see the first one is the wild type kidney there, the second one pre-transplant, the pig kidney, and then the first transplant, the second transplant. And of course, the histology shows, like in the human system, that there's an antibody-mediated rejection. And they've also looked into the molecular profiling of the xenograft after transplantation. This side, of course, is the pre-transplant. And in the post-transplant, of course, there are a number of genes that are expressed higher, that the endothelial cell activation, NK cell response, myeloid cell response, and even the gamma interferon response. So you can see both the transplant that they did, the molecular profiling shows. So the transcriptomic analysis of the xenograft helped to confirm the ABMR phenotype in the host. The next question was, what cells are involved? So the cell type spatial profiling estimates the proportion of each cell type in the various kidney compartments there. And this is a very meticulously done study in those two transplants, the pig transplant 1 and 2 over there. You can see certain molecules are expressed very high, particularly the NK cells, the monocytes, and the dendritic cells over there. So these are the cell types that really come up, in particular the NK cells. So when you look at the human microvascular rejection in such transplants, the 2 cells are very important here. The 2 effector cells involved in MVR are the NK cell and the monocytes. Both are closely equipped to work in concert and deal with the antibody opsonized targets. Whether SLA class 1 sufficiently engages the human inhibitory NK receptors is not yet determined. But given the absence of glycan xenoantigen, because these were ex-gal knockout pigs, the risk for early antibody-mediated damage has been averted. Regardless of the exact activation pathway, human NK cells get activated within the xenograft and initiate the rejection process even in the absence of antibody. Now, this was a very remarkable finding. So the enigma is whether the cross-reactivity between anti-HLA and SLA has clinical implications is a subject of the debate. And MVR can manifest even in the absence of donor-specific antibodies. Graft outcome is similar in DSA positive and the DSA negative MVR. The question, therefore, is if it's not the antibody-mediated damage that is occurring, then how are these xenografts being rejected? So obviously, there's a lot more that needs to be learned. When you look at the NK cell recognition of non-self, this is a cartoon which shows you the NK cell activation is based on a balance of positive and negative signals from engagement of activating and inhibitory receptors respectively. And you can see picture A and picture B, NK cells are activated when the balance shifts to the positive sides by the absence of a negative signal leading to the destruction of the target cell. Other people have also looked into the physiological function, physiological homeostasis, not just putting the kidney from the pig to the human there. They also want to make sure that the kidney functions, all of those functions that we know of that the kidney should do, and this study has done and found it to be absolutely good. So the functionality is fine. And this study, of course, they had a very low follow-up. So they didn't examine the impact of the immune response on the physiological function due to the short-term follow-up there. I want to talk to you this heart transplant. I'm sure many of you have seen this. January 7, 2022 was a really historic day when the University of Maryland School of Medicine first time did all of the time the kidney transplants were done. The first time the heart transplant was done. Again, the genetically engineered organ there, but they made sure that they switch off the growth hormone receptor genes to prevent excessive growth of the heart. And the US-based biotech company called Revivicor is now going after this, that they are really established a very big setup over there of the GE organs that are available. The transplant team was led by Mohammad Mohayuddin, who had considerable experience in genetic engineering. The biggest challenge, of course, is how to prevent spontaneous immunological rejection due to the antibodies of the pigs. By the way, there are fundamental differences between the pigs and the humans, and there have to be. While humans have a well-established ABH blood system, that's not the case in the swine. And of course, I already talked to you about the knockout, particularly of the X-gal or the alpha-gal. So Mohayuddin, this is the guy who then survived for 2 months. Pig heart, the guy survived for 2 months. And Mohayuddin said that Bennett was having infectious episodes. We were having difficulty maintaining a balance between his immunosuppression and controlling the infection. A lot of people ask, why do a diseased, brain-dead recipient cardiac transplantation? And there are a number of questions that need to be answered. And I think xenotransplantation field has all of these questions that need to be highlighted. I don't want to read each one of them, you can see, but I think one very important issue is there a transmission of any porcine pathogen to the human recipient there? And last, of course, we need to increase the opportunity for education about the xenotransplantation with the layman and garner acceptance of this procedure as a bridge or a destination therapy in future. But mind it, pigs are going to be promising organ donors for humans, for heart, skin, kidney, heart valves, axon tracts, corneas, and you name it actually. There are, of course, ethical and societal concerns as well. A lot of people have raised those societal concerns, and I've tried to really highlight some of them. Fair distribution of organs, animal welfare issues. There will be animal ethics guys who will come up with those, and living conditions of the donor animals. They might fail to meet the animals' requirements. Some people feel xenotransplant violates the God's will. And there are also societal concerns for using heart of pigs for humans. Bollywood songs will go off. You're not going to sing about Dil Diya, Dar Diya. And science and society. So you need to prepare the society for the xenotransplantation. This is my last slide. And we in India, we believe in our most revered God, Lord Ganesha, who has been titled as the God of transplant surgery. And there are other examples as well, Daksha, for example, and the chimeras. So at this point of time, xenotransplantation is in transition. Despite initial success, immune hurdles still remain, and these include the clinical application of xenotransplantation. Xenoreactive antibodies are the most important hurdle. Nevertheless, new insights into the specificities of these antibodies, pathways and kinetics of their production, as well as genetic determinants of pathogenicities, offer novel opportunities for intervention. We need to develop efficient laboratory tools for identification and assay of pathogenic immune responses and evaluation of response to therapy. Friends, my title was Xenotransplantation, How Feasible? And I think the verdict is feasible. It is feasible. We need to learn more about the immunology, the immune response behind it. And our surgeons are really excited about it because you saw that the gap is so very large. 17 to 18,000 transplants being done in India. We need about 200,000 transplants every year. That's only for the kidney. If you add also the liver and the heart. And this is not true for only India. everywhere around the world. So I think xenotransplantation is around the corner. There have been initial successes over there, and I, like my surgical colleagues, I'm also very excited, and I need to really encourage a few youngsters over here who can look into the immunobiology of xenotransplantation. Thank you so much, Avdesh.
Thanks, Professor Mehra, for an elegant talk. We could have one burning question if any one of you have. Okay, I'd like to ask then one question. Do you have any question there?
Yeah, yes, sir, one question.
Yeah, please.
Professor Mehra, as usual, it's wonderful to listen to you. As many times as you give a talk, it's always wonderful. My simple question is, in the xenotransplant, the death was due to the infection. So why the transplanting organ can't be practically made sterilized and then, transplanted?
In organ transplantation, this is also in kidney, human to human organ transplantation, we use still, even though you have a well-matched kidney, you use very high immunosuppression. Immunosuppressions are a part of life for the transplant team. Of course, if you have a well-matched kidney transplant, coming from your HLA identical donor, HLA identical sibling, the doses might actually low down, but the immunosuppression is given. And when you're giving high doses of immunosuppression, infection will come.
No, my question was whether you can soak the tissue into varieties of antibiotics so that, it becomes...
All of these questions will have to be answered.
Right.
I think...
I had a question here, over here. You didn't say anything about porcine retroviruses. There are chances of retroviruses jumping from pig cells into human cells.
I think we can discuss it later.
Okay.
I think during break. I had the same question, and because of the constraints of time, we have to move on. Thanks, Professor Mehra, for excellent lecture.
Thank you very much. Thank you.
Thank you, Professor Mehra. I would request the speaker and the chair for a group picture. Please come up on the dais. Yes, sir.
अरे भाई, ये तो हमारे इतने पुराने दोस्त हैं, इनके साथ तो गले मिल के फोटो लेनी पड़ेगी.
You can connect his laptop.
Let me just pick up my... This one? It's asking for keyboard. The keyboard doesn't work, I can just use that. Okay, let's see. This works. Keyboard works. Good morning. I guess somebody is going to introduce me.
Yes. Okay. Good afternoon, everyone. I think this session, the speaker is more eager to keep the time. So we will respect his wish. So it's our pleasure, myself and my co-chair, Professor Nuruddin Khan, to welcome you for your talk. Please take the dais and it's all yours. Thank you.
Okay, thank you. It's always a pleasure to be here. I've been coming to IMMUNOCON for a long time. I'm an Indian Immunology Society member for I don't know how long. For some of you that do not know me, that I did my PhD from IISc like 1991 to 1999. And you don't know that you miss how much you miss the campus when you leave. It's a beautiful campus, and it's hard to find such a beautiful campus and overall the scientific environment elsewhere in the world. And I also really miss is the food in the AMS. That's one thing you realize when you're here, you're fed very well. Like you're fed 4 times a day. Right? I used to be the first person in the mess before they even opened. That I used to go through the back door and eat. But once you go, for example, to US, the thing you realize is you have to cook on your own. So be prepared if you're trying to leave, and it's very important. And I'd like to thank Dr. Nandi for giving, and the other organizers for giving me this opportunity. And my lab's focus is not the monkeypox research. My lab works on various vaccines, including HIV, hepatitis C, TB, and influenza. So I put this talk together just the day before yesterday from scratch. I built every slide by myself. So I hope it's going to be a smooth talk. Let's see. Okay, so. Okay, so the monkeypox virus, MPXV, causes this mpox disease. This is an orthopox virus that has caused periodic outbreaks of mpox disease over the past 50 plus years. It is related to smallpox and vaccinia viruses. However, unlike smallpox, which only infects humans, mpox is zoonosis, means that it can transmit from the animals with an animal reservoir. So here primarily the rodents. So it makes it very hard to really eradicate this infection. Historically, most outbreaks occurred in Africa with spread limited by inefficient human-to-human transmission, along with pre-existing population immunity conferred by smallpox vaccination. The smallpox was eradicated from Africa in 1977 with widespread vaccination, obviously. But this was stopped in 1982. After that, you start seeing cases of mpox emerging. So now it is a public health emergency of international concern. So in 2022, a large outbreak occurred with a newly recognized subclade 2B, over 95,000 documented infections, primarily transmission via sexual contact, but the contact itself is enough for the transmission. Most cases were mild, featuring fever, malaise, and smallpox-like pustular skin lesions. Some severe cases occurred. Fortunately, the mortality rate is only 0.2%, compared to smallpox, for example, probably 30% or so. And then a lot of GI issues. So the way the virus is spreading across the globe, that the WHO declared this as a public health emergency of international concern. So there are 3 clades of the virus exist, clades 1, 2A, and 2B. Obviously, they're called different clades because there are differences in the genome sequences. People are still trying to understand the pathogenesis of these 3 clades. Bernie Moss recently did an experiment in these inbred Castaneous mouse, compared the clade 1, clade 2A, and clade 2B, and then they found that the clade 1 is the most virulent, and the clade 2B is least virulent. So interestingly, the sequence, there is a great sequence similarity between 2A and 2B. 2B was not lethal, even at 100 times the lethal dose of the clade 2A in the mouse. So we had to see as the human data comes up and how it pans out. So today I'm going to talk about the original smallpox vaccine, that is Dryvax, and the current smallpox or mpox vaccine, MVA, and newer vaccines for mpox and the human immune response in mpox survivors in the US. So the smallpox vaccine, Dryvax, I'll be talking about immunogenicity of this vaccine and correlates of protection. So the smallpox vaccination has a special place in the history of vaccination. Way back, Edward Jenner did an experiment in humans vaccinating a child. So this is how this experiment was actually described. And you cannot do this experiment now. But this has changed the vaccinology altogether. Those days, there were no IRBs, consents. But what he observed was that the dairy mates were protected from smallpox naturally. And also people who had the cowpox infection were also protected. And they also transmitted that protection from one person to the other. So he wanted to test that. So he basically took lesions from a person named Sarah Nelms who had the cowpox infection. He literally took the matter from her and inoculated on the skin of this 8-year-old boy, James Phipps. And then he felt a little bit sick for some time, a few days, and after that he felt okay. And then, it's terrifying to hear this, that then after a couple of months, he went back and gave smallpox to that boy, and then showed that he was protected. So this kind of jump-started this concept of vaccination. Imagine this was done when there was no knowledge of B cells or T cells. So the B cells and T cells were discovered about 150 years later. So all the vaccinology started even before that. So what is the smallpox vaccine? This is called Dryvax, and it has a few other names. It is a replication-competent vaccinia virus. It's a cowpox. Along the way that there are other strains came. One of them is the NIVAC, New York board vaccine strain. The amazing thing is a single dose of vaccination can induce the lifelong immunity, but it can cause significant adverse events in certain groups of individuals, including immunocompromised. So here is the data that published by other labs, the Marks Klifka on the left, and this is from Rafi's lab, who is sitting here. It shows that here the individuals received vaccinations, different numbers of vaccinations. In the black, you can see that individuals who got single shot, even after 70 to 80 years, you can still show the presence of these vaccinia-specific antibodies. And around the same time, Rafi's lab also showed that this was also associated with presence of these long-lived memory B cells in that individual blood. And in the same paper, in Slipka's paper, and actually Slipka was Rafi's trainee, and he also showed that the CD4 T cells induced by vaccination also persist for that long. And then around that time that I was a junior investigator. I know there was a big buzz about these studies, and Mark was publishing in Nature Medicine. Rafi went in quickly with JI Cutting Edge, and I was a poor new investigator. I went into JV. So we also showed that these smallpox vaccinia-induced T cells, they can persist for a long time. And then we showed that the CD4s persist better than the CD8 T cells for unknown reasons. But when you vaccinate these people again, and then you do see a recall of CD8s, again, they go down quicker than the CD4s. And then we did non-human primate studies where we gave the animals the Dryvax vaccine, and then you can see the generation of these cells where you can track the contraction of the response pretty closely. And then they contract in the first 8 months. And then after that, they're pretty stable for several years. So we followed these for 4 years, and we can show that the T cells are still there. And then same is true for the antibody. So around the same time, Frankini had done an experiment to understand what is important for protection. So from the human data, there is evidence that both antibodies and T cells are important. And then she did this experiment in monkeys using a monkeypox as a challenge. So in one group, she depleted CD8 T cells. So they all got the Dryvax vaccination. In one group, she depleted CD8 T cells before exposing to mpox. In the other group, she depleted B cells before vaccination, because you can't deplete antibodies after vaccination, because there's the plasma cells in your bone marrow, they make tons of antibodies, and you can't deplete, they are CD20 negative. So that's why she first depleted B cells and then vaccinated and then infected them. So these animals don't have antibody to Dryvax. And then this is the control, that there is no depletion. So what you see here is that this is, sorry for this table, but basically in your control that there is no virus replication, there is no skin pox because the vaccine protects. But when you deplete B cells, when there is no antibody, that then you start to see. But when you depleted CD8 T cells before challenge, that you still are protected because these animals have antibody. But there are some caveats to this experiment, but it proved that if you have antibody response, you can get good protection. So then the current smallpox vaccine, which is MVA, because there are a lot of issues associated with vaccinia virus, because it's a replication competent virus, people were looking for alternatives, and now this is marketed as Genios by Bavarian Nordic. And I'll show you some data that we and others have generated with MVA as a smallpox vaccine. So the MVA was derived from vaccinia virus by just passaging in chicken embryo fibroblasts for over 500 times. It underwent 6 internal deletions and became replication defective in human cells. And it has an excellent safety record, including in HIV-infected people. So Bernie Moss showed that the MVA can act as a vaccine for mpox. He was mostly focused on using this as a vaccine for smallpox, using mpox as a model. Here, this one group of monkeys received 2 doses of MVA, one here and one here. Then 1 dose of MVA was not enough. And when you give the second dose, you can induce these strong neutralization titers. So these are compared to, first you give MVA followed by the Dryvax. And this is single Dryvax vaccination. And this is the control. So you need 2 shots of MVA to get the same levels of titers that you get with a single shot of Dryvax. But then they all protect nicely after challenge. This is the monkeypox genomes in the blood. So this is the first time that it was shown that the MVA can act as a vaccine for mpox. The same analogy for smallpox. So the MVA has extensively been used as a vaccine as a vector for our vaccine development. And we have used DNA followed by MVA as a heterologous prime boost for our HIV vaccine. And then those days, we were interested to see if the same HIV vaccine can provide protection against mpox. So there's one group that got the Dryvax vaccine and then got the mpox. So this is many years after the single dose. And then this group of the animals got the HIV DNA MVA vaccine. And the 2 shots of the MVA HIV is given here. And a few years later, we exposed these animals to the mpox. And then what we showed was that if you look at the neutralizing antibody titers that come up after the second shot of the MVA HIV vaccine, and they're pretty stable for the next 2 years or so. And then when we challenge those animals, and they are nicely, the DNA MVA vaccinated animals, they all get infected, but they quickly clear those skin pox and then clear the virus, as compared to the controls that you euthanize them by 13 days or so. So this, again, showed that the MVA can provide protection against the mpox disease. And then we also worked on further improving the MVA. So we made a new MVA that is called MVA B13R. B13R is a caspase 8 inhibitor. And this is deleted in MVA when it was derived from vaccinia virus. So because of that, MVA infected cells undergo rapid apoptosis within about 8 to 12 hours. So we put this gene back into the MVA and then showed that it actually enhances the immunogenicity of our HIV vaccine. So this can also be used as a non-recombinant MVA B13R, could be a better vaccine than the MVA. So we are currently testing that. And then more recently, that people are using other platforms like mRNA and AD35 to express these specific genes, immunodominant genes, that induce neutralizing antibodies. And they showed that they can provide protection from mpox challenge, and which is encouraging. So you'll see more on that. So now moving on to the human immune response to mpox to the mpox in survivors. So this study is done by 2 people in my lab. One is Sam, and he recently celebrated his birthday. And then Shiny, this is during our Christmas party that we were having fun. These 2 individuals did this work. So we used a cohort from US in Atlanta with people living with HIV. So we studied 2 groups, the mpox group, 15 individuals with long-standing HIV and the recent mpox infection. 4 of these patients were born before 1972, and we presume that they received a smallpox vaccination. And the 3 patients received 1 dose of Jynneos, 1 shot before they were infected, or at the time they were infected. And we collected samples at about anywhere from few weeks to 6 months after infection. And the control group, these were the archived frozen PBMCs and sera from the same people living with HIV, were taken prior to the mpox epidemic, and also from individuals born after 1972. So they would not have received any vaccination for vaccinia. So we looked at the cellular immune responses and humoral immune responses. We did the intracellular cytokine assays to measure interferon gamma, TNF alpha, IL-2, IL-4, and cytolytic potential by perforin expression and degranulation. And then we stimulated cells with either a live MVA or orthopox peptide pool. So these are the peptides that are predicted to derive from the vaccinia proteins based on the predictions for their prediction, based on the prediction to bind the MHC class 1 and class 2. And the same is done for the mpox peptide pool. So we also compared responses against HIV and CMV specific cells in the same individuals. And then we measured the binding antibody and neutralization assays for mpox and vaccinia. So here is an example of, I'm just watching my time. example of the ICS assay is unstimulated cells. Here you have interferon gamma and TNF alpha and IL-2 on the x-axis, and their co-expression on the CD4s and CD8s. And these are different stimulations like MVA, orthopox CD4 pool, and mpox CD4 pool, peptide pools. So if you see now that here is the legend here. So this is the MVA stimulation, orthopox CD4 peptide pool stimulation, and mpox CD4 peptide pool stimulation. These are the HIV in positive individuals infected with mpox and no mpox. You can see there is a decent amount of CD4 T cell response making interferon gamma in all three stimulations. But when you look at the CD8 cells, you saw that more with MVA stimulation than the peptide stimulation. It may be because the MVA is covering more proteins, and it's also maybe the presentation was better, but we're not sure. You see a similar situation for TNF alpha expression, similar expression of TNF alpha across the three groups, three stimulations, but again, mostly interferon gamma, TNF alpha with MVA. And then overall, we saw a nice IL-2 response as well. And in general, CD8 T cells generally make less IL-2 than the CD4 cells, and this was expected. So this was pretty encouraging that it looks like these cells are polyfunctional. So when we'll compare these immune responses to, in the same individuals, responses to GAG and CMV, you can see that there is quite comparable. For the CMV, we use the PP65 peptide pool. You're looking at response to one protein of CMV, and then one protein of HIV. So they are quite comparable across the board, except that the production of IL-2 by CMV specific cells were actually lower, which is actually expected for the CMV because it's a chronic persistent infection. But again, importantly, that it shows that these individuals have good frequencies of CD4 T cells making all three cytokines. So then we asked the question, are these polyfunctional? The way you ask this question is that these are, if you have cells making only gamma, IL-2, or TNF, you can derive 7 combinations of cells making all three cytokines, or any 2 cytokines, or any 1 cytokine. So the triple producers, double producers, and single producers. If you now look at what it looks like for these mpox specific cells, and the CMV specific cells mostly made interferon gamma and less TNF, whereas the mpox specific cells made both interferon gamma and TNF. These are single producers. There are low levels of triple producing cells for the CD4s, and you can see based on the colors here, the CMV is mostly interferon gamma, and the mpox you have both interferon gamma and TNF, and some of the triple producers in the blue. So overall, if you see the cells that are having more than one function, the mpox specific cells are higher than the CMV specific cells. And interestingly, some of these CD4s also express perforin, co-express perforin. This is the gamma and perforin positive cells. They show a little bit of degranulation. This was based on the frequencies, they were higher with the CMV than the others. And it again shows that there's some cytolytic CD4s present in these individuals. And then looking at the CD8s, that here you see the CMV specific cells are in magnitude are higher for gamma as well as TNF, but not for IL-2. And then the mpox specific CD8s are at a lower frequency, but they're there in some individuals. And then polyfunctionality wise, again, that you'd see that most of them are single producers. We don't have triple producers in these HIV positive individuals. So, and similar to CD4s, we see much higher frequencies of those perforin positive, interferon gamma positive cells. And then qualitatively, they're at higher frequencies in these mpox individuals. So again, these show that the cells that are there that probably are cytolytic, and they could provide protection from a future infection. So these are the conclusions. In terms of in the interest of time, I want to quickly show you that when you look at the neutralizing and binding and neutralizing antibody, so you have good binding antibody to mpox, and there are also neutralizing activities there against the mpox and vaccinia. And then there is a relationship between good relationship between the binding and I guess there is the binding antibody correlates nicely with the neutralization, either against vaccinia neutralization or mpox neutralization. And we asked the question, is there any association between the time after infection and the titer of the antibody response? So the binding antibody decreased over time, depending on the time after infection, whereas the neutralizing antibody didn't have a significant decline. So maybe they persist better than the binding activity. And was there any influence of prior vaccination on humoral and cellular immunity? So what we found was that if the individuals received vaccinia vaccination before, then they had higher antibody response in general. And interestingly, there was no effect on the T cell response. So they were comparable between the two. So I'm done. This is one more slide. So basically, the conclusions are that mpox infection induces a strong binding and neutralizing antibody. They correlate, associate nicely, and then prior vaccination leads to better binding and neutralizing antibody, but not T cell response. With that, I'd like to thank people who did the work in my lab. Again, Shiny and Sam, and clinical group, Colleen Kelly and Andy Sheth. And the neutralization assays were done by CDC, people at CDC. And then the peptides were provided by Alex Serres' lab at La Jolla. And then the funding from NIH. I'll stop here.
Thank you so much, Professor Ramarao. We not only enjoyed your science, but we greatly appreciate your time management. You are only 40 seconds up. We have great time for interaction. Thank you.
Ramarao, I'm here.
Yeah. So,
here.
Where?
Here, this side.
Yeah. There,
okay. Hi, sir. How are you?
We will take a couple of questions only. Okay, because as usual, Rama is always on time. This time you have kept on time. Yeah. Okay. So we'll take a couple of questions if you are.
How are you, sir?
Yes. Go ahead.
Can I
Go ahead, please.
So mpox spreads exclusively via physical contact, and worldwide about 95,000 cases. It's way less than COVID, and symptoms are mild and mostly self-limiting. So what is the rationale behind WHO declaring it as a health emergency?
So it's a policy question, but I think they're afraid that how fast it is spreading, right? Because in 2022, there were cases in the US, primarily in the MSM population, and then they went away, right? But then a year later, then there is a big increase. So it hasn't come back to US yet, but in Africa, the cases really increased because it was spreading to the rest of the world. That's what I think made them.
The spread
fear.
wasn't that great.
The worry is that if the virus is changing, right? So if the virus is changing, and if it changes the way it spreads from human to human, it could get out of control.
Yeah.
So next question, Professor Nagaraj. Yeah.
Clade 2A and B, there is a large difference in the pathogenicity, or what's the basis? Have you figured it out?
No, I have not.
Anyone else?
I think people, I'm pretty sure that they'll start looking into all those.
So we don't know anything yet.
No.
Some large deletions or something between the two?
We haven't really done any research in that.
Yeah. So two more questions we can take. One,
Thank you very much. I'm just wondering about the long-term memory B cells after vaccination with mpox. Is there any data available in people who have exclusively been vaccinated with this, if it has a similar effect like the original?
Yeah, that's
smallpox?
we haven't done that yet, but we are planning to do that. And in fact, Rafi and I have been talking about it. And getting samples is going to be a challenge if you want to do it in the US, because the number of cases that are very few right now. But if something can be done in African cohorts, it could be easily done.
Yeah, I think, we can take a last question. Rama, another.
Thanks, Rama. That's a lovely talk. So first of all, a comment, so Dan Griffith's lab had a similar study where they showed that depleting CD8s actually didn't control measles virus. So maybe it depends on the virus, right? I got to defend the T cells.
So no, I think, there are no hard feelings.
Yeah. Yeah.
But the way also the experiment is done, that first of all, you don't deplete everywhere, right? And the second is that the timing between vaccination and infection is only 3 weeks.
Yeah.
Right? It's the cells are still in the effector phase, and so there are a lot of issues with that experiment. So my point was to that.
Sure. But I really think it depends on the virus, maybe also where it. Thing is your human data with mpox, and you showed us single producers also. Do they include all gamma, TNF, IL-2, all single producers, or that data you just counted just gamma producers? The reason I ask this is often
this
in
is
papers,
based on the Boolean analysis. Yeah.
Sure, but you include all three, right? Because I don't know. To me, just a CD8 T cell that makes only IL-2 doesn't make sense. And with
But
TNF,
those
you often
frequencies
see
are very
background.
low, though. So
I
I
see.
wouldn't.
So
read.
majority you would say are gamma single producers?
Yes.
All right. Okay. Thank you. Lovely
Yeah,
talk.
so we'll take, if somebody has other questions, we can take in the lunchtime, okay? So we are running short of time, please. Okay. Thank you so much, Rama. It was wonderful listening to you.
Thank you.
Thank you, Professor Rao, and the chairs. Please stay back for a group picture. We now move on to the next segment of our event. So now 3 parallel sessions will be happening in the main hall and Hall A and Hall B in J.N. Tata Auditorium. So for those of you staying in the main hall, we have an exciting session lined up. The others, I kindly ask you to make it quickly to the other halls to attend the other sessions. I would like to invite the chairs for the next session, Professor Uday Ranga from JNCASR Bangalore and Professor P.N. Rangarajan from IISc, to introduce the speakers onto the dais.
Okay, I welcome you all to this session on viral immunology. We have 4 speakers. I will introduce the first and the third speaker, and my co-chair will introduce the second and fourth speaker. The first speaker is Dr. Ambak Roy. Dr. Roy obtained his PhD from AIIMS, and I'll make the introduction very brief as we are running behind schedule. He has been studying human endogenous retrovirus mediated regulation of CD5 proteins, and he's also interested in regulation of visual leishmaniasis, and is currently associate professor at the Department of Biotechnology at Motilal Nehru National Institute of Technology, Prayagraj. So today he will talk to us about human endogenous retrovirus mediated regulation of immune response. Dr. Roy, you have 15 minutes and 5 minutes for discussion.
Okay, sorry. So I'm thankful to the organizers for giving me this opportunity. And by the time they... Thank you. So human endogenous retroviruses mediated regulation of immune response. Means I have just one small announcement. Next year, we are going to organize IMMUNOCON 2025. So I would be hopeful that everyone would be attending that. And all the means flyers, the first announcement will be happening on 1st of December. So I would be communicating means all of you. So human endogenous retroviruses are actually the impressions of the prehistoric period. They had infected the germline cells, they got transmitted over the generation, they acquired certain mutations, and they became non-productive. Now there are three classes, the class 1, class 2, and class 3. I'm working on the HERV-E particularly. And there are different arrangements. There are some deletions, there are solitary LTR. So only STLV-K is actually productive, right? But the rest of them are not productive. However, they are regulating the immune response. Now, we are working on CD5. So there are three forms of CD5. There is one soluble CD5, which is the chopped off product of the membranes CD5. And I'm interested, our team is interested in cytoplasmic CD5, and that is actually originated from the HERV-E element. And the interesting point is that when it gets expressed, you will find a downregulation of membranes CD5, and obviously there will not be any interaction with the CD72, and that makes the T cell prone to the activation induced cell death, and gradually you will find, though it unleashes the full capacity of the T cells, but it does not actually last longer. So how does that happen? There is a segment here, exon E1A, that is the conventional CD5 gene. It goes up to CD11. You will find if the mRNA expression starts from here, you will find a full-length protein, and that would result in the membrane expression of CD5. However, if that starts from an HERV segment, there is an exon E1B, and if it starts from here, it will go up to exon 11, but the intermittent region, particularly the exon E1A, will be spliced out. Now that would remain inside, and we have observed this phenotype interestingly in the leukemic T cells, where we have observed increase in the cytoplasmic CD5 and decrease in the membranes CD5. So now our finding shows that if you see, we have demarcated the CD45 based leukemic and non-leukemic, and the non-leukemic cells, they express low level of CD5. However, the normal unaffected non-leukemic cells, they express high level of surface CD5. And when we compared in the same individual, you can see there is a clear-cut shift in the membrane CD5 expression. And when we compared in a large number of patients, we found that the membrane CD5 expression was decreased. And when we checked the conventional even A gene and the conventional means the unconventional HERV-derived even B mRNA variant, we found it was almost reversed. You will find in the healthy subjects, even A was higher, and the even B, which is HERV derived, is actually low. And in TLL cases, it has been reversed. So here is means the final outcome is that we found cytoplasmic CD5 high and the HERV even B transcript in leukemic T cells. Means a slight means we are deviating, but the unaffected T cells are not actually proliferating. So we were interested that they are suppressed. So we used membrane CD5 blocking, and in 48 hours we found that the normal populations were producing IL-2, and it has been increased in number, and simultaneously that resulted in the decrease in the leukemic population. Now we know that leukemic T cells, they express increased level of cytoplasmic CD5 and low level of membrane CD5, and we found that certain a segment of the LTR region and certain segment of the GAG, they form the exon E1B region. And so what we did, we analyzed the upstream sequences for the promoter sites, and interestingly we found AHR dioxin and xenobiotic response element, and simultaneously hypoxia response element and CBP beta binding region. I'm showing data which we have completed, and interestingly IL-10 induces CBP beta expressions significantly. So we took the example of we took polycyclic aromatic hydrocarbon, and we identified 4 sites upstream to the E1B segment, and when we used benzoapyrene, we found that E1B has been increased and E1A has been decreased. Means the HER we derived, the variant has been increased and the conventional one has been decreased. We showed with the help of the flow cytometry that the cytoplasmic CD5 was increased upon PAH, that is a polycyclic aromatic hydrocarbon. It has been increased, and when we used AHR expression, obviously it was increased, and there is internalization of the pH inside the cell. And when we use the AHR inhibitor, it shows that the increased cytoplasmic CD5 was decreased. Now we use the qPCR to show that AHR complex, whether it is binding to the upstream or not. And we showed here that except the LTR region, that is the E, which is very close to the start site, you will find that rest 4 are showing significant binding of the AHR complex, but LTR is not showing the binding of AHR complex. So what we did, we went for the silencing. So it became a bit tricky for us because the control shRNA, when we stimulated with EAP, we found significant increase in E1B. So we had to reanalyze ourselves, and we showed % increase in the means E1B, and we showed that in both the shRNA, the targeted shRNA, we found that there was a significant decrease in E1B. Now the second factor we had identified, that is the hypoxia, hypoxia factor, which binds to the upstream of the E1B. So what we did, we identified sites upstream, and we showed the E1B is getting upregulated under the hypoxic condition. And since the hypoxia treatment was given only for the 4 hours, we found there was a shift. There was an increase in E1B. And here is the hypoxia verification with the help of the FDH and HIF1 alpha expression, which confirms that our setup was working. Now we showed the qPCR, we showed that all the 3 sites were showing significant binding of the HIF1 alpha complex, and we took COCL2 as a positive control. Now, just to prove our point, we just took samples from the repository of the Rajiv Gandhi Cancer Hospital, and we showed that hypoxia was there in the matched and normal and the tumor tissues, and we showed that the E1B means we actually confirmed hypoxia with the help of the beta-actin and FDH. Surprisingly, these were also getting increased, which is actually manipulated by the hypoxia condition. We showed that E1B has been increased, and we are actually currently involved in demonstrating the cytoplasmic form of CD5 with the help of the immunohistochemistry. I'm hopeful that I would be able to show in next presentations. So IL-10 So IL-10 treatment, the third factor which we had identified, that is the CBP beta, we showed that when we treated with IL-10, there was an increase in even B mRNA variant, and we identified 4 sites, CBP beta binding sites upstream to even B segment, and 3 binding sites upstream to the conventional one. And we showed that when we treated with recombinant IL-10, there was an increase in CBP beta, and when we used the inhibitor, that is the inhibitor of JAK1 STAT3, that resulted in the decrease in the CBP beta, which confirms the involvement of IL-10. We showed there are 3 isoforms. We showed that IL-10 particularly increases the lip isoforms. We are still involved in this work. So we showed here that when we treated with IL-10, we found that it has there is a significant increase in the lip isoforms, and surprisingly, we did not find the increase in the binding or enrichment of the CBP beta binding region in the ChIP-Q PCR. We've only found that CBP beta binds to the conventional the site which was present against the even A segment, which is the conventional one. And with the help of the shRNA, we showed that actually CBP beta is involved in the regulation of means the cytoplasmic form of CD5, that means even B variant. So it's a summary slide. I hope I have maintained the time. So in solid tumor, here we have shown that in the periphery, the CD5 high antigen specific T cells were there, and when it entered inside the solid tumor, there was an increase in even B variant. As I said earlier, we are in the process of showing cytoplasmic form of CD5. And as we showed earlier, IL-10, which is being produced by number of cells, so they actually also involved in reducing the even A expression and increasing the even B expression. Here is the CD means hypoxia was also one of the factor, and we showed that it also reduces. So there are multiple factors actually acting and reducing the CD5 expression on the infiltrating T cells, and that is something novel, that there is a possibility of axonal switch which resulted in increase in the cytoplasmic form of CD5. So now, as I said earlier, it's a future part that we showed that PAH is also involved in increasing cytoplasmic form of CD5. However, we are also interested that if leukemic T cells are showing increase in cytoplasmic form of CD5, so whether it's a cause or consequence, whether increase in cytoplasmic CD5 actually facilitates the leukemogenesis or it's otherwise. So we are in the business of doing that. There are certain information we have added in this figure. So here is the summary slide. I would be happy to take any questions. Thank you.
Thank you, Dr. Rai, for finishing well ahead of time. We have time for questions. Any question? Okay, there.
Hi, great talk. I was wondering if any cell biology is known about what the cytoplasmic CD5 does in terms of, say, protein stress or anything at all. Thank you.
Not exactly protein stress, but we are working on the localization of cytoplasmic CD5. It's novel. It has not been researched. So currently we are doing electron microscopy to figure it out. We have certain bioinformatics data to support that where it is localized. But to confirm that, we are actually exploring electron microscopy to figure it out that whether it is localized in ER, mitochondria, or in some other compartment.
Yeah, there.
Hi, that's a lovely talk. Thanks. I was just curious about I don't know if I missed this, but did you look at the expression of CD5 in exhausted T cells, A? And B, in just say a viral infection, I'm just curious how the expression goes in naive effector memory, because maybe it's just something to do with cell proliferation, or you have some T cell activation there, or yeah.
So as I said earlier, cytoplasmic CD5 is not a very well-researched molecule. And as far as exhausted cell is concerned, there is a possibility. And I believe that in T cell development in the thymus, where variation in membrane CD5 occurs, there is a possibility of cytoplasmic CD5, but we do not know. It's a purely hypothesis. But it would not be right if I make a comment because I have not done that work, but there is a possibility.
I think okay. So it's just a question of doing surface on one color, cytoplasmic on one color, and checking.
Right.
Exhausted.
That's the way we have done.
Okay.
That's the way we have done.
Yeah. Thank you.
Thank you, sir.
Okay. As Dr. Keshav is setting up his slides, I will introduce him to you. He is a PhD from IISc, and he worked on structural aspects of HIV envelope with the object to generate broadly neutralizing antibodies. Then he did his postdoc at St. Jude's Children's Hospital, and there he studied the relation between G, DNA, and infection. Currently, his lab is interested in zoonotic viruses, especially those viruses that infect bats and birds, and he is using structural as well as immune strategies to ask questions. Keshav?
Thank you. I would like to thank Professor Dipankar and the IMMUNOCON committee for inviting me. And I'm happy to see Ranga and Uday on the stage. And like Uday mentioned, I did my PhD in IISc in MBU in 2009 to 2015. And I'm back again at IISc in biochemistry though. So I echo with Rama about having good campus in IISc. I was in CMS though, but I was the last person to go to MES at that time. So, and also I would like to say that my talk doesn't have any B cells and T cells, and please bear with me with my talk. Before I start my talk, I'd like to thank my students who involved in this work, Sanchita, Aishi, Disha, Subham, Priyans, and Subham Saha, because I don't know at the end I'd be able to thank them. So we are mostly interested in understanding why suddenly certain viruses turn into pandemics. Especially we try to understand why bats and birds, they could able to tolerate these viruses, but when they jump to human beings and they cause the pathology. And you all must be knowing that there are a lot of human infected viruses which are pathogenic from bats, like coronaviruses, rabies, and Nipah. There are a lot of them. But the question still remains is that why they are able to tolerate in bats. Okay. The first kind of immune response that you see the moment your virus enters into any host is the innate immune response. And cell death is one such response where it tries to kill the cells infected so that it can destroy the replicativeness, so that it can clear out the infected cells from the system. But unfortunately, some of the respiratory pathogens like SARS-CoV-2 and influenza, which originate from bats or birds, they tend to cause heavy cell death and inflammation in the lung. You must have heard about cytokine storm or respiratory epithelial damage. People don't know why this happens, but somehow host cell death response gets screwed up, and that leads to the pathology. Let me go a little deep into the cell death. We are particularly interested in one particular type of cell death called inflammatory cell death. So in this case, the cell dies by a lytic form of cell death, unlike apoptosis. So there are a specific set of machinery that deals with it. It's called pore-forming proteins. Once they get activated, they form pores on the membrane that leads to the cell death. So I'd just like to show this video which is primed for inflammatory cell death. You can see that it just dies by like popping up a balloon, and then you start seeing the nucleus. So this leads to the release of all intracellular contents into the extracellular space that triggers heavy inflammation. And in general, this inflammatory cell death has a big role in homeostasis. It not only initiates inflammation, it primes adaptive immune responses early on. One such cell death pathway is called necroptosis, is mediated by a set of proteins called Rim proteins. This Rim is a homotypic interaction between protein. This motif, if any two proteins have this Rim motif, they try to pair up with each other through protein-protein interaction. There are only 4 proteins available in mammals that can have this Rim protein. So they tend to, once they form this interaction through Rim, they assemble into large complexes like filamentous in structure. Those are a kind of signaling complexes that tend to activate the signaling pathway and ultimately lead to the formation of channels or pores on the membrane, and that leads to the cell death. But you already know that viruses are smart. There are certain DNA viruses. They tend to mimic these Rim domains so that they can block this activation of cell death at molecular level so that you don't see the formation of these complexes, and that leads to the inhibition of cell death. It's kind of adaptation for them so that they can evade the host responses. But this is not the case with RNA viruses. We don't know why. And people know that in RNA viruses, these kind of cell death pathways are excessively activated. So we started asking simple question, what about bat viruses? Because most of the pathogenic viruses are RNA viruses. Do they employ these Rims or viral Rim mimics like host Rim proteins? So to cut short our analysis, we did find some of the RNA viruses in bats, especially coronaviruses, hantaviruses, and some other picornaviruses. They do have an intact Rim mimics that can mimic the host Rims to navigate the cell death. Particularly, we explored one of the SARS-CoV-2 protein called NSP13. It's a replication complex protein. It has a Rim domain like host protein, and it's also a RNA binding protein, which is required for SARS-CoV-2 genome unwinding. But we wanted to really see whether it's a viral Rim protein. So we in vitro, we characterize them, and we see that unlike the DNA virus viral Rims, this NSP13, which has the Rim domain, that actually is spontaneously inducing cell death, which is surprising for us. Not only that, we see that when we mutated the Rim domain that we identified, it abolishes cell death, and also it depends on the RNA binding pocket. And then we did this experiment with the SARS-CoV-2 virus as well. But in this case, it was difficult to make NSP13 mutants because the moment you mutate NSP13, it actually affects the virus replication. So in this case, we just ectopically expressed NSP13 in our mutants and then infected with SARS-CoV-2 and see that NSP13 indeed regulate cell death for SARS-CoV-2, and it depends on the viral Rim that we have identified. And we also did same experiments in bat cells, but surprisingly what we have seen is that, yes, this NSP13 can induce cell death in bat cells, but the problem is it was not dependent on the Rim domain that we have identified. So the take-home message here is that the bats, which are like mammals, they do have host Rim proteins that might have elicited evolutionary presence on these pathogenic viruses that leads to the evolution of viral Rims. But in this case, they actually promote cell death in both bats and birds. However, the viral Rims, they don't show any function in bats, but they do show function in cells. But we also solved the mechanism how they activate it, and we figured out that this NSP13 gets activated by a type of pathogen-associated molecules called zRNAs or zRNAs, and we see that these are actually required for sensing a host protein called ZBP1, and that is leading to the cell death. And SARS-CoV-2 NSP13 protein actually mimics the host cell protein in activating cell death by sensing SARS-CoV-2 gRNAs that we have identified. And not only that, what we see is that this particular viral motifs that we detected, there are 7 coronaviruses that have infected humans, but only 3 of them have these conserved viral Rims, that too pathogenic coronaviruses. So at this point, we think that we're trying to develop methods to see that can we use them as a molecular cues to predict the pathogenic potential of the viruses, which are there in the bats, but they haven't come to the humans so far. But this is good that we identified that bats can be eliciting pathogenic traits in the viruses that they have been harboring, but it doesn't explain why bats able to tolerate these viruses without causing tissue inflammation and cell death. So we wanted to see that are there any other pathways that are regulating. So there is another pathway called pyroptosis, just like necroptosis. In this case, the pores are formed by a protein called gastermin. Okay, that leads to the activation of inflammasome. This is the gastermin pore. The structure has been solved. And it's been very well established, not only in SARS-CoV-2, in multiple pathogenic viruses, that the activation of this pyroptosis is directly associated with the disease severity. And especially for the release of IL-1 beta-IL-18 cytokine and and then leading to the respiratory epithelial damage. So we just asked a simple question: what if bats have an adaptation in the pyroptosis pathway, especially its executioner called Gerstenmann pore structure? So we come to this hypothesis because this Gerstenmann pore activation is downstream of most of the innate immune pathways. Not only that, adaptive immune pathways as well, that all of them can at the end can converge at the activation of this Gerstenmann pore. So to summarize our results, we did find that some of the bat species, they have very interesting mutations in the Gerstenmann pore structure, and that actually required for the pore assembly. So we actually model these things, and we see that bat Gerstenmann pores, they actually, when we introduce bat mutations into human pore, they actually destabilize the pore formation. And not only that, we actually characterize this experimentally by developing two cell-based assays where you can just express this Gerstenmann pore along with the caspase that actually cleaves between these two domains, and this N-terminus goes to the membrane and forms the pores. And we have seen that here when we express intact human Gerstenmann, it actually induces cell death, but when we introduce either single mutations of bats or in a combination, all of them were actually reducing the cell death activation in human cells. So we were very interested in one particular mutation called C191, which is cysteine. This Gerstenmann internal domain has 3 or 4 cysteines, but they don't form disulfines. One of them called C191Z, which is there on the surface of that. So we became more interested on that. We started characterizing. So for the characterization, what we have done is that in this case, we took only the N-terminal. If we just take this N-terminal and express in the cell, it spontaneously induces cell death. Now you can see here is that the cysteine mutations of bats, they actually do not induce much cell death. So we characterize this in 2 different cell lines. Not only that, this phenotype is not because of the expression of protein level differences. So, and also these cysteine mutations were affecting the release of cytokines like IL-1 beta, IL-18. So these cytokines, they generally do not come out of the cell. They need pores to be formed, or the cell should undergo lytic cell death. Then only they come out of the cells. So now you can see that having BAT mutations, they actually inhibit the release of the cytokines too. Okay, and then we asked why bats have these mutations. So I know it's good that bats having these mutations in the Gerstenmann pore, they will help them to have the viruses, which are so that they can induce less inflammation and they can harbor these viruses. But why did they adapt these Gerstenmann mutations first of all? Because bats must have adapted these for different reasons, and that is being used for hosting the viruses. So there recently, I think last 2 years, there many papers came out and show that the reactive actogen species directly regulate the Gerstenmann pore formation and pyroptosis. And this is something to do with the bats as well. So bats are the flying mammals. So when they fly, they generate lots of ROS, but keep in mind that they never undergo any kind of tissue damage, despite they have higher ROS levels than humans or many other organisms. So we hypothesize that what if the Gerstenmann adaptation in bats is because of their need to handle the high ROS levels in their during the flight system. So to this end, what we have done is that we used 2 different ROS triggers called rotenine and arsenate, which eventually trigger the ROS production from the mitochondria. We can see that this Gerstenmann pore activates cell death, and the moment you add ROS triggers, this cell death goes up. But when you have the BAT mutation, they do not induce cell death at all. Okay, and not only that, we tested. We wanted to see whether this mechanism is useful for the viruses to host in the bats, and we have seen that when we infected the cells which are bearing BAT mutations in the Gerstenmann, they do show less cell death with SARS-CoV-2 as well as influenza infection. So to summarize this part, we have seen that bats have got adaptation in this pore structure compared to the humans, where they activate suboptimal activation of this pore formation, and that is leading to the less inflammation so that they can harbor the viruses. So to summarize our talk, and we could able to show that bats, they actually develop pathogenic traits in the viruses because they are like mammals and they have many proteins like mammals. Not only that, they got adaptations in the immune machinery or inflammatory immune machinery that makes them adaptable for hosting the viruses. So this is the last slide. Not only that, we are working on recently happening pandemic H5N1 infection all over the world. So you might have heard about that. In US, it became a chaos because it infected the cows. It shook the cow industry. So this H5N1 has been there for about 1990 onwards. But again, this suddenly a new variant of H5N1 has emerged as a pathogenic strain. And people don't know why this sudden emergence of H5N1 appeared. And not only that, now this has jumped to a lot of mammals. And generally, this H5N1 is happy to live in birds. They generally don't propagate or replicate in mammals or humans. But this particular strain has been moved to a lot of mammals and it infected humans. And that's a concern for people that it can become a next pandemic. So in this study, we have shown that this particular H5N1 gained a very unique mutations in its hemagglutinin and neuraminidase. Like it has got unique pattern of glycosylation on its surface and unique pattern of neuraminidase, which has a long stack that leads to the evolution of this particular strain that helped them to spread to the multiple mammals. Okay. With this, I'll stop and I thank all my lab members and collaborators, and I'm happy to take questions.
Thank you, Keshav. You completed well on time. Open for questions. Yeah, first one right there. Can somebody pass the mic?
Yeah. So, should I go ahead?
Yeah.
Yeah. Hey, Keshav, yeah, very nice talk. And just a curiosity, this is just a variant of bat that you were talking to. And you were articulating evolutions. Have you looked into the evolutionary tree and keep wondering that why you do not have those variants in higher mammals, or maybe we do?
Okay, I think we did look at it, but these mutations are specific to being seen in bats, but this particular cysteine has been mutated in some of the patients too. And they have a different kind of inflammatory disorders. Wow. Especially they are more prone to the bacterial infections than the humans who have intact cysteine.
And any IL-1 beta linked to it?
Yeah, definitely. Because since the pore doesn't form properly because of the cysteine mutation, it definitely affects the IL-1 beta release as well.
Great talk, doctor. Have you noticed any cellular pattern in the pellet of the bat or the cartilaginous portion of the bat? Because you see, all the RNA virus, including the rabies, involve those pellet areas, and there is a predominance of Merkel cells on those. Have you seen Merkel cells proliferation with all these pyroptosis changes in your study?
Yeah, we haven't done, but there are other people have done this kind of things. They do see a difference in them.
Murali?
Yeah. Beautiful talk. But so the question is, what you're telling me is that the bats actually establish some kind of chronic infection, and whatever happens, we don't know anything about the adaptive responses there.
Yes.
But what happens to very early type 1 interferon innate mechanisms? So my question there is that, are there some defects there, and as a result, are we getting interferon resistant viruses that can jump from bats to other mammals? That's my question.
Yeah, that's possible that they can develop interferon resistant viruses because bats do have at basal level high level of interferon than humans and many other mammals. And not only that, they have a multiple copies of interferon genes, like duplicated than human beings. So they naturally have high interferon levels. Whether this is because of their harboring virus or their intrinsic, we don't know yet, but they do have it.
Okay, you have a question?
Yeah.
That is the last one.
Yeah, quick one. So pretty much follow up to what's already been asked. So you've mentioned that bats have this tolerance to the virus they harbor, but I wanted to ask, from an evolutionary standpoint, is harboring virus, say, a side effect or an outcome of the mechanisms they had to balance the redox biology that you noticed in the cell, or is it that nature wanted them to harbor viruses to maybe spread across places? So do you have any insights into
Yeah, I mean, this is definitely a big question that people are trying to understand, but what I can answer based on the current knowledge is that I think it's the bats' adaptations for their flight movement and their high metabolic needs, and that became a suitable environment for viruses to be standing in them. So that's the current understanding, but it can be changed.
Thank you,
Thank you
Keshav.
very much.
It's a very wonderful presentation.
Okay, our next speaker is Dr. Sudhir Pai Kasturi from Department of Pathology and Laboratory Medicine, Emory University. Dr. Kasturi obtained his Bachelor's of Engineering degree from Manipal Institute of Technology and then got his PhD from the University of Texas at Austin. And his lab has been developing carrying out preclinical studies using non-human primates and rodent models for testing new vaccines. He has designed a novel adjuvant delivery system that enhances the longevity of HIV vaccine-specific antibody responses. And one such adjuvant is actually currently in phase 1 clinical trials. And today he's going to talk to us about programming durability of vaccine-induced antibody responses with TLR7/8 targeted adjuvants. Dr. Kasturi.
Thank you. Thank you, Dr. Nandi and team for a wonderful IMMUNOCON 2024 and the invite to share our work here. Let me start off by introducing, I don't know in the audience if everybody is familiar with adjuvants. After having lived through the COVID-19 pandemic, I think this is more important now that we invest in researching adjuvants. Adjuvants are simply substances or ingredients that you mix in a vaccine inoculum that can simply strengthen vaccine-induced immunity. Okay, these are natural or synthetic molecules. They have it's well established. It's researched over 100 years now. They can improve responses to protein-based vaccines. As pointed out by Professor Amara previously, that live vaccines, such as against smallpox, yellow fever, and even measles, one shot, one vaccination can potentially give you long-lived or lifelong immunity, and that doesn't happen with proteins. So you need adjuvants. Adjuvants can improve immune responses in multiple different ways. It can improves the rapidity of, it can increase the rapidity of how fast immune response evolves. That's critical during a pandemic, so that's important. It can enhance immune responses in immune-compromised people, in aged people, and particularly I would like to highlight about the need to reduce the number of immunizations, right? So there's increasing vaccine hesitancy that's induced by the COVID-19 pandemic, and hopefully adjuvants, better adjuvants that are continuously researched can help us reduce the number of vaccinations by enhancing the longevity of vaccine-induced immune responses. And that's something that my lab is interested in. To understand how adjuvants work, it's important to appreciate the different phases of an immune response where adjuvants can make an impact, right? So adjuvants and the adjuvants, the flavor of inflammation induced by adjuvants are important in jump-starting an immune response, for example. Adjuvants can influence by stimulating dendritic cells to really trigger very strong T-cell responses. Specifically, it's important for us to appreciate adjuvants that can potentially give rise to very strong follicular T helper cells. Helper cells are important there to trigger germinal center responses that are extremely critical in viral vaccines, right? Germinal center responses are extremely critical to ensure B-cell affinity maturation. You need that to cover viral variants. That's important in influenza, that's important in SARS-CoV-2, and that's important in HIV as well. One area that I'm particularly interested in is understanding how adjuvants can program the induction of plasma cells, right? We have Professor Ahmed in the audience here who has established a really important paradigm that plasma cells that successfully migrate traffic to the bone marrow and reside there for very long periods of time are extremely critical. To understand how adjuvants can influence them, I think is a really important question. And we had the good fortune to collaborate with Professor Ahmed in my postdoctoral days with my mentor, Dr. Palipalendran. We worked closely with Rafi's lab to understand if adjuvants can influence migration of plasma cells to the bone marrow and mediate long-lived immunity. And that's where we were really, really excited about working with TLR7/8 class of adjuvants. And it appeared at that time that this was a feature of this class of adjuvants to really drive very strong frequencies of plasma cells that were not only seen at the peak time point, but were quite stable for at least 1 year when we had the opportunity to really look at these responses. Look at what happens with alum. This got me interested, and that's what I'm going to talk for and present for the rest of my talk today. Alum, which is an important adjuvant, it was one of the first adjuvants discovered about exactly 100 years ago. Okay, so it's approved for use in about 30 plus clinical vaccines. But in our hands, with this particular GP140 HIV envelope protein that we used, it gave us pretty good blood ASC responses, but we saw pretty poor and weak or almost non-existent plasma cell responses in the bone marrow. So that raised an important question. Can we understand if there are changes or if there are qualitative differences in the quality of blood ASCs that we've induced with these two different classes of adjuvants? Can we ask that important question? And that's where I invested some time, and I'm going to present that in an independent study that we set up as a follow-up. But let me introduce what is this TLR7/8 agonist. For people who are unaware, it's Bharat Bio. We've all lived through the COVID-19 pandemic, and India has the distinction of approving one of the first TLR7/8 adjuvants that was part, I don't know if people are familiar, but a TLR7/8 adjuvant was used in the Covaxin vaccine that was introduced by Bharat Biotech, right? So India has the distinction now for getting this approved. We worked with 3M Pharmaceuticals in the US. They had worked actively in designing this particular molecule called 3MO52, which is hydrophobic by nature for a reason. The scientists designed this molecule so that you could minimize systemic toxicity. You could minimize the systemic dissemination of this compound. This compound was designed so you could effectively formulate it because of its hydrophobicity and lipophilicity. And of course, there's a new theme that's emerging. It's actually an old theme, but that's been revisited by multiple groups in the US that have highlighted that vaccination strategies, slow release vaccination strategies that could potentially lead to very strong germinal centers and long-lived immune responses. Perhaps 3MO52 can contribute to that as well, owing to its to persistence because of the nature. I would like to highlight also an important finding from the published work that I just shared with you, that it was really interesting to look both at the peak and at the durable time point, the enrichment of vaccine-specific plasma cells that were found in the bone marrow among the total pool of immunoglobulin secreting cells. In comparison with what's known with human vaccines, so against influenza or tetanus or even with SARS-CoV-2 infection, as shown here on the right-hand side, if you compare these numbers, these are quite high. And of course, this is given there's a distinction between using a non-human primate model. It's highly relevant because the immune systems are quite parallel. The take-home message here is adjuvants potentially can contribute to improve the overall frequencies of plasma cells that not only arrive but stay stable in the bone marrow. And that's exactly what I wanted to test in an independent follow-up study to what we've already published, okay? So we set up an experiment using the non-human primate model using state-of-the-art, the latest envelope trimer immunogens representing clade A, B, and C of HIV, and we adjuvanted those immunogens using alum or the TLR7/8 agonist. And we vaccinated animals in a standard dosing regimen. And the goal here was very simple. We wanted to now sort and harvest these blood ASCs. Blood ASCs come up after boost vaccinations. Again, quite well established by Professor Ahmed's lab and Jens Rammert. We were interested in investigating the transcriptional makeup of these cells. We were interested in understanding the phenotype of these cells. Do we see clues that are different between blood ASCs that are induced by these two different classes of adjuvants? Can we find out if they have chemokine receptors that facilitate migration, increased migration to the bone marrow? And more important, there was a wonderful presentation by 10x Genomics yesterday. So we made use of that technology, and the goal was to resolve immune responses at the clonal level, right? So the idea was to then try and track these clones right into the bone marrow to understand how vaccine-specific plasma cells are distributed, right? So we suffered some setbacks. Everybody knows COVID-19 pandemic shut everything down, but we used this opportunity and turned this into a positive. So we waited, and we could now monitor the longevity of this immune response in the study. And we revaccinated the animals. So now we use this unique opportunity to revaccinate after 2 years, okay? And now we could use, we resorted these blood ASCs, and now we could compare the clonal makeup of if every time you vaccinate animals, in this case, do you recall the same clone? What is the transcriptional makeup? And do they traffic every time you vaccinate the bone marrow? So it was a very unique opportunity that we thought was very exciting to pursue. It was a highly collaborative project with Professors John Moore and Maurice van Gilt in Netherlands, who kindly provided the immunogens. Maurice is a close collaborator who's helping us understand memory B cells, and also helps, and I'll touch upon monoclonal antibodies that we've isolated from this study. Steve Bossinger is at the Emory Primate Center and School of Medicine, who's got considerable expertise in B-cell informatics. Okay, so highly collaborative. Let's jump right into the data that resulted from this study. So once again, we reproduced what we have shown in our published work, that yes, indeed, you could generate very durable responses, okay? Much higher with the TLR7/8 adjuvant in comparison with alum. But something really stood out. It was very unique. If we calculated the half-lives and the decay kinetics, the decay rate of this antibody response, it was surprising. It was not that different. So alum is not a bad adjuvant, right? So your TLR7/8 can increase the magnitude of responses, but the decay kinetics are very simple. That's the first take. In HIV research, you have different classes of antibody response. You have what's easy to neutralize, so it's a tier 1 antibody response, and what's difficult to generate, which is tier 2 autologous neutralizing antibody response. Now, that's where the distinction exists between the two different classes of adjuvants, right? So 3MO52, or the 3 TLR7/8 targeted adjuvants, was able to not only induce but sustain responses against those difficult to neutralize autologous, those tier 2 viruses, and whereas alum failed, and it was extremely transient. So we have to be very careful when we distinguish these different classes of adjuvants. Now, if we also break down the half-life calculation into multiple intervals, this is where we were also able to appreciate that the difference, if at all that existed, as shown here, was in the early time points, which highlighted now these early time point antibody responses are contributed by blood ASCs, right? So that makes that blood ASC population that much more exciting to investigate as hypothesized, right? So we did just that. We isolated the blood ASCs. This is an established technique. We know the kinetics, we know the phenotype now. And here, as shown, yes, CXCR4 is a chemokine receptor that helps lymphocytes traffic into the bone marrow, and that was significantly enhanced when using the TOL7/8 adjuvant in comparison with alum. We also saw a significant enhancement of CXCR3, and we have some data now that there's a specific subset of plasma cells that carries the CXCR3 receptor on its surface. And there's also reports that it could perhaps play a secondary role, not only to guide cells to the sites of inflammation, but also to the bone marrow. And here are some quantified numbers. Yes, it is indeed significant, significantly higher when you use the better adjuvant in comparison with the clinical grade alum adjuvant, right? We also looked at the total transcriptional makeup of these blood ASCs sorted at that peak time point. And we saw, not so surprising, but very strong type 1 interferon signatures in these blood ASCs that are extremely enriched. Okay, so that's well established. If you run LS spots at this time point with these cells, there's greater than 50 to 75% of these cells that are vaccine specific, that are HIV envelope specific in this experiment, right? So in addition to type 1 interferons, we were also able to appreciate there were several genes that were significantly higher in the blood ASCs when using the TOL7/8 agonists that have been previously reported in mice or even in human experiments that are critical for a better quality, such as CD93, BAF, several TNF superfamily receptors. And my mentor, Balapalendran, has previously reported that some of these molecules, some of these genes at early time points serve as biomarkers for the efficacy of the yellow fever vaccine. So we've now gone to the source of these antibodies and reproduced those signatures in vaccine response enriched blood ASCs. We use the single cell technology to investigate, are these blood ASCs quite heterogeneous, right? So are there different subsets that we can appreciate? And as you can see, there's at least 4 different clusters that stand out. And I'll highlight in a second that it is actually this red cluster that you're seeing that carries most of the vaccine-specific antibodies that are predominantly IgG. The rest of them have a mixture of IgG and IgA, and this orange cluster is quite interesting. There are the phenotype of these cells match that with what's been reported by Professor Ahmed and Ali Bedi in the humans, in human vaccines, that these are activated B cells that could perhaps commit to the memory lineages. It's quite puzzling why we are picking them up in blood ASCs. And here are some canonical markers validating that we have identified the right population of blood ASCs. We've validated that our signatures, even with single cell resolution, match what we have shown with bulk RNA sequencing. The signatures stand out. You have type 1 interferon, you have CXCR3, as I pointed out, and so on and so forth. And as highlighted in the paper that we've already published, we were able to recap again and reproduce that, yes, vaccine envelope-specific responses are significantly enriched in the bone marrow. With that, okay, a few slides. And we've spent time and invested with time in also pulling out subsets of plasma cells. We've done morphological analysis. We have validated that these subsets of plasma cells indeed are vaccine specific. By using that, this is what I wanted to present. This is what I would like to call the money slide for the experiment, right? So this is where we've really done clonal analysis, which shows now that the blood ASCs that were found in migration contained a lot of clones that were subsequently found in the bone marrow. And the clonal overlap that we saw between blood and bone marrow was substantially enriched when you use the TLR7/8 agonist as hypothesized, highlighting in part one mechanism that the adjuvants might be influencing effective migration of the blood ASCs arriving in the bone marrow. Keep an eye on the top. These are blood ASCs from what happened 2 years ago, right? So we still find overlap between those ASCs and the bone marrow, potentially highlighting that we can identify LLPCs in the bone marrow that were found circulating in blood 2 years ago. One last slide. We've also improved the resolution of these responses using serum antibody proteomics, where we have pulled envelope-specific antibodies down and run mass spec to identify bona fide envelope-specific clones that now we can overlap with the database that we created with the blood ASCs that allowed us to select antibody sequences to express them as monoclonals and then test them. And we had remarkable success where we chose 84 antibody sequences at random, and 81 of them were envelope binding. And of course, a subset of them, at least 30, had very strong neutralizing activity as well. Okay, so in conclusion, I hope we have shared that yes, adjuvants can impact how antibody responses are maintained long-term by influencing trafficking and influencing qualitatively blood ASCs that are induced early on that migrate to the bone marrow and persist there for very long durations of time. I'm going to skip the future directions. Please feel free to come and talk to me about what else is going on in the lab and what are the follow-up studies that we are setting up to build on these findings. And I would like to acknowledge Arpan Pradhan in my lab in collaboration with Amit in the Possenger lab who led these studies, and all collaborators who helped us with all the primate studies that are ongoing in the lab and the funding from National Institute of Health. So happy to take questions.
Thank you very much. We'll have 2 questions. Yeah.
Thank you very much. So it was a beautiful talk.
Thank you.
What you say is somehow familiar to me. I work in allergen-specific immunotherapy. We use alum. What you present is basically the finding for a basic course of immunization and the follow-up. And it's clear that when you use a more powerful adjuvant, you will, of course, more load the bone marrow with the first shots, and then you will have, of course, a longer memory. However, in allergen-specific immunotherapy, we give boostings, regular boostings, very often. And in immunotherapy with ARDOM, we see the rise of, after the boosting of another IgG class, subclass, it's IgG4, and this is long-lasting, and it builds up slowly but steadily and remains high, whereas the IgG1 goes very strongly up and then goes down. And what we see is this twofold course, and I'm just wondering, what would happen now if you would use your schedule in a booster scheme, where you have not just one course, but you would boost regularly?
We've done, so if I understand the question right, you're asking me about the boosting schedule, right? So if that has an impact on accumulation of cells in the bone marrow, is that what you're asking? Yeah, I mean, we have varied the intervals in multiple follow-up studies. The short answer is, if you boost with these adjuvants that we are seeing, the number of cells in the bone marrow keep building up. I don't think the intervals, we pull the intervals out. It impacts the quality of the immune responses, but regardless if we are boosted early or later, we've continued to recruit memory-derived blood ACs that have successfully gone into the bone marrow, and they've stayed there. So I don't think interval matters as much, but it's an important question. And you also highlighted IgG4, right? So in our experiment, we have seen predominance of IgG1, and that is classic because of the adjuvant that you're using, correct? So we've not seen a dip in IgG1, and we've not seen IgG4 come up. I think it's a consequence of the adjuvant that we've used.
Yeah.
Have you ever looked at the T cell response, particularly CD8?
I'm sorry?
Have you looked at any T cell response, particularly CD8 response?
Yes. You get extremely poor CD8 T cell responses when you use protein-based vaccines with even with these adjuvants. And we've done experiments in mice first. You need to formulate the antigen to enhance cross-presentation of proteins to elicit CD8 T cell responses, but we have failed. We haven't seen any appreciable CD8 T cell responses in non-human primates, in larger species. We've heard it's the same problem. If you've looked at human clinical trials with HIV vaccines, we don't necessarily see CD8 T cell. That's to see good CD8s, you need viral vectors. To my knowledge, that's the best. Now, I think with mRNA vaccines that have been introduced, I think you're seeing much better CD8 T cell responses. But adjuvants and protein-based vaccines, we don't induce very strong CD8 T cell response.
Okay, very nice talk, Dr. Kasuri. Just a small clarification. The adjuvant used in Bharat Biotech Covaxin is called L-hydroxyQM or something, from a company called Viravax.
Correct.
Is the same as this?
It's not. It's the same class of compounds. They both target TLR7 and 8.
Right.
This is highly lipophilic.
So this has not been used in a approved human vaccine.
It's been used in human clinical trials as we speak. In fact, they published the first findings this week. I don't remember the journal, but 3MO52 is being used in clinic, but it's in clinical trial. That's why I highlighted L-hydroxyQM jumped ahead, and Bharat Biotech has the distinction of approving the first TLR7 class of adjuvants in humans.
Thank you. We have to close. Sorry.
Hello, sir. Sir, as I studied that matrix M is a very good adjuvant in case of HIV. So is there any comparison like you did between alum or 3M052? What you will suggest about matrix M or this 3M022?
Yeah, I wish I had the opportunity. Novavax is a protein-based vaccine that's been introduced now. Finally, it's not FDA approved, but there's an emergency approval for Novavax, which carries the matrix M adjuvant. My understanding, I think my mentors just published a paper comparing the 3M adjuvant with matrix M in the context of malaria. It's a Science Translational Medicine paper that was published last month. I don't remember the exact numbers off the top of my head. I think 3M still does slightly better than matrix M. We've worked with other groups that use that particular class of QS21, the saponin-based adjuvants. I mean, we're still working out the details, but I think the TLR7 class of adjuvants perhaps has a slight edge over matrix M. So we'll know in follow-up studies when we do the comparison. But there's a paper out there by my mentor.
Thank you, Dr. Kasturi. We'll stop.
Yeah.
Let me invite Dr. Kaja, Murali Krishna Kaja, for the last presentation of this session. Dr. Kaja is a alumnus of IASC, and he is an associate professor with Emory University, and also he is the associate director of ICGB Emory Vaccine Research Program. His laboratory interest remains in viruses, vaccines, and protective immune response evaluation.
This is the cursor, right? Yes. And this is the back and forth. Yes. Yeah. All right. Thank you. Thank you for introduction. And thanks to the organizers for giving me this opportunity. Yesterday, Dr. Dipankar Nandi said it has been 30 years since the IMMUNOCON happened at IASC. Actually, I'm probably one of the few people who had this wonderful opportunity of presenting 30 years back at IASC in the IMMUNOCON, and now at that time as a PhD student, almost at the end of my PhD. And thinking of the time, it's 1:30. I'm sure that you are all hungry. I'll try to be less than 15 minutes. And thinking about the food, and Rama likes to go to AMS and Keshav go to CMS, and I used to go to CMS actually, but not at the end, not at the beginning. I used to sit almost all the time there. And so I'm going to talk about some of the interesting findings that we are making on dengue, human immunity and disease. Actually, this talk could have been given by either Anmol or myself. Anmol is traveling today, so I am giving this talk. So I don't want to spend too much of the time, but because all of you know about dengue problem in India, I'm sure that all of you know that there are 4 serotypes. It can cause hemorrhage and shock and death and mosquito-borne, and we don't have yet licensed vaccines and therapeutics. But I just want to focus on these 2 problems. We don't really understand what are the correlates of protection and what really contributes to protection versus pathology. That has been the major problem in our inability to have effective vaccines, I think, from last 30 to 40 years. And but then there is some significant things that developed in the field. It came almost around the time when I was born, actually. Scott Halstead did these studies in Thailand in very highly dengue endemic area. At that time, he found that almost all the dengue severe cases were secondary infections. And that led to the idea that, primary infections are usually not at all dangerous, and secondary dengue infections with a different dengue serotype is likely to be the most dangerous thing, high risk. And that's been a feast for immunologists, because that conceptually, it defies all the fundamental immunology of that, how we learnt about it. The memory, comes up and should protect us. So, but then there were so many attempts based on this kind of idea and try to make vaccines, design vaccines and implement, and so many challenges that happened in that whole process in the last 40 years. So, and one of the things, is that unlike many parts of the world where dengue has been there for centuries together, India, although has had dengue sporadic outbreaks, the dengue took off only from the last 20, 25 years. And so dengue is pretty recent in India. And so we were very interested. At that time, I think around about 10 years back, I'm kind of diverting from my talk, but it just came into my mind. Actually, myself and Dr. Rafi Ahmad visited here in St. John's Hospital during the early part of dengue season. And then what we found is very interesting. Anita Sheth was our collaborator, clinical collaborator. We went to a pediatric ward, and then we saw like 50 mothers sitting on the beds, and each one of them have a small child or infant, all very young, for 3 years, 2 years, 1 year children, and all with either hemorrhage or shock or something like that, or close to the dengue warnings. And then, but that was very different picture than what we had seen in the adult medicine department, very fewer adults. So then we wondered, what's happening with the vaccines at that time. The few vaccines that are there, most of the vaccines are being undergoing clinical trials only in the adults or advanced age children, or that 1 or 2 vaccines that are actually licensed in some of us, for example, Sanofi, is recommended only for children more than 9 years age. And assuming that more than 9 years age children are the ones who are likely to be already exposed to dengue virus, and those are the ones which are at higher risk for severe disease. So this whole thing didn't make any sense, but nevertheless, we wanted to, our biggest strength is to understand human immunity. And when we started the ICGB India program, Emory India and ICGB program, we put a major emphasis on understanding the human immunity to dengue. And in the following eight years or so, we did a lot of studies on understanding, starting from innate immunity to B cell responses to T cell responses, and trying to map just the human immunity, and with an assumption that we would see all these responses going big bang in secondary infections or severe cases, assuming that the severe cases would be more or less synonymous to the secondary infections. And that's not at all what we have seen, in fact, and we could really not interpret what's happening. So that's where we thought that let's take a step back and try to understand what actually happens to severe disease or disease in India between primary and secondary infections. So the next 5-6 minutes, I'm just going to focus on only those studies. And the studies are published recently, and I may not be able to go through every bit of the data, but if you are interested, you can look at this paper. So basically, there are 619 dengue patients, and they were recruited. This is the analysis, and they were from 3 different hospitals from all 3 parts of India: Delhi, AIIMS, and Bangalore, St. John's, and CMC Vellore. All are children around 7 years age, and all were analyzed during the acute febrile phase of infection. And then I don't mean by we, our clinicians, wonderful clinicians, they characterized the disease based on the WHO criteria: mild dengue and dengue with warning signs, which is intermediate and severe dengue. And then in the lab, we did classify primary as secondary infections based on the WHO criteria, but using very well standardized and calibrated pan-bio capture ELISAs, which are highly calibrated and used all over the world. Basically, they would characterize primary infections if the individual doesn't elicit IgG at all, or only IgM, or a lot of IgM but very little of IgG. So this is the first summary of the result. So we have 619 children. And then we first asked, what is the proportion of the cases with severe disease, severe dengue disease? Pretty high actually, 32% of them had severe disease. And now if we divide these all cases into primary and secondary dengue infections, we actually did not find much difference. It's basically, it didn't matter. Primary infections has pretty high amount of severe disease, and secondary infections had pretty high amount of severe disease. that was pretty shocking for us, because it's not telling me what has been conceptually out there in the field from last 50 years, and what's the most of the vaccine approaches as well as vaccine implementation approaches are based on. So we thought, let's look at it a lot more carefully. So one thing that we did is that we increased the stringency of how do we classify primary versus secondary infections. So here we use the usual ratio, but here we use much more stringent ratios, and basically, a lot of IgM here in this group. And even if we put that extraordinarily stringent ratio, we still see pretty high proportion of severe cases in the primary infections. And of course, similar with the secondary infections. And then we thought, let's go one more step more stringent. And then because if you take very early cases, at the early points after onset of symptoms, if it is primary infection, they should just make only IgM antibody. There should not be any IgG. And if we just look at those, so we looked at the patients who are less than day 4 post onset of symptoms, and they didn't have IgG using the dengue specific IgG using the PanBio capture ELISA, but they had IgM, and even there pretty high proportion of severe dengue cases. And and then we thought that let's look at, based on what I said about our experience, just by looking at, severe cases in the children ward versus adult ward. So let's look at the distribution of cases, by age. So if we look at very youngest children, obviously almost 100% of them primary infections. And then as you go later, it's a mix of primary and secondary infections. That's not unexpected. Soon after birth, it would be primary infection. But if you look at the disease severity, these youngest children are the ones who had actually the highest disease severity. And those are the ones who need the vaccines. Those are the ones who need the therapeutics. And we are completely ignoring them in our vaccine trials and going with higher age. And then, of course, we look at the older people. They are not just completely made of secondary infections. They are also had a mix of primary and secondary infections. And if you take even the 4 to 16 years old age group, and if you take only severe disease cases of 4 to 16 year age group, again, half and half of them are primary versus secondary infections. So it tells that there is a lot of primary infections have severe disease. And during this, there was seven unfortunate fatalities in Delhi site, and we happened to have their first sample collected at the time of recruitment. So we did a careful analysis of their primary versus secondary infection status. So basically 5 out of 7 fatalities were primary infections. It's not just the severe disease. And then, so we thought, let's look at the neutralizing antibodies, which is a lot more stringent criteria, but it also tells us what happens to these children depending upon primary versus secondary infection. So these are all different serotypes. And then we are looking at the neutralizing antibody titer, and these are secondary, and secondary. Obviously, they have very high neutralizing antibodies. And these are primary in different serotypes. So primary infections are the ones actually are not making the neutralizing antibodies, which is expected. And the secondary cases actually make ton of neutralizing antibodies by the time they reach the hospital. But they still are suffering from severe disease. So there is something going on with the mechanisms, and we don't know what it is. So we thought that let's go ahead and look at it, because serologically, serum tells us what happened, from last few days or past few months. But if you look at the B cells, if you isolate the B cells, it will tell us what's happening right at this moment. So we took the primary and secondary patients' blood and then stained B cells, sorted plasma blasts, and then used our high throughput human monoclonal technology platform here. we made human monoclonals, and close to around 200. Nearly half of them are primary and the other half monoclonals came from secondary patients. You look at binding to different dengue serotypes, the secondary patient-derived antibodies, which are the red color here, bind pretty good, whereas the primary patient-derived monoclonals bind very low. And the same thing gets reflected in terms of neutralizing titer. So again, it tells that the B cells that are taking place there in the secondary patient, they are making pretty good neutralizing antibodies, but the patient is still suffering from severe disease, at least some of those patients. So that's where, where we are going. And so overall together, it tells us that, whatever the dogma in the field, which is basically the severe diseases associated with secondary infections, our findings are questioning that. And much of the vaccine developing and implementation efforts currently are based on this dogma. So these findings have to be put into that picture, and we have to try and readjust, our approaches. And so that also brings something very interesting questions about it. In all these primary cases, there is not even an antibody. So where is the antibody-dependent enhancement coming up? So what is the mechanism by which the severe dengue is happening? So we have to go back and revisit, understand what are the mechanisms of pathogenesis and how do they diverge from primary versus secondary infections, so that we are taking a holistic approach in the next few years, try to look at different aspects of innate and adaptive responses. And so I'll end there. And a lot of people contributed, either directly or indirectly, from many institutions in India, many hospitals in India. Our major collaborators are from AIIMS, St. John's, and CMC. So I want to thank all of them. But more importantly, I want to thank children's parents and who actually consented to take their. It's not easy to consent to take your child's blood for research when your child is going through hemorrhage. And I really want to thank them. So any questions, I'll be happy to address.
Thank you, Murali, for completing on time. Open for any questions?
Thank you for beautiful presentations. I have just two very quick things that I would put in your mind. The first is you have a gap in the very young children, yeah? And is it a possibility to vaccinate adults, the women, to drive the titer up very highly and to hope that some of the antibodies will go to the blood of the children and at least protect them in the first few months of life? Just for you to think. And the other thing is about the severe disease. Despite high antibody titers, we have found in COVID there actually the formation of immune complexes. And this depends on the ratio of the antibodies versus the antigen. And then, of course, antibodies can bring a chain of viruses to the cell.
Yeah, I mean, those are all good points and very relevant, something to consider. But there are other factors to consider too. We think that most of the dengue studies about the antibodies, and they came up with the idea that, antibodies does this and enhance the infection and all, but they are just, we humans are not like a test tube with only antibodies. We do have T cells, we do have memory B cells, which undergo, which get activated and go on recall responses. So I think there's some of these things need to be lent and as well as put into the applications when we actually come up with the ideas. Yeah.
So I have another question. So I'm here. For the severity, actually, some serotypes are also responsible, like held responsible. Maybe dengue 2 is more severe, and also what is circulating at that time in that area. So if you can, number one, tell us like what was the serotype circulating. Then second thing is, there is a role of NS1 also recently kind of proved. Have you looked into the NS1 secretions as well in these samples?
Yeah, very good questions. So here is the serotype data. It's basically, just to summarize it, I don't want to go through all that thing. In our study, we have dengue 1, 2, and 3. Dengue 4 is very, very few cases, so I can't make any interpretations. But in these 3 serotypes, we basically see pretty high proportion of primary infections having the severe disease. So the second thing that you asked is about the NS1. Yes, absolutely. A lot of people and many studies come up with the NS1 could be the most, could be the pathogenic by causing the endothelial dysfunction. But the thing to consider is, it is obviously is very important, and a lot of studies show that. An intriguing thing is that because we were trying to kind of interpret some of our results, and then what we found is that most of the secondary infections, which actually are supposed to be at high risk for severe disease, they have a lot less NS1 compared to the primary infection group, which actually indicates that the virus is replicating a lot higher in primary infections, and which is not surprising. They are not making antibodies. They don't have pre-existing memory cells.
Can you keep it brief?
Right?
Can you keep the answer brief?
Yeah. So I think, so at this stage, I think all those things need to put together and then look at it carefully.
Thank you.
Yeah, I'm not trying to conclude anything about it. I'm just telling what we are observing.
But second, after that, Chair.
Yeah, I have a very quick question. The secondary infections, and you had a cohort, and any comment on the separation between first and the second infections, and if it gave you any insight?
No, the studies that we did are not a longitudinal cohort.
No, but the time separation in those individuals, were there a consistent time separation between primary or secondary, or in some cases the primary infection may have happened 2 years before, in some cases maybe 2 months before?
No, these are all the cases that are recruited at the same time. Basically let's say in this ongoing dengue season, if you recruit it, and then we divide them into primary and secondary.
Yeah, the secondary was determined based on IgM-IgG ratio.
That's right.
Do you have any other information beyond that to articulate what exact time point when they were primarily infected?
No. We did not take the previous history, and I think it's not reliable.
Yeah, Murli, yeah. Very nice talk. Just 2 short questions. So what is the overall mortality rate among these children? Among 600 odd, you said, right? Only 6 or 7 died in the entire thing, or?
See, the mortality rate depends upon the medical situation, right? In countries.
In this study.
Yeah, in this study, we only had this 7 cases,
7, okay.
So it's percent-wise comes very
7 out
low.
of 600.
Yeah, they are in the hospital.
Yeah.
So yeah.
And the other question is there any link between malnutrition of these children and severity of disease? Is there any data?
It's a good question. We didn't really look at those things, at least in this study. Ranjan Nanda and ICGB is trying to do some proteomics, and hopefully we will try to understand something.
Thank you, Murali. It's a wonderful presentation.
Thank you.
See, if other speakers come on the stage, we'll take a photo. I'm trying to save time.
Delete that.
We will delete all presentations. Thank you to the speakers and the chairs of this session. A gentle reminder for the speakers in the afternoon session to upload the PPTs with the technical team during the break. We request everyone to please attend the poster sessions and also proceed for lunch now, and also visit the company stalls that have been put up in and around the auditorium halls. The participants who have been assigned slots for posters today are requested to put up their posters and have their lunch before the evaluation. We kindly request everyone to reconvene sharply at 3 p.m. so that we can start the next session. Kindly go over the schedule for the next session, and we have an exciting afternoon planned for all of you.
It belongs to God of Chakravarti, Professor, right? Please collect it from us.
Welcome back, everyone. I hope you have enjoyed your lunch and are all refreshed and ready to continue. Our next session has 3 parallel sessions happening simultaneously in different halls. For those of you staying in the main hall, we have an exciting session lined up titled as T-cell biology. For those attending the parallel sessions, you may now proceed to Hall A or Hall B. Please refer to the event program for specific details on the topics in each hall. The main session will continue here in the main hall. I invite Professor Sarath Dalai from Nirma University to chair this session.
Welcome all of you to the afternoon session. Our first speaker for this session is Dr. Sudha Kumari, who is currently Assistant Professor at the Department of Microbiology and Cell Biology at Indian Institute of Science. Her work focuses on investigating the mechanism of immune surveillance in lymphocytes at molecular and cellular and organ scale using interdisciplinary techniques. Previously, she was research scientist at Darrell Irvine Groups at Koch Institute, MIT. There she devised imaging methods to assess immunogen trafficking to lymph nodes during vaccination and lymphocyte infiltration within intact solid tumors using cancer immunotherapy. Prior to this, she carried out her postdoc research with Michael Dustin at Oxford, NYU, where she studied the process of T-cell activation in autoimmune and immunodeficient diseases. So, and Dr. Kumari also did her PhD from NCBS, so it's Satyajit Mayer. Welcome, Dr. Kumari.
Thank you so much, Professor Dalai. Thank you so much for inviting me and giving me this opportunity. How does the... All right, yeah. Okay, sorry. Hi everyone, so I'm Sudha. Where I started my... Okay, good. So where I started my lab very recently, where I combined techniques. It's highly interdisciplinary lab. We combine techniques from hardcore cell biological techniques, immunotherapy and immunology to investigate how T cells interact with other cells, talk to other cells during immune response, how these communication channels fail when some specific diseases develop, and how do existing therapies actually support these communication channels. Our system of choice is largely using reconstitution systems to understand T cell signaling at the cellular and subcellular level, but we do expand them often to tissue and organism level for a better scale understanding of these questions. So today I'm going to talk largely about the cell biology of T cell activation, primarily because it's a fascinating area, but also because I do want to contribute a different flavor to the meeting. So For healthy immunity, T cells must perform immunosurveillance, which entails that T cells can go long distances in the body, migrate everywhere, can infiltrate the most complex organs. They migrate around scanning different cells for presence of antigens. Okay, it's exemplified in this movie, where you can see the T cells are in green in the lymph node, and they're aggressively migrating, scanning the other cells, including the red-coded antigen-presenting cells for presence of antigen. There's no antigen here, okay? So see how aggressively they migrate. If there was an antigen, then the T cells would arrest. And they will form something called an immunological synapse, which is a specialized cell-cell conjugate interface across which there is crucial transfer of biophysical and biochemical information. And a T cell will never be the same after forming the immunological synapse, okay? And since all the recognition process in adaptive immunity are dependent on the physical scanning and synapse formation, this is arguably one of the most important event and structure that lays the foundation of adaptive immunity. Okay, so one very interesting feature of immunosurveillance is that all of these steps, migration of T cells to long distances, complex tissue infiltration, scanning of other cell types, forming transient interactions for scanning, they all rely on a very specialized and unique cell biological properties that immune cells have. In fact, it's specifically afforded by them. In developed metazones, you won't find many cell types that can actually migrate around like that. And if they migrate, there is a problem. Okay, so immune cells are uniquely equipped with cell biological properties that enables immunosurveillance. The property that I'm very interested in is actin cytoskeleton, and I'll describe briefly why. So when I started studying T cell, there was a very interesting gap that I found. So this is a highly simplified model of T cell activation, where a T cell sees antigen on antigen presenting cell. Of course, there is a whole host of other molecules that have to engage in this process, but for the sake of simplicity, this engagement happens. And T cells form immunological synapse. At synapse, there is a lot of actin polymerization and rearrangement that goes on, and somehow that's associated with calcium flux. T cells get activated, they disengage and migrate on to perform effector functions, to perform memory, and so on and so forth. So when I started this journey, it was known that actin polymerization is very important for calcium flux. Why? Because there are a number of contexts where a genetic lesion in actin regulatory protein affects calcium flux, and there are immunodeficiencies. But it was not known how actin actually does that. How does actin help in calcium flux mechanistically? Okay, and it's a complex question to ask because actin is important for everything. It's important for adhesion, it's important cell to migrate on. What is special about actin here so that it's enabling calcium flux? So to understand that, I decided to go to these actin-related immunodeficiencies, genetic lesions in actin regulatory protein that result in defects in T cell activation. I worked on a number of these deficiencies in collaboration with Professor Raif Jaha at Boston Children's Hospital. But my most favorite molecule is viscotaltic syndrome protein VAS, causal agent of VAS. So why? So truly special context. So viscotaltic syndrome happens when the individuals lack VASP, and VASP itself is actually expressed exclusively in immune cells and platelets. And it's one of the molecules downstream of antigen receptor that can mediate actin polymerization. So actin, you can imagine it's like a Lego piece. Okay, you have a lot of monomers, and then you can polymerize it to generate different architectures. And people believe these different architectures actually have different functions. Okay, so one very interesting feature of the viscotaltic syndrome was that there are some definite phenotypes that T cells showed there. One was T cells see antigen, but they do not flux calcium. They engage synapses, but they're not stable. And there's very high incidences of malignancies in individuals that have VAS. So I said this is a natural perturbation system where there's a very unique opportunity to understand perhaps how actin polymerization or polymerized actin actually mediates calcium flux, might stabilize synapses, and perhaps, if you're lucky, also contributes to tumor immunity. So let's stage each of these phenotype and see what's going on. So it's easy, right? So what you do is you take T cells from VAS individuals and see what's wrong with actin there so they're not fluxing calcium, right? In theory, yes. but in practice, a little bit challenging question. And the reason for that is that synapse is not easy to image. T cells are highly round, dynamic, tortuous, and this is a classic synapse assay. If you put an antigen-presenting cell and T cell together, what we saw, others have seen that too, that the actin levels shown in the gray here are actually identical in the normal T cells or T cells that lack VASP. Okay, so then I argued that if content is similar, it still doesn't tell us much. Because if actin is like a Lego piece that's going to generate architectures that may be responsible for function, then we should be looking at architecture and not just levels. So what is the architecture of actin here? This is not a good assay to look at that because you can't really see what's going on inside the synapse. To look at synapse, then I deployed the system that was originally pioneered by Mike Dustin, my postdoc co-advisor, which is nothing but you make a fake antigen-presenting cell, lipid bilayer that is reconstituted with molecules derived from antigen-presenting cells. These are ligands. And one very sort of interesting, very important feature of this system is that 3 critical parameters that dictate T cell activation, namely the dose, affinity, and diversity of ligand, can be very precisely tuned in this system. And of course, it's planar, so it's amazingly amenable to microscopy. So T cell sees this system, forms immunological synapse, and now you can couple it with the microscopy technique of your choice and see what's going on in the synapse. When we do that, so now we're looking at synapse from the bottom, not the side, okay? So a single T cell synapse seen from the bottom, the footprint, the junction of the antigen-presenting interface. So first thing I noticed was actin is not uniform because in the cell-cell assay, it looked like a belt, right? So in fact, this was the first examination of actin at this detail at the immunological synapse of a primary T cell. And guess what? These actin heterogeneities actually are associated with signaling T cell receptor clusters, so much so that they even co-migrate, okay? So active T cell receptor clusters are associated with this actin enrichments. So I decided to call them foci, and these foci are not there in the VASP patient T cells. So it seems that VASP generates these foci. VASP also seems to be important for calcium flux. So do these foci help in calcium flux? Turns out yes. And so I did a series of pharmacological and genetic perturbations to build an understanding of how exactly this foci could be helping in calcium flux. And turns out that when the antigen receptor is engaged, there's a whole host of molecules that are recruited. And also eventually VASP is activated. It forms actin foci. Actin foci directly enrich PLC gamma 1, which is a mediator for calcium efflux from ER. If you do not have VASP or if you do not have foci, in either of the case, PLC gamma 1 activation goes down. And even though you have early signaling going on, antigen recognition going on, you do not flux calcium. So fantastic. If following the first phenotype, we arrived at a basic understanding of how calcium flux may be boosted at the immunological synapse. So encouraged by this, we decided to follow phenotype 2, which is VASP deficient T cells do not stabilize synapse. So we decided to look even deeper within this small interface, just 5 micron across, of a primary CD4 T cell. And what we found was that the foci that look like dots in light microscopy, in electron microscopy, it shows an interconnected network of highly tense filaments, and that is lacking in VASP deficient T cells. What do these filaments do? They generate a lot of tension, so T cell is not able to disengage from the synaptic state. So this led to a mechanical model of how T cells stabilize synapse, which is that highly aggressively migrating T cells, when they transiently arrest, they lay down this actin architecture, which is very, very tense. And the only way it breaks is that T cells actually eventually downregulate VASP. When that happens, this whole network collapses and they can migrate on, okay? So it's actually a symmetry breaking problem. It's actually very unique to actually immune cells because the time scale at which you engage and disengage these contacts is actually not very often seen in physiology in other cell types. All right, so second phenotype, unstable synapse led to understanding of synapse stability. Now third phenotype, much more complex, right? I'm sure you'll agree with me. Malignancies you can't understand very easily using a unique mutation. However, we decided to take it to give it a stab anyway. So for this, we switched from CD4 to CD8 T cell system because CD8s can directly recognize and kill tumor cells. So it's a sensible place to start. So first we decide to check whether the findings that I've seen in the human patients can be replicated in mouse model, and they can. If you implant tumors, then they grow out much faster in wasp knockout animals compared to the wild type animals. But that can be a problem in any immune compartment and not necessarily T cells. So for that, we switched the model and we took the wild type animals and then implanted tumors and then tried to change the tumor growth rate by adoptive transfer of antigen specific, either wild type or wasp deficient cytotoxic T cells. And when we do that, then it's very clear that wasp deficient CTLs indeed are a little bit compromised in their ability to tackle tumor growth. So there's a problem in CTL compartment. So then I actually went after where the problem could be, and turned out the problem was everywhere. So cytotoxicity was an issue, activation was an issue, cytokine secretion was an issue, degranulation was an issue. Turned out the problem was actually the most fundamental step of antigen recognition itself. So very first of antigen recognition was compromised. And when I looked at actin in these cells, the CD8 T cells that are just seeing the antigen, okay? Here the foci look very different from CD4 T cells. They actually have a Z aspect to them, almost like comets, right? They've been described in serology as comets. Comets, what do? They generate forces. So it led to actually a mechanical model of force feedback between the antigen-bearing cell and T cell. So what we think is happening is following. So T cell, initially few receptors are going to see peptide MHC on the tumor cell, will lead to wasp activation, foci formation, foci are gonna push. If this force is resisted by tumor cell, if the tumor cell is hard, then there is a mechanical benefit to the other T cell receptors, and you have more and more and more T cell receptor activation that feeds into foci generation and then more. So this feedback mechanism keeps on going. However, if tumor cell is soft or T cells lack wasp, this system crashes. You may have biochemical signal present, you may have peptide MSC present, but T cells are not going to recognize efficiently. Even the T cell receptor phosphorylation goes down. Okay, so this was actually quite fascinating to see that the initial force exchanges and the actin organization can actually affect very early steps of the antigen recognition itself. So again, encouraged by this, we decided to go even deeper into the T cell cortex and see what kind of actin organization actually help in decision making after initial antigen search, scanning, and recognition. So I will take 3 more minutes to just tell you a little bit about it. So we found that it's very surprising, a surprise, an enigma, and a drug. So we found 3 things. So first, let's go through the surprise. So I'm sure anybody who's worked with lymphocytes has seen that there are surface projections, the surface very tortuous in lymphocytes, right? So classically, they are considered to be microvilli, okay? So, and there's a prevailing hypothesis in the T cell activation field that the T cell receptor actually sit on the tips of these microvilli, and it helps in efficient search, fast search actually, that is still pretty accurate. Given our data on CTLs, I hypothesize that perhaps not all of these are microvilli, and a sizable number of them are these protrusions that are made by WASP and R2-3 pathway. So we could test it by just doing regulatory perturbations in the molecules that generate either microvilli or these protrusions. And when we did that, we took T cells from different knockouts, from knockout animals where microvilli are supposed to be compromised, or animals where we should have the other protrusions compromised, and we found that it's neither. Actually, there's a third kind of organization that regulates the dynamics of T cell cortex so that they can efficiently search, find, and recognize the antigen. So that after that surprise, we were quite baffled that, okay, so the T cell cortex seems to have a very different organization than previously believed. Let's revisit synapse and see whether the dynamics of actin still holds up. So what's known about the synaptic actin dynamics? So the Kuffer model of synapse is that the antigen receptors, they start in the periphery, they will end up in this signaling dead zone. So each antigen receptor has a couple of seconds to signal, and then finally it's spat out. So Genial Griffith and Mike have done really good work on that. And there's a nice actin flow, this centripetal flow, that antigen receptor, engaged antigen receptor clusters bind on, and they go to a signaling dead zone and they spat out. All great. Then you go to the primary T cell. Now the system changes. So I hope that you can appreciate that while the flow here is all centripetal, here there are two components to the flow. One flow that's centripetal and one distinct flow that goes outwards. In fact, it drags 40% of antigen receptors outside. Okay, so not all antigen receptor is degraded, 40% is not. And what is enigmatic is that it still rides on actin. Okay, so what is enigmatic here? So if you think about a river that is carrying, like the actin flow is like a river, is carrying the antigen receptors to the center. Now you have two actin flows simultaneously in time and space. Can you imagine a river that has two flows in space and time? It's actually extremely enigmatic phenomena. So to understand that better, thankfully at ISC we have a lot of departments. So I reached out to my colleague in physics, Sumantra Saka, and he actually found a fluid dynamics explanation to that. You can have simultaneous flow, both antigen receptor as well as actin at the synapse if we actually use those understanding. So I think we have a solution to that. If you want to understand more about it, I'll be happy to talk about it. So by now my students were like, Sudha, I think there's too much going on. There's a lot of confusion. Lot of actin organizations previously unknown that we are discovering. And some organizations are causing projections, some organizations causing flow, all using the same monomer, right? So to investigate further into these processes, we need better handle on these organizations. We need to be able to perturb actin polymerization in space and time on demand, where we want, subcellular level. Of course, supercellular is a little easier. So, and none of the existing perturbation methods actually cut it, okay? So to do that, Srishti Mandal in my lab, which is one of my first graduate students, took on a big task of actually generating this inhibitor. And we used light as a tool to exert control. So we now have a light activable, photoactivable actin inhibitor. And because light has really good resolution, you can scale the inhibition from few sub-micron scale to the supercellular scale, to the organismic scale, wherever you want. And at whatever time scale you want, when you want, okay, on demand. And it has a lot of benefits. It's just one of the examples of this system. So this is what I'm going to show you is a actin dynamics movie of Jurkat T cell synapse. And this is a lot of actin flow that goes on. It has inhibitor. The moment you shine light, there's change in dynamics and cell contracts. So now we can actually do subcellular, spatially defined, temporally controlled actin polymerization inhibition. And using this inhibition, now we can ask questions that are outstanding in the field that has never been asked because the perturbation methods are lacking, which is how do the cell topologies actually guide T cell decision making in the first few seconds of interaction between the T cell and the antigen presenting cells. These are very, very critical for affinity discrimination between actually telling for the fundamental property of the adaptive immune system to tell apart different affinities. So with that, I'd like to close and thank everyone. I've only spoken about the synapse work, but we have other interesting areas in the lab. Hopefully next time I'll get a chance to share that. And thanks to the group, these guys make it happen. So thank you all.
Thank you. Any questions, please? Can you give me mic there? While mic is, I have one question maybe before. Dr. Sudha here. Can you hear me?
I'm sorry.
Here.
Oh, okay. Hi. Sorry.
I have a question for you. When T cell get activated, many TCRs have to be engaged, and this is where the immunological synapse plays a critical role. That is for T cells to be activated. But when T cell is going to kill the cancer cells, do you think that many TCRs need to be engaged with MSC peptide on the tumor cells?
When the T cell is going to be cancer cell itself?
Yeah. It only requires for recognition. It is not required for activation. Already activated T cells are going to engage with the tumor cells.
Yeah.
What would be the requirement of MSC?
I mean, my easy guess would be that it will be less. I think we have actually some data on preactivated T cells. But I mean, it's a very, very interesting question because the proliferation and T cell activation are often coupled. But there are contexts when they are not. So you may not need T cell activation for proliferation also. So if you're talking about the uncontrolled proliferation, then may not be. I mean, it's not necessary that you need it. When Jurkats are a perfect example. They go on dividing without the. So about the number of T cell receptors that are required for T cell activation, turns out even that is okay. Even that is very few. Apparently, even 10 T cell receptor engagement can cause a full-blown T cell activation because there are a lot of amplification mechanisms in the early signaling.
Hi. Yeah, I was interested about the WASP activation mechanisms. I mean, are there any role of integrins, for example, in the DCs in terms of activating WASP in T cells? And do you recapitulate that in your artificial antigen presenting cell surrogate?
Yeah. So we routinely put integrin ligands here. Certainly ICAM1, but sometimes also VCAM. So this activation of WASP has baffled us too. So it's not classical CDC42 dependent activation. And Mike Rosen's work has shown that for NWASP, it's just clustering of NWASP can cause activations like phase segregation. And it might be that for WASP also, because WASP has a lot of SSC domains and it can bind to a lot of early signaling proteins at T cell receptor cluster. That might be the mechanism for activation, and then it starts to,
So we may take one more question.
Hey, Sudha, wonderful talk. It's a WASP-mediated calcium control, and as I gather that puts these T cells in the intimate contact of cancer cells. And coming to the first questions, if it is not required for TCR activation per se, would that probably engage a lot of co-inhibitory molecules and do the checkpoint controls or any thought about it?
Yeah, I think there is a very, very interesting question again. So the coupling between the calcium signaling and exhaustion has been investigated before. But between WASP levels and exhaustion hasn't been. So actually, I don't have a clear answer to that. Yeah, I don't have a clear answer to that.
So very nice talk. Looking at that, the dynamics of T cell activation in naive cells versus memory cell or effector memory cell. Is there any difference in this kinetics of actin filament with WASP? Anything is known?
So no. So subcellular actin dynamics measurements are extremely challenging. Very few groups do that. But what is known is that the migration patterns as well as the synapse duration is different. Naive cells engage longer synapses, memory less. In fact, even between CD4, CD8s, there are differences, and those are actin dependent. So I would think there is difference. But quantitatively, mechanistically, that has not been.
Okay. We'll go to the next speaker. Our next speaker is Dr. Ritubrat Goswami, Associate Professor at IIT Kharagpur. He's working on T cell differentiation. And his lab is basically focusing on understanding role of specific molecules, including micronutrients, steroids, in the differentiation of T cells. And in fact, work in his lab has explored how fat-soluble vitamin A and D, as well as estrogen, regulate Th9 and Th2 differentiation. Today, he's going to talk about homeoboxin containing transcription factor HOXA9 regulates differentiation of T helper cells. Please.
Very good afternoon. Thank you, Professor Dalai, for that kind introduction. And I also extend my cordial thanks to Professor Dipankar Nandi for such a generous invite and having an IMMUNOCON of this scale. And I also thank for your time and attention. The afternoon sessions are really challenging. So I hope I can do a little bit to make you awake. And as you can see, I have changed the title of my talk quite a bit, but the essence of the talk remains the same. So we are going to talk about one particular transcription factor that belongs to the HOX family and how that plays an important role in CD4 T cell differentiation. So needless to say, for this talk, this slide really doesn't need any space, especially after all the talks we started hearing since yesterday, and of course today from Vijay Kuthru and Professor Wilfred. But I still do not want to resist this opportunity to skip this slide. All you need to know is that naive CD4 T cells, they get differentiated into different T helper subsets. I apologize in advance because I have not put all of them. And the main factors which are responsible for this differentiation would be mostly the three things apart from the antigen recognition by these CD4 T cells. And those will be the cytokines present in the microenvironment, the epigenetic modifiers, we had to talk about that. And of course, it's going to be the transcription factors which are expressed in that vicinity. So the transcription factor which I'm going to talk belongs to the HOX family. Now it's a transcription factor which has been associated with development. So if you see, so these HOX genes, they're conserved and they determine the course of embryonic development in animals. And these studies actually, if you go back maybe almost 50 to 60 or 100 years back, when the studies regarding development happened, people would believe and people would see that up to a certain level in the embryonic development, the frog embryo and maybe the human embryo, they would look same. And when the scientists worked on them, they figured out that it is because of the HOX genes. So in vertebrates, the HOX genes have been duplicated twice, and that's why you have 4 clusters. And these are for A, B, C, and D. What you can see here is the 4 clusters in chromosome numbers in humans. What you can also appreciate that over evolution, some of these genes have been lost out. And so at the end of the day, in vertebrates and humans, we have 39 out of these 4 clusters. What is also important to note is that among these 4 clusters, there are 3 paralogs, the 4 paralog, the 9 paralog, and the 13 paralog, which are present in all the 4 clusters. And so again, this is basically a cluster of genes which are important for the determination of the general body plan, starting from the anterior to posterior. So HOX 1, 2 would be the anterior, 3 to 8 will be the central, and 9 through 13 would be the posterior. So my interest would be talking about one of the family members, which is HOXA9. And so this slide summarizes the discovery in the HOX field research over the last more than 100 years or so. More importantly to figure out, is that we have in the 1983, the structural determination of these HOX came into the picture. And in 1985, you have these HOX clusters which have been identified in human mammals. So moving forward, so if we look into the roles of HOX genes in immune systems, if you just modify the classical diagram of Bob Weinberg and Robert Hanahan, you could see that various members of these HOX genes are involved in various types of cancers and impacting various reasons for having those cancers. And on the other side, what you can have is the various members of these HOX family members. They are important, or they play a critical role in angiogenesis, vascular remodeling, and inflammatory response, out of which you can see that HOXA9 is one of those molecules. So if I take HOXA9 separately and see what exactly it is doing in the immune system, and again, there are lots of literature which shows aberrantly expressed HOXA9 and various different malignancies, and they can impact various phases, which would include apoptosis, EMT, drug resistance, and probably recurrence. And along with that, you can also appreciate the expression of various transcriptional factors, other regulators, cytokines, which will be aberrantly expressed in these different phases. So now, when we looked all this literature, all these studies, we also wanted to say how this HOXA9 works and interacts with additional partners. So very classically, HOXA9 interacts with another class of homeodomain containing protein, which is PBX1. They can form this dimer, and to stabilize this interaction, sometimes you can have a trimer including another homeodomain containing protein called MEIS1. So you can have the formation of this trimer over here. So we actually looked into a study which came from Nick Lucas's group about 7 years back, where they actually looked in one of these paralogs, which is the HOX5 paralog, and they saw that this HOX5 paralog genes modulate the Th2 cell function. So based on that information, we looked what exactly was the expression level of HOXA9 across various different T helper cell subsets, and we could appreciate that the Th2 cells express a good amount of HOXA9. So what we did in next was we took Th2 cells and retrovirally ectopically expressed with HOXA9 constructing viruses. And you could see that when you do that, we could see a decrease in IL-4 secreting cells. And that is also true when we look into the MFI as well. So what happens with the other cytokine, interleukin-13, which is also a critical cytokine expressed or secreted by Th2 cells? We see the similar thing. We could see a decrease in IL-13 production when HOXA9 is overexpressed in Th2 cells. So now, if I show you the constructs of these HOX molecules, this is the domain structure of HOX, and you could see that the main domains which is responsible the function of HOX would be this homeodomain and the hexapeptide motif. And in this hexapeptide motif, you have this invariant expression of the tryptophan amino acid. And there are different 3 helices which are part of this homeodomain, helix 1 and helix 2. So homeodomain is going to be binding to its specific DNA sequence. Helix 3 is the one which will be responsible for that binding, and that binding is going to be stabilized by helix 1 and helix 2. So we constructed few mutants in the lab, which where we actually changed the tryptophan to alanine. This is the mutation W1992A. And then in one of the mutant, we removed the helix 3. And in one of the mutant, we removed this region, which is intrinsically disordered. And in the final one, we actually kept this one intact and removed both the hexapeptide motif and the homeodomain. So when we do that, we also have to keep in mind that these 2 cytokines secreted by Th2 cells, interleukin-4 and 13, they not only share, they actually share the receptor as well, so which is kind of known to everyone, but they also share a conserved region between them. And that's why you can find that there is a core ECR, which is about 649 base pair, which is located between interleukin-4 and interleukin-13 gene. So we did some, we took this information and we constructed these IL-4 and IL-13 promoter in luciferase expression vectors with or without this core ECR region. And what we can find is that when we have A9 transfected, we do not see any change in the activation of IL-4 promoter. But if we remove the helix 3 domain, we could see not much of a change. But when the IDR region is there, so that means we could see complete opposite effect. But if we take the IL-4 promoter along with the core ECR, very interestingly, we could see that HOXA9 is actually interestingly transactivating the promoter. But if you take either of the Del H3 or the IDR mutants, those activation is completely lost. What happens in IL-13 promoter? So IL-13 promoter, it's interesting again, because A9 now it is decreasing the activation of the IL-13 promoter. And if you take those 2 mutants, those decreasing activation is getting lost. But it changes again when we are having the core ECR along with this IL-13 promoter. We do not see any change in the activation mediated by A9, but if you take the IDR mutation, that is completely going to be opposite. In fact, it gets increased. So the other cytokine which is expressed by Th2 cells is IL-10, and if we overexpress A9 in these Th2 cells, we could see surprisingly an increased level of IL-10. And when we do the reporter assay using the similar constructs, what we could see that IL-9, HOXA9 is really not changing that much in terms of the activation of IL-10 promoter. But if you take these mutations, surprisingly, the DLH3 mutation is reducing the activation of IL-10 promoter. So we could see very distinct role of different domains of HOXA9 in these 3 genes. So we've delved into further and tried to figure out what exactly is the mechanism behind this regulation of Th2 cells mediated by HOXA9. And we could find out that HOXA9 is not interacting, or it is not really interacting with the master regulator of Th2 cells, that is the GATA3. Interestingly enough, in Th2 cells though, when we look for HOXA9 interaction with P1, which is responsible for the heterogeneity of Th2 cells, we could see that there is actually an interaction between HOXA9 and P1. And we did the same thing in the presence of a DNA sequence to which this P1 would be binding, and we could see that HOXA9 is actually occupying the P1 binding site. And for that binding, you really need to have the intact the homeodomain for this interaction. So you have seen that I've mentioned that HOXA9 interacts with multiple cofactors. PBX1 is one of them. And PBX1 is, the cartoon of PBX1 is kind of shown over here. So this is also a homeodomain containing transcription regulator. This has a 3 amino acid loop extension that is kind of shown over here. And we also check the expression of PBX1 in various T helper cell subsets. And like what we had seen in the case of HOXA9, PBX1 expression was also higher in Th2 cells compared to the other 2 subsets. And this we verified using the immunoblots. Interestingly, when we do the overexpression studies in Th2 cells using the PBX1 retroviral vector, we could see there was actually an increase in IL-4 production and also in IL-13 production. So when we did the experiment using the luciferase assays to figure out what exactly is happening at the promoter, we could see the effects are very different. So this we have already shown. And now if we just activate the construct with just PBX1, you really do not see any change. But with together A9 and PBX1, you could see that there is an increase. But if you take the IL-4 core ECR region, which we saw with A9, if we put PBX only, we could see that there is a decrease, and the HOXA9 fails to change that this decrease mediated by PBX1. And we saw a very interesting observation in IL-13 promoter with or without the core ECR region. So we could see that PBX1 by itself could be upregulating the activation of IL-13 promoter, which can be mediated or downregulated by A9, which is kind of shown over here. And the similar thing is kind of shown in the core ECR region as well, where A9 is negatively impacting the role of PBX1 in regulating the activation of this PBX1. So if we overexpress again, PBX1, when we did to check the level of IL-10, we could also see an increase of IL-10 production as well. And when we do the reporter assay, what we found was that PBX1 was also enhancing the IL-10 promoter activity, and A9 was not changing that when A9 and PBX1 were put together. So in terms of potential interaction of PBX1, what we could find out in our lab that PBX1 could actually be interacting with GATA3, but we have not been able to figure out whether PBX1 is interacting with P1 or any other Th2 specific transcription factors. So what exactly is happening in this whole story, which I have kind of zipped through in the for the sake of time, is that for both IL-4 and IL-13, one thing we have not really shown is how these histone modifying enzymes are relevant in regulating the gene. So we have an interaction of HOXA9 with P1, GATA3 is interacting with PBX1, and they are differently regulating the IL-4 and IL-13 gene expression in Th2 cells, which would be additionally regulated by histone-modifying enzymes. But for IL-10, the roles may be different. We could have the relevance of PU1 directly, or it could be the difference in the histone-modifying enzymes, which would be responsible for this different regulation. So this work has been carried out by Shayanti in the lab, and she has been supported by Shagnik. They also had their own posters yesterday. And the funding has been provided by ACRB. And this is the whole group who is part of the team, and I'd like to thank for your kind attention.
Thank you. We have time for 2 questions, please.
So the DNA binding assays that you showed, did you actually determine if HOXA9 is directly interacting with particular DNA sequence, or it's a protein-protein interaction?
So DNA, that was the DNA affinity precipitation assay.
Right, but that doesn't distinguish between direct DNA.
Right, so it's a very good question, and we are trying to do those experiments in the lab. It's a very relevant question.
Thank you.
I think your interesting story. Have you tried to overexpress both molecules? So who would win?
That is something which is ongoing in the lab. Okay. So it is a developing story, so I don't have all the data, but I thought of sharing something which is going on, and hopefully we can have those data soon and probably get back what exactly is going on. Because it's interesting, we don't know, because we don't know what exactly is going to happen with MEIS1 into the picture in this Th2 cell, so then maybe we have to do triple transduction assays. Very challenging, but we'll see.
Okay. May I just add, have you tried, do you have an antibody where you can stain HOX9? Do the cells which express IL-4 have the higher levels of?
Precisely, we are doing that. We are sorting this based on the expression of IL-4 high and IL-4 low. And again, I'm afraid I do not have all the data to show, but very good. We are doing those things.
Okay, thank you.
Thank you. If there is no question, yeah. Thank you, Ritu. So our next speaker is Dr. Nimesh Gupta. In the interest of time, I'm not actually introducing his full biosketch here, but he's basically a viral immunologist working on T cells. He was really famous during COVID time. Did a lot of really good work on T cell immunology in COVID.
Yeah.
And in fact, last year he got mid-career scientist award. So with this, I invite Dr. Gupta to deliver his talk.
This is forward. Perfect. Thank you so much, Professor Dalai, and a very good afternoon to all of you. I'll start thanking Dr. Dipankar Nandi and the team for this wonderful organization of, I have a bad habit of moving out of the podium, but let me try. So I think this is at this scale of IMMUNOCON. I think it's a fantastic organization by Dr. Nandi and team. So I think we must congratulate him, and I'll ask him to have a SOP available, after the meeting is over, so that we can have the good organization of such meetings in the country. So what will I do is in next 15 minutes or so that I'll share the glimpse of some of our work that we are developing in lab in understanding T cell, B cell collaborations, in virus infections and controlled human vaccination. And the major emphasis of these programs are mostly on the biology of follicular T helper cells at the scale of both antigen specificity and at the single cell resolutions. So I think here what I'm sharing today is the program on dengue. And I must thank Morley. I think I need not to go into the details of the introduction because he has very nicely introduced about the dengue aspects. I'll just highlight one or two aspects here that among almost all mosquito-borne infections, if we see dengue is probably one of the most widespread infection. And recently there is much more concern in the health agencies when it has reaches to Europe. I think we have been talking about mosquitoes available in European countries now, those who have the capacity to transmit the virus. But I think now we have also cited some of the autochthonous dengue cases in the European region. So it's probably bringing to a stage where I think we are looking at the development of dengue vaccines, which has been challenging. I think it was a major challenge in the field, but with much more knowledge now, which is available from the human immune system. So this is, just an aspect of dengue in India, which is mostly coming from the Delhi NCR region. And I think if you see the scale of, basically the magnitude, the scale of infection, even in Delhi NCR, which is actually endemic, is actually amazing, right? Because every 2 years, every 3 years, we are seeing the expansion in the cases. And that rise in cases is continuous. And I think Murli has mentioned that in his talk, that this we have started seeing in past 2 decades. So we can imagine from a highly endemic area, which we talked about primary, secondary infections, we have majority of secondary infection, still there is a huge rise in the dengue infection. And I think epidemiologically, we always see the dengue infection in these regions from August to November. It is something which is coinciding with the monsoon season in these part. But now there is also an extension of these months of the transmissibility of the dengue virus. And I think it is ranging now, reaching from July, and now it is touching even November. And so I think this is a major concern now, even for the health agencies to control, particularly the spread of this, the virus. So if we talk about the factors, which are responsible for dengue severity, I don't think we have a well-classified factors which can correlate or which can associate with the dengue severity. What we know is there will be genetic factors, there will be virus-associated factors, and there are also the immunological factors that probably either work in hand-to-hand or there have some dominance of some of these factors in bringing the severity to an individual. But majority of these knowledge came from the clinical studies which were done in World War II time by Albert Sabin. And I think he at that time did some of the challenge human infection models, and we got some information from there which has been carried forward till today. And I think slowly now we are getting into the details of those knowledge. So what you see here is, particularly when you have a homotypic infection, you are still protected, but that is actually limited to a particular time. And when you have an infection with the heterologous serotype, there are chances of getting severe outcome. There is a short-term cross-reactive immunity, even for the homotypic infection, which according to those 7 studies are reaching until around 6 to 8 months post the virus inoculation. And that wanes down, and then it brings out, to a possibility of severe outcome when there is a heterotypic infection coming out, from a particular, another serotype. Something which is interesting, which has been followed up in the field, even from those studies, and also later some of the studies in human infection model, that there are cases of heightened plasmablast. There are cases of antibodies that may target, our self, which can actually bind to the endothelial cells or causing some kind of autoimmune kind of scenario. And the antibody-dependent enhancement, which has been largely talked about. So all this information led to a scenario where we do know that the antibodies or the humoral responses play a key role in, let's say a virus pathogenesis or the immunopathological scenario. But many aspects of that humoral immunity, like how does that come out? What kind of cells, like the T cells, which play the role here, which are implicated in causing or leading to that humoral responses, were not much clear. Now, why it is important? Of course, it's important to understand the virus pathogenesis, the human response to the virus, but it's also important for the vaccine development, because for the rational development of the vaccine, we do need, some of the immunological parameters that can allow us to evaluate the vaccine at a very early stage of development. So some of these questions, we are trying to address in the lab, that, what kind of CD4 T cell subsets are actually implicated. Of course, the major emphasis is on follicular T helper cells, but we were looking with open eyes in terms of understanding what all subsets are actually implicated here in this immunity development.
and what kind of access of the T cell and B cell collaboration exists during the humoral immunity development to the dengue virus. So what we started with is, we made a small cohort. This is a hospital-based cohort of dengue patients, which was actually established in Delhi NCR region. It's an adult dengue patient cohort. And we actually followed them up multiple times, at least 2 times during hospitalization, 1 time during convalescence, 1 time at least after a year of the recovery from the symptoms, which could be either of 3, where we utilized, as Murali has mentioned this morning, we utilized the WHO criteria to classify these patients in 3 different outcomes of the disease: the mild disease, the disease with the warning sign, and the severe dengue disease. And of course, as we utilize those criteria and classified them in primary, secondary dengue infection, and we found around 64% of them with the secondary and 36% of them with the primary infection. So to just address first of our questions, we looked into the T cell activation, and this is more the T cell side of the story. What Murali has talked about, morning, he's looking more into the B cell side of the stories in the dengue patients in this area. So what you see is in these patients in the acute stage, you have like around 8 to 10% of the CD4 T cells, those who are proliferating. And these proliferating cells, if you see here, you find that they have majority of the activation markers, particularly like ICOS, CD38, as well as PD1, which is widely expressed on these proliferating cells. So we looked into the Tisne analysis of this subset, and what we found is that majority of these activated cells, because our major focus was on the TFH subset, which is actually, can easily be identified using the CXCR5 marker. What we found is that the CXCR5 expressing cells in these, proliferating population do not much coincide with the PD1 expression. So this is something which was interesting for us because we were expecting to have much more activation of the TFH population. That was according to 1 or 2 studies which were available in the field, not directly on TFH, but on the basis of the IL-21 cytokine, in the serum. So we then looked into the details of these subsets where we had more PD1 expression, but not much of the CXCR5. And what we found in the longitudinal analysis that first, a lot of activated population you will find in the acute patients when they are hospitalized. And when you follow them, these activation levels comes down to the baseline. And majority of these activated cells are actually PD1 positive. So more than 80% of these cells are actually expressing PD1. And that was interesting, observation for us because this was beyond a population in terms of the TFH as well as non-TFH groups, which we found in the dengue patients, which has so much high quantum of the cells expressing PD1 in the CD4 compartments. So we then tried to look into more details of it. I'm not putting up all the data here, but we importantly look into the different disease outcome. That does it make sense for a particular disease outcome in these 3 different disease scenarios where there is a febrile illness, when there is a warning signs of the severity, and when there is a severity. So what we found is that this subset, which is actually expressing a high PD1 in the activated population, is much more higher accumulated in the severe condition as compared to the TFH, which does not show much change in the 3 different outcomes of the disease. And then we further looked into the more detail because this was more into the activated kind of T cells in the blood. We then looked into the dengue virus specific repertoire. So we did all that, analysis at the antigen specific level. And, it was actually surprising that if you see this red color, this is mostly the antigen specific cells, which are actually PD1 positive. And it is the major compartment in the almost all dengue virus specific pool, which is expressed by the red dots here, which has minimal CXCR5 positive cells as well. And if you see their kinetics, longitudinal kinetics, in terms of the acute dengue patient, those who even recovered, you find them the similar kind of kinetics like what you see for the TFH cells, which are in red, but the magnitude was much more higher for this particular population. So we looked into more details. We also looked into their cytokine functions, the effector functions, multiple cytokines they are secreting, and we found that they have much more closer phenotype, the functional phenotype like TFH cells in terms of providing
help to the B cells. They secrete a lot of IL-21. They do secrete IL-10. So when we decided to link into out of that box that maybe they are making cytokines, do they also have some correlation with the humoral responses, not only the antibodies but also the plasmablasts? We found that these subsets do have a very strong correlations with the plasmablast population, and even the antibodies which are targeting to the different proteins of the virus, like NS1 protein or the PRM or M protein, envelope protein of the virus, and they have much more significant correlation with these antibodies as compared to TFH, which do not show much association with the antibody responses to these part of the viruses. So in the lab, we were actually trying to develop some of the assays to understand the T cell and B cell collaboration, and successfully we published one of the methods where we can actually identify the function of T cells to provide help to B cells at an antigen-specific level with only 90,000 T cells in the pool. So that is actually available both here as well as in the STAR protocols after a lot of demand from the community. So what we did, we sorted all this population from the blood, which were PD-1 negative or positive in both CXCR5 positive and negative compartment, along with appropriate controls. And what we found is that these cells, which are actually expressing PD-1, has much more similar capability to help the B cells to get differentiated into the plasmablast, like what you see in case of the TFH population. And this is in an antigen-specific setting. So then, this led to a question: are they somewhere, related to the bona fide B cell helpers? Are they kind of, some progenies or the outcome of the TFH population? Or they have some different identity, in terms of their different transcriptomic landscape, or in terms of, having a different or unique role in, at least in dengue or in many maybe other viral infections. So what we did is we tried to look into the more details at a single cell resolution of these cells in the dengue patients. So we took the cells from the patients, we sorted them on the basis of their activated phenotype, and we pooled them and we performed a 10x based a single cell genomics here, which was 5 primed, so that we can look into not only the gene expression of these cells, but also can look into the major aspects of the clonotype diversity, etc., as compared to the TFH population. So we found that they are highly heterogeneous. You can find multiple clusters here, which are ranging from around 10 clusters. And some of them are here, which is a TH1 kind of, which we were annotated on the basis of the gene expression analysis. And they were actually having a high PD-1 expression levels as we were expecting, but it were much more heterogeneous beyond our expectation as we were actually thinking to have a very close or a homogeneous population. And both in case of the RNA, the gene expression, as well as the protein expression, we found a very similar observation. So it was further confirmed that this pool, which is around 5 clusters, are coming from the TH1 kind of phenotype. And the cluster 2, which is mostly accumulating the TFH cells in the TCM cluster from these dengue patients. When we looked into the clonotypes, what we found is that they do not have a substantial or a significant overlap with the TFH clonotype. So you can see here the cluster 2, which is a TFH representing, and cluster 3, 5, and 6, which is actually representing more the TH1 kind of cells which are expressing PD-1, they don't show much overlap in terms of the clonotypes. So, of course, we then looked into more details of it, that if they are distinct, they are transcriptomically distinct, and they are also clonotypically diverse. So how does it make sense that they are actually helping the B cells, and how different they are in this heterogeneous population of PD-1 positive cells? So we looked into more effector functions of them, and we found these 2 clusters in which the cluster 3 showed more IL-21 positive cell accumulation as compared to the cluster 5, which was more granzyme B, the kind of cytotoxic cells in this PD-1 positive population. And this was also further validated utilizing an ex vivo stimulation assay, and we found that these cells are actually distinct even as compared to the cytotoxic population in this PD-1 positive pool, and there is a very marginal overlap of the granzyme B positive cells and IL-21 positive cells, suggesting they are a diverse population in this pool of PD-1 positive, which have the IL-21 positive traits. I will not go into these details, but we further confirmed that these cells are actually providing help to the B cells
via IL-21 axis. And that actually further confirmed that maybe in terms of a continent-dependent mechanism, IL-21 is the major axis which these cells are actually using. So just to conclude, what we are now working upon is trying to understand what is the biological relevance of these cells, because if they are coming into the same pool of PD-1 expressing cells along with cytotoxic population, they will also migrate to the different sites in the peripheral tissues, where they can actually lead to a scenario of cytotoxic cells to control inflammation, but these cells now have the function to help B cells. So what they exactly are doing there, that's the major question that we are trying to understand. And we have some interesting data which is now coming out where we found there very strong correlation with the extra follicular B cells. We looked into the dengue patients, and we also fetched some of the lymph nodes from the healthy individuals from the dengue endemic region, and we found that these cells do exist in these secondary lymphoid organs as a memory in the antigen-specific repertoire. So it means they actually, just to conclude, it means that they actually have a very devised and specialized function to help B cells, at least in case of dengue. They do actually have a very distinct transcriptomic and the clonotypic diversity and the divergence in their differentiation than the TFH as well as the cytotoxic population. And they have the capacity to migrate to the peripheral tissues where, now which is our hypothesis, is where they might be talking to the B cells in the periphery and might be leading to a kind of tertiary lymphoid structures or making a lot of antibodies in those tissues. With this, I'll just stop here. I'll thank my team. This work was majorly done by Asghar Ansari, who was the PhD fellow in the lab. Now he has moved to Pennsylvania University for his postdoc. And our footprint is there in the different areas of the world, but this was majorly done with our clinicians friends in AIMS in New Delhi, and also with Alex Sete and Shane Crotty in La Jolla, USA. And thanks to our funders, majorly the work was supported by NBM and the SERV, and of course for other funders, those who have been continuing to support and showing the faith in our programs in India. Thank you so much for your time. Thank you so much for your attention.
2 quick questions we can take. Any question?
Yeah, one here.
Yeah.
In your initial presentation as an introduction to dengue, you said 4 billion people are at risk for dengue. Does it not appear too much? Half of the global population?
Yeah, I think it is more than half of the global population, because I think these regions where, in the tropical or subtropical regions, the population is the highest. So I think if you see the number, even this number is undermining the actual number. So I think there's of course a surprise for even the clinicians and epidemiologists that, we are talking about majority of the global population who is at the risk of these infections.
So I have one quick question.
Sure.
So have you tried to do culture experiment where you take these PD-1 expressing T helper cells we are talking about and culture with B cells and try to understand how they are modulating the B cells?
Can you please repeat that question? I didn't get the first.
Have you tried to do any co-culture experiment where you have taken this type of T cells with the B cells and see how they are influencing?
So we did some experiments where we found that, majority of these cells are actually, helping the B cell via IL-21 axis. That's what some of the data that I was showing. And in terms of the contact-dependent phenomena, I think there could be, the multiple axes that might be implicated. So right now, we have developed some new models where we are trying to understand the biology of these IL-21 expressing PD-1 positive cells in case of dengue virus infection. And I think we'll have much more answers in the near future about the biology of these cells and their role in the dengue virus infection.
Okay, thank you.
Thank you.
Our next speaker is Dr. Ram Gopal Udupi. In fact, he's a physicist from Indian Institute of Science. And he has really contributed to the areas of de novo design of peptides and proteins, structure determination of proteins using weak analog signals. And in fact, he was responsible for structure determination of several immune receptor ligand complexes and used this insight to develop soluble version of mutant high affinity immune receptors as an alternative to currently approved checkpoint inhibitors. Please, floor is yours.
Okay, good afternoon. And I thank Dr. Dipankar Nandi to invite me, a physicist, and know a little bit, at least I can spell immunology. And yes, I worked on immune receptor extensively. And I also solved a lot of structures of this checkpoint blockade molecules. And later designed, because I worked on peptide design and protein design. Now I'm combining these two. It's a very recent work, and I just want to, this is for the students who are not, because there are a lot of people from interdisciplinary field, I think. When this is the first signal when a T cell Can you hear me now? Yeah. This is the first signal where a peptide loaded MHC is recognized by TCR on the T cell. Then the second, that is the stimulatory cell signal is required, that is through CD28 B72. And similarly, the inhibitory pathway is CTLA4 B72, or there are so many other molecules. And what is checkpoint blockade? You know, we already heard the first talk by Jim Allison, Professor Jim Allison. Basically, this is the pathway where T cell is suppressed by cancer cell, where they express more and more amount of PD-L1 or CTLA4, which are negative regulators, mostly PD-L1. And the first drug which came was basically a blocker of CTLA4 B72 pathway, that is the called Yervoy or ipilimumab. And this was the first in 2011 and 2013 already. There were so many molecules coming out of which block PD1 and PD-L1 pathway. That is this pathway, PD1 pathway, and already science announced its breakthrough of the year 2013. That means it is going to change a lot of things. And then comes the flood of so many drugs, which are mostly the blockers of PD1. And even though the other thing is, the President Jimmy Carter became the ambassador of, because he was saved at 86. And in another 5 years, you know, the Nobel Prize was also given. And I was fortunate to work with one of the Team 3 molecule with Jimmy Allison, Professor Jimmy Allison. And also I was fortunate to work on the structure of the first molecule, that is the ipilimumab Fab with the CTLA4. And that then if we are so successful with MAB, then is there anything to do, anything else to do? That yes, there are so many drawbacks, particularly when it comes to India, we only maybe 2% of patients can access this drug. It's so expensive, particularly in US, dollar sits around, in Indian rupee, it may be more than 1 crore actually. But now things are slowly coming, and we can get for lesser money also, but the thing is still expensive. Only 2% people can access this. And it's not only India, in many economically unfortunate patients around the world still can't access these drugs. And there are so many efforts, and there are also combinations which are also tried, and but there are still more to understand, because only small percentage of patients this is working fantastic. Okay. Now, having worked with so many checkpoint blockades, all the greens ones are the first structures I determined. At the time it was just structures for me, and this was the one I failed also. And even I can try to continue to work on these molecules here too. Then I decided, let me design this, because I also had some idea of protein design. Basically, what we do here in our lab is, now you say PD1 path, if now you're looking for this blocker, take the PD-1 itself, modify, make the high affinity PD-1, which will bind to PD-L1 tightly, and connect it to FC to keep its, say, renal clearance and other things as same as the antibody. That's the thing. And there are many other advantages here. That is, it is a smaller one, so tumor penetration is expected to be better. And generally, this kind of molecules that are FC fusion molecules, generally the minimum immunogenicity is seen. And we think that if it is, say, one day we can make it, it can produce in India, it can be economically better also. That's the idea. And and also other thing is there are so many drugs like this, that is FC conjugates, like say Etanercept, which is the TNF2 connected to FC, which is for use for autoimmunity. And most of these drugs are for autoimmunity and nothing for cancer. Basically, here what we are trying to do is PD-1 connected to FC, but PD-1 is made high affinity PD-1. For this, there are a lot of efforts towards this, which is actually directed evolution. There are 2-3 patents already. But being in a small lab with less money, I have to work on different, somehow they have to find a different avenue, way to make these molecules. What I did is, in this case, the Shankar worked on this pathway. Basically, he went through all the IgV domains from, say, antibody, T-cell receptor, adhesion molecules, ICRs, everything. Did the analysis to understand what is important for IgV domain fold to be intact. First thing he did that. And we found some of these, see, for example, if you see that this is the sequence conservation, and you see that some of these interactions, either E or Q, it is maintained in all the, for example, antibody heavy chain, antibody light chain, kappa, and so many, everywhere it is maintained, if you take the V domain. Similarly, you can see some of these interactions like the salt bridge. For example, in this case, you can see both arginine and aspartic acid is maintained through many of these families. And it's unpublished data. But I am not putting all the observations here. Shankar was able to find out that many of these conservations, which are, that tells that these residues we cannot touch. Because if you touch these residues, say we might even lose the fold itself, because they are maintained all through the evolution, has somehow maintained that all through the evolution. Why I should change it? Or even if you change it, it may be dangerous. That's how we first did the analysis. I am not putting all the results we found, but because of the time constraints. And similarly, other thing we did is, see, when an antibody, because we have to now design a molecule, modify PD-1, which has the affinity similar to an antibody. See here, we know that there are, say, 3 CDRs from IgV, VL, and other 3 CDRs from VH domains. Together we have 6, like 6 fingers catching a ball. Here, you can see that it is very different. It means one, if you imagine a forefinger and this, and it's just holding, it's like a clamp. Okay, but what is in the literature, interestingly, because in most of the antibodies, the CC prime loop, what is in blue here, is not discussed at all. Because all the CDRs are discussed, this is not discussed. But when we come to the IgV domain interaction, which is seen in most of the immune receptors, this is the CC prime which plays a bigger role. That's what we found, and we went ahead and did a lot of analysis. This is one case I will show you here. For example, when the PD-1 binds to PDL2, even the CC prime, not only just conformation, it goes from a loop conformation to strand. Similarly, there are a lot of conformational change happens in the CC prime loop, and even compared to the FG loop, the CC prime plays a huge role in binding of an IgV domain to IgV domain. Similarly, you can see here, these are special IgV domains, which are very rare, and you can see that when they bind to small molecule, the CC prime loop, which is in blue here, moves almost towards the red, that is FG loop, that is CDR3. And when it binds to a small molecule, see here, for example, this is CD22. Similarly, in TIM3, it binds to phosphatidylserine. You can see that it forms a small pocket. That means what we understood from this, what are the key residues we should not touch and where we should do modification. Probably one of the places is CC prime loop. I will show you that I did not get any grant from any agencies for this, whatever I applied from any DBT, DST, because probably it went to all crystallographers who thought maybe I'm again doing one more lysozyme mutation. Nobody gave me money, but it was supported by Bristol Myers Squibb in a small grant. And sorry. And basically what we do here is, oh, sorry, something is missing here. It's not moving. Oh. It's moving. Okay, it's moving. Basically, we, from the structural analysis and sequence analysis, map all these conservation on the, say the IGV domain we are modifying, and that way we know that what is conserved. For example, in this proline is extremely conserved in say a particular region, we don't touch that. At the same time, if nature is changing a particular region, okay, change it. If that is in the interface of say PDL1 and PDL2, we are more confident. That's how we worked on it. We made a lot of mutation and kept it, but there was no money, but still went ahead with something like 4 mutations we selected, which are definitely safe, okay, with all these analyses. And when we refolded the protein, because I did not have a cell culture facility, and when we refolded the protein, all the folded proteins, all the mutants bind, you can see that bind better than, bind to PDL1 better than the wild type 1. This is what we did with the single mutants. Then we went ahead with the, we made some small combinations of mutants also. You can see that the one in red, it is a wild type. All the other mutants, that like single, double mutants and triple mutants, actually the triple mutants are better than double mutants. You can see that it is 2 different concentration. And in this case, we bought actually HEC293 expressed PDL1 and again tested, because ours is refolded, whether it works with a folded protein, expressed in HEC293. All the mutants are high affinity mutants. And we also did this is the assay which from Promega, where this is luciferase based assay, where if the interaction between the PD1 and PD1 is there, then you don't get the glow. But when PD1 is L1 PD1 pathway is blocked, you can see the glow. That's how it measures the blockade of this pathway. And we can see that one of the only we were able to do with 1 mutant. And because when we started the initial experiment already, our kit was over, then we just did with 1 mutant. And you can see that all the, we can see that different concentration, our mutant is having a better capacity to block this PD1 PD1 pathway. And this has been patented this year. And what we are doing now, I have a grant, at least finally, people believe me. And basically, I'm trying to do this, basically the FC fusion of PD1 and even the single chain and bispecific, because I also have a modified TIGIT. And the reason is the smaller the size, we have a better penetration, tumor penetration. With this, I thank Dr. Dibyendo, Gayatri, I think she's somewhere in the crowd, and the students from her lab for helping me with all the assays, and Bristol Myers Squibb Small Scholarship Fund, and my previous institute. Thank you.
Thank you. Any questions? I have one question, if others are. If you make a peptide instead of having a fusion protein, your whole idea is that here you are trying to interfere between PD-1 and natural PD-1 and PD-L1 interaction. If you have a peptide which will do the same job, don't you think you can achieve your goal?
Yeah, see, this kind of studies are done actually. Many are doing these small molecules, like for example, to disturb the PD-1, PD-L1 interaction. There are small molecules which are some big companies are also doing. But what I see is the interaction area is very big actually. And probably it is not only just the interaction which is disturbed, maybe on the surface how they associate, because I don't have time actually, we show that most of the cell surface molecules makes a 1D array. That's the other thing. I'm actually recently went to PNS and came back, and that's the other thing I'm seeing actually. Means in the signaling, now how they control the signaling is not just like a molecule A interacting with molecule B. There is much more to that. Actually.
Okay. Thank you. Any question? It's okay. Thank you.
Thank you to the speakers and chair for such an intriguing session. I would request everyone to come on the stage for a group picture. Our next plenary lecture is by Professor Harinder Singh, Chair of the Board of Scientific Counselors at the National Cancer Institute. I invite Professor Shoman Vashak from NII and Professor Arun Kumar from IISc to chair this session.
All right. Let's come to plenary lecture 9 by Professor Harinder Singh. He's a chair of the Board of Scientific Counselors, Basic Research of the National Cancer Institute and a member of an advisory group to NIS Director, Dr. Francis Collins. And he's going to talk about regulation of germinal center B cell dynamics and the specification of plasma cell phases. Dr. Harinder Singh.
Okay, thank you very much. I have to start off with a correction. The two things that were mentioned were things in the past. In other words, I'm not serving in those roles at the moment. I am a professor and a director of Center for Systems Immunology at the University of Pittsburgh. So what I wanted to do was to actually talk about two stories and more importantly, keep to time. In my lab, we have been interested in B cell development and B cell dynamics, primarily initially using the mouse as a model system, and then more recently analyzing such dynamics in the context of human B cell responses. And actually just sitting through this past session, I noted a lot of connections between, I think, some of the things that I'm going to talk about and what previous colleagues have presented to you. So the first story is going to focus on analysis of human B cell dynamics at single cell and at single base pair resolution. So the single cell part should not surprise you, given that we are inundated with technologies that enable us to analyze individual cells, their trajectories, their transcriptional chromatin states, proteomics, and so on and so forth. But to molecular geneticists and genomics-minded people like me, analyzing cellular trajectories at single base pair resolution, I think does represent an important breakthrough. And so I'll try to explain what that means, what the implications are as we go forward. So the system that we use are naive human B cells that we isolate from PBMCs. Those cells are activated under defined conditions. We have not developed these conditions. We're taking advantage of really decades of work that other human immunologists have carried out. We've simply refined the conditions so that we can use them for high resolution analysis of these cells as they undertake their dynamics. And I'll just remind you that B cells start off like T cells being quiescent in G0 of the cell cycle. And upon activation, upon sensing antigen through their B cell receptor, co-receptors being activated, the cells initially undergo a substantial metabolic reprogramming, get activated, and the first cell division actually takes a lot of time. It takes almost 48 hours in this culture system, and then the cells begin to rapidly divide. And in the systems, the cells can be shown to undertake a primary bifurcation. In other words, you have some cells upon activation and cell division generating plasmablasts. So that's mimicking the extrafollicular plasmablast or antibody secreting response that we see in the context of human infections or human vaccinations. But the cells also generate from clones germinal center precursors. And many of you will know that that's a very unusual response where the cells induce a somatic hypermutator targeted to specific regions of the genome, particularly the V regions of these antibody heavy and light chains, and enable somatic hypermutation to occur. And if you have the process of positive selection occurring, then you can generate clones, B cell clones that express higher affinity antibodies to the antigens that instigated the response. So what's really nice in this system is that we can, in fact, when we activate the cells over the course of 6 days, have them undertake clonal expansion, and then the bifurcation, we see the generation of two groups of cells that are distinguished on the basis of expression of transcription factors IRF4 and 8. And these transcription factors we have shown in the mirroring system, along with other colleagues, as being critical for enabling the bifurcation. The cells that express higher levels of IRF4 are destined to become plasmablasts, the short-lived response, whereas the cells that express higher levels of 8, lower levels of 4, are the ones that give rise to germinal center precursors. So what's really nice in this system is that we can analyze this bifurcation, which in fact predicts, in a sense, the quality of a humoral response as it will unfold. The rapid response dictated by these cells and the slower response, the higher affinity response, which will be dictated by those cells. I won't have time to go through a series of cell surface proteins and other regulators that are consistent with the annotation that I've suggested. The other thing that I'll point out is that we've done this reproducibly using purified naive B cells from many different individuals. What's different is that each individual differs in terms of the relative frequencies of these two groups of cells that you generate. And we don't understand yet what the underlying basis of that is. In other words, is it genetic heterogeneity, or is it due to their immune status, prior infections, and so on and so forth? So now, to a molecular biologist like me, you want to use a system like this to do two classes of important experiments. One is, can we get down to single cell, single base pair resolution in terms of deep analysis of these cell fate dynamics? It's one thing to have phenotypic analysis. It's quite another matter to be able to do deeper molecular analysis. And the second point, of course, is that we also have to be able to perturb the system. In other words, deliberately perturb particular genes and then look at the alteration in the dynamics. And I'm going to argue that, in fact, both are possible, and therefore it becomes possible really to take apart human B cell activation dynamics at high resolution. So what we've done here is to use this system to analyze the changes in state of these cells, both at the level of the transcriptome as well as at their chromatin level, by using a combination of single-celled RNA and ATAC-seq profiling. So these are paired profiles. For each cell, we're capturing both the transcriptional status of the cell as well as its chromatin state. There are a variety of computational tools that can be used to analyze these massive data sets, and I'm not going to belabor the one that we are using. It's called MIRA, and it uses topic modeling instead of the individual RNA or ATAC-seq features to organize the cells in coherent clusters. Needless to say, this particular computational tool does a really wonderful job in terms of organizing the dynamics of these cells as they proceed from activation, clonal expansion, and then this bifurcation. And so what you see here is a low-dimensional, a two-dimensional projection of all of the cells from day 0 through day 6. And I've color-coded the cells. These are the naive cells. You can see this huge separation in the two-dimensional space of cells 24 hours after activation. And we can actually nicely temporally resolve the cells, day 2, day 3, 4, and then the bifurcated groups of cells beginning initially to be seen at day 5, culminating in day 6. Now, when it comes to those two major states, the plasmablast state and the germinal center precursors, the plasmablasts actually occupy this part of this particular two-dimensional representation, the UMAP, whereas the germinal center cells are accumulating over here, although you can see the onset of a germinal center program as early as day 1 of activation in terms of the genes that are turned on. So now, what about the single base pair resolution? So that's, I should emphasize those dots on the UMAPs, for those of you who are not looking at those, are actually individual cells, and they have these distinctive transcript and chromatin features. So the single base pair resolution part, really, I want to point out Anshul Kundaj's work at Stanford. I think he's really led he's a computational biologist who has developed neural network-based models that allow you to take chromatin data. So if you have genome-wide chromatin analysis where you can determine which regions of the chromatin are accessible to cleavage by a nuclease, a probe, and these accessible regions are regions that are reflecting control regions of genes, essentially. So you can train neural network models on these DNA sequences and their accessibility profiles. And these neural network models get so good, essentially, at analyzing DNA sequence patterns and associating them with these accessibility patterns, that they can eventually make predictions at single base pair level as to how much a given base pair is contributing to the accessibility profile that you're observing. So once you have that, you can ask the model, okay, for all of these regions in the genome that are accessible in my naive cells versus my activated cells versus my plasmablasts versus my germinal center precursors at single base pair resolution, what are the regions that are accounting for that accessibility? And then you can interpret those regions, it turns out, because they represent binding sites for known transcription factors. And so, in a sense, this method reveals or predicts the underlying nature of the regulators that are binding and acting on those regions. Now, I have to digress a bit. This tool is so transformative. And, you know, there's a lot of hype about AI, but in this case, this is an example where it isn't hype. It's actually quite real. And let me illustrate that. When I began as a postdoc nearly over 30 years ago, working with Philip Sharp and David Baltimore, we actually were amongst the first to discover mammalian transcription factors. So we were working at a time where a couple of these transcription factors were known, and we developed tools that advanced their discovery and analysis. And it took 33 decades, lots of scientists, to catalog essentially all of the transcription factors that are operating in human cells and the nature of the DNA sequences that they act on. So let's wipe out all of that history, okay? And fast forward and say, at this point in time, we have only one experimental method, ATAC-seq, which can give you chromatin profiles in given cells. And we have one computational tool, ChromeBP-Net, developed by Anshul Kundadje. Well, if you combine those two things, you can reveal the entire lexicon of the human transcriptional factor regulatory code that took 3 decades of painstaking biochemical and molecular biological work. So that's how important I think this particular advance is. Not a surprise, 2 Nobel Prizes actually went to either the generation of neural network models or their application in the context of predicting protein structure this year. Okay, so now if we turn this powerful model onto our data sets, it will allow us, so what I'm showing you are the predictions of the model. The model is either being trained on chromatin profiles for the germinal center precursors or the chromatin profiles of the plasmablasts. And these are the dominant, this is the dominant lexicon that it's revealing of the sequences that are controlling chromatin accessibility and by inference transcription in these cells. I won't belabor this, but if you now look at these predictions in the context of what we already know, the predictions line up. In other words, transcription factors that are known to regulate these alternate cell fates are predicted correctly by the model. But it also begins to pick up new lexicon, which we haven't known about. And I'm going to actually talk about this particular extended motif because we think this turns out to be quite important in terms of the generation of plasmablasts and human plasma cells. So now to turn to the perturbation part of the analysis. So for this, what we use are arrayed CRISPR screens. So we basically perform gene editing in these cells by transfecting them with Cas9 RNPs that carry particular guides to genes that we're interested in. Specifically, we're testing the sets of transcription factors that ChromeBP-Net and MIRA are predicting are important for controlling cell fate. And I'll just show you examples of such a screen. So here we have perturbed, I think it's 30-some transcription factors in these cells. And the perturbation is done at day 2 of activation. And then we monitor what the consequence of that perturbation is phenotypically in terms of the relative frequency of germinal center precursors and plasmablasts we generate. And then we do single cell profiling of the perturbed states so we can see to what impact is the perturbation having on the transcriptional state of the cells. So this is on the top are a set of transcription factors which when they are perturbed reduce the frequency of germinal center precursors and increase the generation of plasmablasts. And conversely, on the bottom are a pair, and only a pair of transcription factors that have the flip result. We basically block the generation of plasmablasts and greatly increase the frequency of germinal center precursors in those cells. Now, both of these transcription factors, BLIMP1, PRDM1, encoded by the PRDM1 gene, and IR4, are well known in the mirroring system to control the generation of plasmablasts and plasma cells. So in that sense, this is not a surprise, although it's nice, obviously, to be able to confirm this definitively in the human system. But then we can pose the following question. Do these two regulators control the same or distinct programs or modules when they're inducing plasmablasts or plasma cell generation? And so here, as I said, we perform single cell RNA-seq on the perturbed cells. And I'll just show you one example of this. So what we're doing here is analyzing genes that are impacted, either upregulated or turned down by one or the other perturbation. And it's striking that a lot of genes actually lie along the diagonal, particularly genes that are repressed by these two regulators, suggesting that they're functioning in a coincident or a concordant manner to turn off expression of a set of genes in the context of inducing plasmablasts and plasma cell differentiation. The reason that's important is if you think about B cells as opposed to T cells, they do something really unusual. When they differentiate into plasma cells, they actually lose B cell identity. And by that I mean they essentially turn off a very large set of genes, including genes involved in antigen presentation, BCR signaling, and so on and so forth. And they induce now a set of genes that are specific to plasma cells. We think that these two regulators are functioning in a concordant manner to shut down B cell identity. And how might they be doing that? And that's where I come back to this prediction that ChromeBPNet makes. This extended ISRE sequence, the ISRE stands for interferon sequence response element, is a sequence that we had noted a decade earlier when we were analyzing plasmablast generation. And we have reason to believe from a molecular and a structural standpoint that this sequence is co-bound and perhaps cooperatively bound by IRF4 and PRDM1. And we're in the process now of molecularly testing that particular possibility. Okay, in the remaining bit of time, I want to turn to a second story, and it's a second system. So now I'm going to use the mouse and not the human system. And now I'm going to go past the primary bifurcation that I spoke about, which is B cells getting activated and generating plasmablasts or germinal center precursors. But as you allow a germinal center response to unfold, and that occurs over weeks or in fact months in humans, the cells are acquiring mutations, they're undergoing positive selection, and therefore they're generating clones with higher affinity receptors for the original antigens that set off the response. Ultimately, though, the purpose of this is to allow the generation of either memory cells with higher affinity receptors or plasma cell precursors, which will then home, for example, to the bone marrow and generate either short-lived or long-lived plasma cells. The long-lived plasma cells, of course, are the key to durable humoral immunity. And so what we want to do here is to address an age-old question that is posed in the context of vaccines. We know that certain vaccines can generate lifelong protection. And the humoral protection that they generate is in fact due to long-lived plasma cells in the bone marrow that can be detected decades after the vaccine was administered. And examples of such vaccines are over here. For example, measles and varicella. Now, other vaccines actually only generate short-lived antibody responses. They generate short-lived plasma cells. And so a fundamental question that has been around for decades is, how is this process molecularly and genomically controlled? And if we can understand the basis, obviously we should be able to rationally design vaccines with suitable adjuvants that preferentially promote the generation of precursors of long-lived plasma cells. Okay, so how do we address this question? I'm actually embarrassed to say, because I'm really not to present these experiments from the standpoint that I thought these experiments had been done decades ago, and when I looked at the literature, they surprisingly had not been. And so we decided to do these experiments. So the idea is fairly straightforward. What we do is we instigate an immune response in our one set of animals. We're using black 6 mice here, and we're immunizing with NP-KLH. And of course, B-cell responses to NP, immunologists have analyzed in great depth. So we're able to take advantage of all that knowledge. In fact, we're able to track NP-specific cells in all of these analyses. Now, the peak of the germinal center response occurs at around day 14. We deliberately wait out the peak of the response and then begin to isolate CD138 positive splenocytes at different days after immunization. Those cells, we have reason to believe, again, because of prior work, are where the plasma cell precursors that are emanating from the germinal center reside. And so what we do is we take that group of cells and we adoptively transfer them into RAG2 mutant animals. Actually, I'm sorry, in these experiments, not RAG2 mutant animals, but MU-MT mutant animals. So those animals are blocked for B-cell development. They have no B cells, they have no plasma cells. The adoptively transferred cells, if they contain plasma cell precursors, should be able to home to the bone marrow and give rise to plasma cells in the recipients. And this should be now, we're not adding any additional antigen at this point. And of course, in the adoptively transferred animals, we can analyze for the occurrence of plasma cells initially by just monitoring antigen-specific titers over time, and then, of course, formally recovering plasma cells from the bone marrow and showing that they, in fact, are secreting NP-specific antibodies. Now, when you do this rather simple experiment, what you find is an unexpected result. At least to me, it was unexpected. What we found was that when we started analyzing these cells at day 21 in the adoptively transferred animals, we could detect NP-specific antibodies early after transfer, day 7, but then the titers actually dropped and they were gone by day 60. And in fact, I'll show you, and similarly, if we analyze plasma cells in the bone marrow, the same thing. And so this argues that if you look at an earlier point in the germinal center response, as it's generating plasma cell precursors, those precursors are competent to give rise to plasma cells in the bone marrow, but they're short-lived. Now, you get a very different result if you go longer. And I'll just show you the result with day 35 after immunization. So here you'll see we've actually more or less stabilized antibody titers out to day 60. And in fact, if we analyze for plasma cells 120 days after the adoptive transfer, we can actually detect at the highest frequency these NP-specific cells. So I know this is a mouse, so the time span is much shorter than in us, but I would argue that what this is showing is that the precursors of long-lived plasma cells, at least in this mouse model, are generated preferentially at late points in the germinal center response. And we know from earlier work that this is the point in the response where antigen is beginning to wane, the germinal center is shrinking, and it's converting somehow into generating a different class of precursors which have this preferential ability to give rise to long-lived plasma cells. So, of course, we're very much interested in high-resolution analysis of these precursors. You know, what are the cells precisely that have this capability of homing to the bone marrow and then giving rise to long-lived cells? And so if you analyze these cells by single cell RNA-seq, I'm going to just draw your attention to, two clusters of cells. This is a group of cells which we think are the earliest progenitors emanating from the germinal center. And I'm only showing a handful of genes, but the thing that I want you to note is they continue to express hallmark B-cell specific genes. So they haven't lost their identity yet, but they're beginning to turn on genes, including turn up IRF4 or turn on PRDM1, which are going to be needed to induce plasma cell generation. So this is the earliest molecular switching that we can detect. But it turns out that these are not the precursors that are capable of homing to the bone marrow. Those cells actually lie within the group of PC clusters shown on the right. And in fact, and this is another surprise, these cells are the subcluster that I've denoted as PC TIGIT plus. So I'm going to emphasize TIGIT because all of you who work on T cells, of course, know this molecule quite well and its ligands. And it's a complete surprise, in a sense, that TIGIT is inducibly expressed on B cells as they're coming out of a germinal center response. And in fact, I'll show you in a moment that these are the cells that are highly enriched for the precursors, the plasma cell precursors that are competent to move to the bone marrow.
So we may have another minute or so.
Thanks. So I will, so if we analyze these cells molecularly, as I said, the TIGIT positive cells are beginning to, they have highly induced a lot of PC genes, and they're also showing evidence for undergoing greater proliferation. We track these cells on the basis of their particular antibody genes, heavy and light chain genes, because we're simultaneously sequencing those. And of course, we know which ones are specific for NP. And so this is just an example of connecting the trajectories of the cells. And in fact, the progenitors connect very nicely in their trajectories to the cells that go on to express TIGIT and give rise to plasma cells in the spleen. We can also connect these clones between the spleen and the bone marrow, and in fact show that the majority of cells emanating from the spleen that are NP specific and that can be connected to progeny in the bone marrow are in fact cells that express TIGIT. The TIGIT protein, so I've only been showing you TIGIT transcript expression, the TIGIT protein is inducibly expressed in this group of cells that is turning down the expression of B220, CD138 positive. Here's this group of cells. And if you analyze if you purify those cells on the basis of either expression of TIGIT or not, adoptively transfer them, you can see we get strong enrichment for the TIGIT positive cells as the ones that give rise to plasma cells in the bone marrow. And I just want to conclude this part by emphasizing that if you've performed genetic analysis on TIGIT, and we've done this not using a floxed allele, but a germline allele, so we make chimeric mice that carry either wild type or TIGIT wild type or mutant cells, reconstitute animals, immunize, and then follow NP-specific responses. The loss of TIGIT results in a loss and generation of plasma cells, both in the spleen and the bone marrow. And this is just the quantitation of that.
So we need to conclude, I guess. Yeah.
Okay, so let me just finish with a model and then take any questions at this point. What I'd like to argue is that there's a fundamental shift in the germinal center that accounts for the generation of short-lived versus long-lived plasma cell precursors. The shift obviously is in part caused by diminished antigen, which would result in diminished BCR signaling. The induction of TIGIT, and we're postulating that just as in T cells, where TIGIT signaling antagonizes T cell receptor signaling, we're going to argue that TIGIT signaling in B cells is also set up to antagonize BCR signaling. The combination of diminished antigen, TIGIT functioning as an antagonist, will diminish BCR signaling in the germinal center and allow amplification of CD40 with IL-21 signaling. And we think that change is what's critical in enabling the shift from short-lived versus precursors for long-lived plasma cells. So I just want to acknowledge a lot of people, both at the University of Pittsburgh, as well as at other institutions that have enabled all of this work. And I thank you for your attention.
Thank you very much. And we probably will have time for 2 questions. Yes. But the microphone.
Yes. May I?
Yeah.
Yeah. Very interesting talk. I was wondering whether the precursor cells that are long-lived plasmablasts towards the later part of the germinal center have any origins from the higher affinity of the binding that the receptors will have to the antigen now because of greater affinity maturation? Or has that been controlled for?
Well, what is not surprising is that the cells that are generated, the long-lived cells, if we analyze the high affinity, they are enriched for high affinity clones because they come out later in the response, having undergone extensive hypermutation and selection. But I'm not sure I'm answering your question.
The question is whether that affinity, increased affinity of antigen binding has a role in there being long-lived.
Right. Now that's a very good point. So you would argue, well, obviously, as you extend that reaction, the germinal center reaction, you're increasing affinity. And might that increased affinity be used as some kind of a signal to, alter the state of the precursors that are generated? Clearly, my bias is the other way, that you need to antagonize signaling through that BCR if it's functioning as a signaling system. But you raise a good point. It could be involved.
Well, for the second question, yes, over there.
Yeah, thank you very much for this very nice presentation. I'm just wondering, you said that the type of antigen is important. So I think it is obvious if you compare live vaccines with inactivated that this is a matter of length of exposure. But the question is, there are also in inactivated vaccines, the possibility that you have actually a long-lived plasma cells. I'm just thinking of hepatitis A vaccines. You have 30 years of protection or even longer, and it needs to be only vaccinated. So what is here the difference, you think? Is it also an antigen-specific effect that can have an influence on the TIGIT expression, or is it really only the length of or the duration of exposure within the germinal centers?
Yeah, I tend, right. So you're quite rightly pointing out, the two aspects of the model, and I stress it's a model that we really have to rigorously test. So you're pointing out to the two fundamental things that I'm invoking. One is the duration of that germinal center response, and obviously adjuvants or the nature of the vaccine would fundamentally control that. And then the other part is, we actually don't have a handle on what's causing that TIGIT expression. And for that matter, we also, since TIGIT has several different ligands, we've yet to identify what the relevant ligands are and who's presenting them. So I think, we're going to need to explore this before we can address the kind of question that you've posed.
Thank you very much. I think the rest of the discussion can happen later.
I would request the speaker and the chairs to come up for a group picture. Thank you, Professor Singh and the chairs. We now welcome Professor Leighton Grimes from Cincinnati Children's Hospital for yet another plenary lecture. It is my honor to invite Professor Pawan Sharma from IISER Bhopal to chair this session.
It's ready to go?
This is the presenter.
Yeah, so this is
Back, front, and the
this is front.
Yeah.
This is what is okay.
Good afternoon. It's my pleasure to introduce the speaker of this plenary session, Dr. H. Leighton Grimes, and friends call him as Lee Grimes. Is that right? Yeah. He's the director of the Cancer Pathology Program, Division of Experimental Hematology, and Division of Pathology at the prestigious Cincinnati Children's Hospital Medical Center. The Grimes Lab works on hematopoiesis, response to the infection, and molecular oncology, and that includes mouse modeling of hematopoiesis, myelopoiesis, marrow failure syndromes, including myelodysplastic syndrome, MDS, severe congenital neutropenia, and acute myeloid leukemia. And believe me, his work on each of these syndromes is mesmerizing scientific saga in itself. I can't hold myself back from alluding to at least some of his most impressive contributions which to me, they have direct positive consequences in patient care and management. Earlier this year, in March 2024, the Grimes Lab, actively collaborating with the Nathan Selamonis Lab, published what has been called the world's most detailed atlas of the many types of stem cells and early progenitors involved in producing human blood. The Nature Immunology paper describes as many as 80 distinct subsets of hematopoietic stem and progenitor cells, documenting perhaps for the first time various intermediate cell states and stromal populations and the most abundant and end states amid dynamic instability. The work is testimony to meticulous harnessing of the power of multi-omics approach, which included single-cell RNA sequencing, matching flow cytometry, and innovative application of game theory and machine learning technologies for data analysis. And as evident from their work published in Nature 2016-2020, along the way, the Grimes Lab collaborators have developed several extremely useful mouse models of human disease like acute myeloid leukemia, severe congenital neutropenia, etc., and harnessed the power of new genomics and computational strategies to create a general experimental platform to unravel the mutations, cell types, developmental stages, and molecular pathways, possibly driving the diseases which include pediatric hematological malignancies, neurofibroma, prenatal inflammation, ulcerative colitis, etc., etc. Ladies and gentlemen, join me in welcoming Dr. Lee Grimes to the stage. Stage is yours now.
Thank you. And thanks to the organizers for inviting me to speak. So if anybody is interested in the Human Bone Marrow Atlas, that was a precursor story. And the next part of that story just came out of Nature Genetics. And that's the sequencing of people with African ancestry who have AML and the discovery of novel mutations that they have that give them very poor outcomes. And the atlas that we're talking about was, used donors that spanned both sexes and race to provide a platform to understand the genomics of the people with African ancestry. It's a fun way to understand things if you want to look at that Nature Genetics paper. Okay, but today, something different. I'm going to talk about a framework to understand hematopoiesis, and I'm going to use the mouse in this study. This is actually a twin study to the one that is published in Nature Immunology. And this is how we understand, whoa. So here's the people who did the work. So this is a lot of work from a postdoc, Kyle Firkin, helped quite a bit by Annie Song and postdoc Sean Zhang. collaboration with Nathan Salomonis and Kara Vithacker and Frank Lee in his lab, and of course my good friend Harinder, and a friend Sam Morris, who has a couple papers that are very interesting that we exploited called Cell Tag and Cell Oracle. Okay, so how are blood cells made? You know, we want all these cells, and you know, you guys are immunologists, so you study all these cells. But how are they made, and why is that even important? You know, so 2/3 of your marrow is taken up by the decision to make red blood cells and neutrophil granulocytes because you burn them. They exit the cell cycle. They never come back. If you have an infection, you're going to burn those neutrophils. They're going to die. There's going to be a call back to the marrow to make more. If you bleed excessively, there's a call back to the marrow to make more red blood cells. So there's dynamics that are happening, responses to the marrow progenitors. They have signals. They understand what's going on in your body. And we don't understand how that works at all. You'd think that we would by now. So we understand that there's quiescent stem cells because we transplant these into people. They engraft and they give all of these cells at the end. But we think that there's compartments here of multipotent progenitors, you know, pioneered by Irv Weissman's lab. And then there's decisions that are made to make these bipotential progenitors. And other than that, we don't really understand the process. And we also don't understand where the decisions are made. Of course, Irv would say that he knows where they're made. But he would say that they're in an MPP, and I'm going to tell you that Irv is wrong. So here's a couple of terms that I'm going to use, just where they came from. So metastable state is just, you know, there's stability, there's really highly unstable things, and a metastable state is something kind of in between that. And then there's dynamic instability, which is taking a term from the Mitchison-Kirshner story about microtubule growth and collapse. And in this case, we're kind of borrowing it to understand the forming and unforming of a lineage decision. And then, you know, a cell state, which I think most people understand. But basically, I'm going to talk about it as just the sum of the expressed genes and transcription factors, which is a view populated by Eric Davidson, who's now passed. Okay, so what do we do with those concepts? We previously, in 2016, isolated marrow cells and did single cell RNA-seq, and then really analyzed them to find that there were these early progenitor cells, which were ruled by GATA2 and MEIS1 enhancers. Those then became deconstructed, and at the same time, construction of Pu1 CBP alpha enhancers was happening. And these are opposite ends of the universe, and they don't like each other. And so these cells had to get out of there. They were experiencing what we call dynamic instability. And there were these populations of cells in which this was happening, which we named multi-lin, because they were kind of sampling different lineage genes, but not coherently. So it wasn't like suddenly you express all the MEG genes. No, it's just like one MEG gene, one erythroid gene, one monocytic gene. And this dynamic instability, we thought, was a sampling of ways to get out. And we showed one way to get out of this dynamically unstable state was to go through this very, very unstable or metastable intermediate where you launch both sides of this gene regulatory network motif. These are opposing, as you can see from the arrows. These transcription factors are acting together. These transcription factors are acting together. We got very excited about this because GFI1, which I cloned a long time ago, when it's mutant in kids, gives you neutropenia, and IRF8, when it's mutant in people, give you monocytopenia. And these guys were at the head of this mouse gene regulatory network, which is very fun because people think that humans and mice are not good models of granulopoiesis. Well, here's the core of it is totally conserved. And so these cells are so unstable when they have this at the same time that they make a binary cell fate choice because they can't stay there. So, but there's many other ways to make cells, you know, many other cells to make. And so there have to be other exits from this multilineage cell state. What are they? Well, we decided to find out. So we did really deep dives into just all c-KIT positive marrow cells, a multi-lin gate that we generated, an HSC MPP gate that we generated, and all the IL-7 receptor positive cells. And I won't belabor it, but we actually titrated molecularly 270 antibodies from BioLegend and the CiteSeq platform, TotalSeq A. And then we found out that only like 130 of them are actually worthwhile playing with for this population. They make sense, they participate in clustering, they give you a good dynamic range. And then we utilized Nathan's student Frank's program called SC Triangulate. If you don't know it, look it up. It's very fun. It's much better than if you have, multimodal data. It's much better than nearest neighbor approaches to form clusters. So what can you do with this program? You can say, here's the RNA data, high, medium, low. Here's the clusters, Leiden clusters. Here's ADT data, the antibodies, and you cluster that high, medium, low, and then you can have all these published references. We had like 13 published references where people had previously captured bone marrow and they put them together. And SC Triangulate actually figures out what's the most stable solution to the clustering problem that you give it. And so we end up with really beautiful clusters here. And here's the proof of that, I guess. Here's the key genes that organize these clusters. And here's all the antibodies that go with them. And here's where they came from. So the bottom line here is just that we have an atlas of the stem and progenitors, hundreds of thousands of cells deep, covering all these transitional populations that are super rare and hard to get to, right? So, and there's specific number of antibodies, 130, that go into this atlas and participate in the clustering. So now we want to translate that into reality, into real world data. And so we used full spectrum flow cytometry and infinity flow. So those who don't know infinity flow, here's the Python version that we wrote that you can work on your laptop with millions of cells. But infinity flow is that you have a backbone of antibodies that are, each one of them is going to describe the major populations in the pool that you're looking at. And then you leave open some bright color like PE, and you put individual colors on that. And you have a series of flow experiments where you're creating these FCS files, and it has the same backbone, but this antibody A, same backbone, antibody B, and so on. And then they're sewn together using XGBoost nonlinear regression. And so the final thing is to make this very large object. It's not a real thing, it's an object, and it's inferred, in other words. But it's every single antibody that's used in the CiteSeq. So what I'm telling you is, I now have a flow FCS file, flow cytometry data, that contains every single antibody in the CiteSeq atlas. So now we have twins. I'm going to skip this. We have twins, and these guys here have RNA and antibody. These guys over here have antibody in real world, and their organization. You can see this is very deep. Even the ILC and NK cell precursors are present here and illustrated. Here's the eosinophils and basophils, et cetera, et cetera. But what we really want to do is to transfer the cluster information from the RNA into the flow cytometry data, right? So that's like a kind of a novel concept. We want to sort the clusters at the end, right? We want to say, look at that cluster. Isn't that interesting? That's a transitional population. I wonder what that does, or how do we understand that population? You want to get the cells, right? But you can't have them from the CiteSeq. So can we push the CiteSeq genomic information into the infinity flow object so we can have them? And the way that we did that was we assigned a target multi-omic cluster population, and then we looked at the population using markers in the infinity object. Basically, like when you backgate in flow, you can actually go back and say, like, well, was this marker on it? Was that marker on it? Et cetera, et cetera. And then we captured we sorted the cells and captured them by 10x, and then we aligned them to the multi-omic atlas reference. So we went back to the CiteSeq and we said, what did we get? How pure are those cells? How good did we do? And the answer is, it's pretty good. So we wanted to get this pre-baso cell. Here it's in the baso thing. We wanted to get immature neutrophil stages, and we got immature neutrophil stages, et cetera, et cetera. And the contaminations are basically coming from the nearest neighbor, which is what you would expect because flow cytometry is only so good also. You know, there's also a limitation of the technique you're using. So we benchmarked the technique that we had by using some of the ones that we'd already captured and going back and forth with SciCombine, Z-score, Mario CCA, XGBoost, and bridge integration from Seurat. And this method works best of all of them. So it's, you know, good on Kyle. He really figured something interesting out. But the biggest thing that's the most important thing that I want to stress to you is that now progenitor populations defined by their current coherent genomic content can be physically isolated and shown to display the expected gene expression. So if you thought, or were told, that hematopoiesis is a continuum, you know, everything's kind of schmooing and moving here and there, there's no real stable states, this is a very difficult thing for you to hear, you know, because what I'm telling you is the constellation of antibodies on the surface of the cells that are progenitors, that are in transit and moving towards differentiation, if you use those antibodies, you isolate the cells, the new cells that you isolate are still going to have that exact constellation of genomic content, right? So there's stability somewhere in there, which means it's probably not a continuum. Okay, and also this is the inverse of classical hematopoiesis, where we use antibodies in the first place, isolate the populations, and then ask what they do, right? So here we're saying, no, we're going to put the genomics first, and then ask, how are these things related to one another? Okay, so now, you know, Harinder already talked about using Chrome BPNet, so I won't go into that, but we did, I'll introduce one other thing. So when you have the multi-omic data with the RNA and the attack, you can also add another layer to that, which is the antibodies on the surface, using something from the Allen Foundation, Peter Skeen's lab. It's called T-seq, you know, for all three of those features that I just told you. Basically, you take your site-seq cells, you permeabilize them with digitonin, and then you tagment them and you capture them on a multi-omic thing. Then you make the three different libraries and you sequence them and you put them all together using the barcodes, the shared barcodes for the cells. Okay, fairly straightforward, I think. But we couldn't get, you know, we just wanted to look at the multi-lin cells for this thing. And then we analyzed we pushed the clusters from the site-seq again into the T-seq. Just as we pushed them into the infinity object, we're pushing them into the attack to organize it by those clusters that were hard won. And then we're using CRO-BPNet to tell us what's there. Well, here's the patterns that it finds for the individual base pairs that are contributing to the peak. And, you know, another way to understand what Harinder was saying is there are peaks in the attack and they're getting bigger or they're smaller, or they're bigger or they're smaller. And how many reads that they accumulate, that accessibility is, that's the contribution score that they're talking about with Chrome-BPNet. The contribution of the nucleotide to the accessibility of the DNA. So apparently these motifs, which are consistent with these families of transcription factors, are what's ruling the generation of the hematopoietic system. And you can see here, these are all the MPPs and HSCs. And then we have these multi-lin clusters here, which appear to be on a really different planet. They're very, very different than these multipotent progenitors. And if we then use the edges between the seclets and their CWMs, contribution weight matrices, to the genes that they're regulating. So just by doing a simple Pearson correlation, right? So we're saying you had a peak and it was going up and down. You had a gene, it was going on or it was going off. What's the correlation between the gene going on and the peak going up? And then within that peak, since these seclets and CWMs explain the peak height, which one of those seclets actually explains this gene going up? So we're linking the transcription factor activity on the chromatin to the expression of the gene. And then that correlation, okay, can be visualized using Cytoscape, which is shown to you here. So these are the kind of blobby things that are hard to understand, but let's look at that. Here's HLF and its activity. These are stem cells. Here's IRF8 and P1. These are the myeloid lineage. Here's GATA1 and GATA2 over here. This is the meg-erith. Okay, there's your orientation. So now you can ask the question, for each one of the clusters that you had, those correlations are going to change, right? As you move along, the gene is going to go up, the gene is going to go down. The correlations are going to change to which transcription factors, which peaks are present as you move through the clusters in hematopoiesis. So you can then ask the question, can I just, don't show me everything, just show me the ones that were good in a quiescent HSC. And it shows you all the orange genes are all HSC genes. And I told you before, this HLF shows you where that is, right? But there's very little going on out here. But now as you move to this multi-lin first population, you start to see a little bit of IRF8 and PU1 active. On this guy, ML2, there's a lot more of that activity. And in ML3, that dies down. And there's GATA1 and GATA2 active. And you can see that this is coherent with the next step of megakaryocyte erythroid progenitors or basophil mast cell progenitors, whereas this is more coherent with the future of a pro-neutrophil granulocyte cell or an MDP. So there's your MDPs. Yeah, okay. Okay, so then you can kind of convert this information by looking at each of the clusters for all the edges that a transcription factor has. In other words, all its target genes, right? And how much it's expressed. And you just z-score that. And then you can say, okay, what's the activity of FOXD2? Wow, it's very specific to quiescent HSCs. That's a known thing. That's really exciting. To recapture that information, you can see the activity of the transcription factors as they move across. And what you can also see is that these guys here, GATA2, GATA1, IRF8, and PU1, and some CBPs, are over there in the multi-lins. And so we wanted to look at those activities. We had reporters for the transcription factors, PU1 fused to YFP, GATA2 fused to venous, et cetera, where we could actually see the level of the transcription factor protein in the flow data, right? And then we can actually say, okay, well, what was the TF activity score for those? And you can actually see that it starts to go up with the level of the protein, with the level of the protein, with the level of the protein. Pretty cool that you can actually see the activity in a cluster-specific manner from something you're inferring from flow data, right? Just showing you that this really works, that the flow and the genomics are all intertwined with one another in an appropriate way and something that works. Okay, so now what can you do with this? We have a tool, right? We had twins born, right? We put them together. We figured out how to sort the cells, but now what can you do with that information? So here we just did the TF activity score for surface proteins, because remember I showed you there was ML1, ML2, ML3. ML2 and ML3 looked like they were splitting the multilineage gate. Maybe they were already making a decision. We wanted to know how to separate them. And so we asked the question, where are there activities that are associated with surface molecules? And I want to call your attention to CD55 and CLEC12A, which CLEC12A, you'll remember, is CD371. Okay, and there, CD55 is going with these GATA factors, and CLEC12A is going with these CBP factors, IRF8 and PU1. And if we now go to Cell Oracle, which is Sam Morris's program, you can use the information that we have, the gene regulatory network, with the sequence, CWMs, the TFs, and their target genes in each cluster, and you can say, predict for me what is happening here, or what would happen if I perturbed all of these things. And so we can actually perturb GATA1. We can say, we can pretend GATA1 is gone. What happens? Well, everybody rushes away from ML3 and towards ML2. And what happens if I cut out IRF8 and there's no IRF8 there? Well, everybody rushes away from ML2 and towards ML3. So according to Sam's program, these transcription factors I showed you that were associated with the expression of those surface molecules would in fact control the lineage fate, because their absence makes the lineage go the other way. Does everybody understand that? The transcription factors are understood to drive genes that are making surface proteins. And if you cut out those transcription factors, the cells are going to go the other way. So we think then we may be able to use those surface molecules to catch the cells as they're making a lineage decision. Oh, I don't need to show that. Okay, so here's what this is what looks like under the hood. So here's the seclets for the CBP family for CLEC12A. You can see them here in ML2, but not in ML3. And then here's the ICE element. So that's Harinder's name for PU1 and IRF8 together. And that's loading in here in ML2, but not in ML3. And here's the GATA family binding here to ML3, but not ML2. And the dots on the side just show you the expression of the gene that's in the same cluster. Okay, so nascent TF activity in the multi-lin populations corresponds to induction of surface markers, CD55 and 371. But what does that mean biologically? I mean, you could guess what it means biologically from the Cell Oracle program, but that doesn't mean that's really what's going to happen, right? That's an inference. So yeah, so here's that clue for you. So we did the cell tagging and we kind of understood what the progenitors are doing with one another. And then we used this 371, 55, and the double negatives of the multi-lin gate to dissect what's really going on. We used CREs that are lineage specific. So here's CD11C CRE, MRP8 CRE for neutrophil granulocytes, EPX for eosinophils, and EPO-R for red blood cells. And we sorted the and you'll notice these guys only mark cells way out here. So this is a Rosa LSL TD tomato marker gene that's being activated. They're way out here. So we're sorting cells from here and we're saying, if I plate a single cell in a well, what are you going to become? Will you become something that launches CRE and marks the cell as red? And so you'll notice here that if you take the cells that are double negative, that they make every single one of these lineages, EPO-R, EPX, MRP8, and CD11C. But if you say, okay, you have to be the 55 positive multilin, then the only thing you can make is the EPO-R, right? And if you take the 371 guys, they don't make the EPO-R anymore. They make the eosinophil granulocytes, the neutrophil granulocytes, and the dendritic cells, right? So there's the split for you. This is where the lineage restriction events are first happening in hematopoiesis, not in the MPP, but in the multipotent progenitor called and multilin. Okay, so here's a cartoon to kind of make a synopsis. So you have this multilin 1 giving rise to multilin 2 and multilin 3. This guy can give rise to MDPs, monocytic progenitors, eosinophil and neutrophil progenitors. And this guy here, ML3, can give rise to the BMCP and the MAP. And there's a backdoor here straight from MPP1 to making eryth and meg cells, but we didn't discuss it. Okay, that's it.
Thank you. Thank you very much. Keeping the time. Questions, please. Okay.
It was so clear.
Yeah. Yeah.
Oh.
Oh.
Saurav.
No, I mean, obviously it's very clear work, so it's not easy to ask questions in the sense, but I had more of a sort of a mathematical type of question, right? So you have some states which are clearly more stable, and there are states that are unstable, and then there are metastable states, and all defined by a particular combination of transcription factors. So what about certain...
Well, the stable versus metastable would be defined by how frequently we can sample them. That's our use of the term.
Okay.
Yeah.
So it's not really a particular combination of transcription factors competing to give
you that
the
state
metastability.
Yeah. What is making instability and instability happen? So we don't know that for sure, but what we think is that you have enhancer landscapes that are not allowed together, and as you start to deconstruct one and construct the other one, you need to get out of that. Harinder may disagree with me. You'll have to ask him.
No, but there's some sort of a symmetry breaking event happening.
Yep.
With the allowability of combinations.
Right.
Okay.
And we don't know what clearly there are external inputs, as I alluded to, like infection, bleeding, whatever, and those have not been put into the model yet either.
Right. And would there be contribution of the niche the cells that are
Yeah.
in contact
Yeah.
with that?
Yeah, for sure. So we published a paper with Danny Lucas in Nature, in which Danny showed that the production of bone marrow cells is very, very specific. There's like a map you can draw of where things are and where they're never, right? And we actually showed that the deletion of CSF1 in only a subset of endothelial cells actually blocks the production of the MDP and the monocytic progenitors, but it doesn't do anything to the serum levels of CSF1 at all, only in the marrow, and it screws up the production. Very elegant story. It's in Nature. Check it out. It's very pretty.
Okay, any other question? Okay, thank you, Lee.
Thank you.
Thank you so much for very elegant talk.
Pardon? A picture. Oh, yeah.
I would request the speaker and the chair to come up for a group picture. Thank you, Professor Grimes and Professor Sharma. Next, I would like to invite the Sony team on stage for their 10-minute technical presentation. Is anyone from Sony team here? Request everyone to reconvene by 5:50 p.m. so that we can continue promptly. Participants who have presented their posters today are requested to remove their posters from the board and enjoy your break. We'll see you shortly. And we'll resume with the technical presentation by Sony. I hope they'll be ready on time.
Good afternoon, everyone. Namaskar. आप कैसे हैं? मेरा नाम थानाका. Sony Japan से आया हूँ. Can you understand? So thank you for waiting. We Sony finally come to India market. So I'd like to explain about our product feature within 10 minutes. So have you ever heard Sony flow cytometer before? So could you raise your hand? Oh, thank you so much. Several persons know our product, but our Sony is known as a TV, a headphone, and camera, and sometimes PlayStation, music, and movies. And our flow cytometer division is under the same umbrella of electrical product lineups. Why we enter this product segment is we are fully utilize data processing technologies such as Blu-ray, TV, and cameras. So we utilize those kind of processing technology into research areas. That is, we started the business in 2012. Today, I'd like to introduce, currently we have a total, so 5 products lineup in the market, and we are going to release 1 new product in the next year. But today, I'd like to touch on 3 products. First of all, I'd like to introduce our spectral cell analyzer, ID7000. So this is recently, spectral analyzer technology has become standard. So many of the players in the spectral analyzer market. Maybe as you may know, so compared to conventional analyzers, so conventional cell analyzer is equipped a bandpass filter. That is detected only specific wavelengths. However, prism of spectral detect all the spectral signature at once and divided each with unmixing. Why it can be achieved unmixing is Sony spectral analyzer is using spectral libraries. If you register the dye at once, you can utilize for future analysis. That saves the cost also. Most important things is you can free from compensation when you use a spectral technology, because spectral unmixing device, the color automatically divide the color automatically as long as you register the dye in the libraries. And if someone is struggling with autofluorescence, spectral is the best solutions. Sony library can be registered multiple autofluorescence also. After the autofluorescence subtractions, result may be much clearer. And this standardization mode is not for spectral feature, but this one is very user-friendly function for Sony products. So we can equip the standardization mode that achieves so day 0 and day next year's result condition will be the same. So as long as the environmental temperature or parts condition, it doesn't matter. We can actually standardize with QCBs. And this is not only same products, but if you want to work with other institutes or other universities in the world, so instrument A in India or instrument B in India or instrument B in Europe or US, that condition can be equal. So I just summarized our ID7000 features like 3, as I explained. So we are very unique function for spectral libraries, and that our ID7000 achieve 44 colors analysis, and the autofluorescence extractions also useful. And there are several user-friendly features, which is standardization, and I couldn't introduce auto sampler function as well as auto shutdown functions. Okay, from next explaining, so I'd like to touch on cell sorters. So our message is cell sorting is sorting made simple. So actually we have several features. So our most strong feature is we are using exchangeable sample line and sorting chip. And the second one is automatic setup. And the other features I'd like to explain detail the later part, but this is I'd like to touch on the seed temperature control equipped with MA900. That is a really unique function for our MA900 because some laboratories is difficult to control the temperature, and that temperature deviation is really a bad effect for the sample condition. But our seed temperature control function is subsidize those kind of actually conditions. So let me touch upon the detail of our flow cell, our product. So what I'd like to emphasize is we are innovative flow cell is using. So sample line and sorting chip can be replaceable. This is really actually useful function across the world. And what we want to say is minimize the contamination from the sample to sample, and also easy to clear the sample clogs using those kind of replaceable chip and sample line. And our replaceable chip can be chosen from 3 types: 70 microns, 100 microns, and 130 microns. And that depends on your type of cells and speed. And if the solar using, if the user is struggling for the setting up, that part also we are free from the setting, because we are achieve fully automatic setup within 15 minutes. Just insert our replaceable chip into the devices and insert and using QCBs. After that, just click in our PCs. It will be set up within 15 minutes. And those automatic setup, including laser alignment, laser delay, those kind of setting is automatically, and those functions achieve highly best sensitivity or high recovery rate and highly reproducibility. And those kind of automatic setup also helpful for the plate sortings. Not only 90 well sorting, but also 384 well plate sorting, also easy to actually enjoy in your lab. So recently, I think single cell analysis also become much popular, and that our function is useful. And someone asked us, so without any reputations in India, so it's really difficult to convince our product. But I'd like to say, our product is already contribute to the worldwide researches. So those Dr. Jennifer and Dr. Emmanuel Professor using our equipment and our equipment name in their white paper. And also this year, Nobel Prize winner of Professor David also using our SH800. That we're showing in our, yeah, thank you so much. And I really hope I'd like to work with India researchers, and I'd like to actually celebrate together with one of you or everyone. So our message is again, sorting made simple. That is an important function portion for using cell sorters. And next year onwards, we'd like to introduce new product, spectral analyzer, that is integrated into our ID7000 spectral analyzer functions and also our cell sorters. So actually, we are using replaceable flow cell also, and we are using automatic setup. That kind of strong feature we already equipped in this new product. That will be launched in 2025. So that, again, now spectral is become standard. So we'd like to explore new action researches area together with you. And this is one of the brochure. Actually, this is official brochure, and this is me actually. So if you want to see me, please come to our website and you can see me anytime. Thank you so much. And our product is made in Japan. And our Sony India team would like to support for your marketing, sales, and also service. And today, I'd like to happy to introduce our partner in India, Spinco and Biotrons. Those two companies, together with Sony India, we'd like to start India business. So don't worry about the sales service support. So we can go immediately anytime. Shukriya, Phil Mirenge. Just 10 minutes. Thank you so much.
Thank you, Team Suni. Proceeding with the next plenary lecture, I invite Dr. Dheeraj Kumar from ICGEB and Professor Siddharth Jhunjhunwala from IISc to introduce our speaker, Professor Saurav Ghosh, Professor of Neurology at Yale University School of Medicine.
Good afternoon, everybody. It's an absolute pleasure to introduce Saurav to all of you here. There's a lot to talk about Saurav, but I'll keep it very brief so that the time is given more to him. Saurav is a good friend, but more importantly, he's an amazing immunologist and does very, very precise work. And so you'll see that in his talk today. And with that, over to you, Saurav.
Can you hear me? Yeah. Thank you, everyone, for attending, and thanks to the organizers for the invitation, Dipankar, Siddharth. I'll try to speak quickly. Siddharth even took me out for lunch somewhere outside, so I have to finish my talk quickly to go to the bathroom, but haven't worked yet. All right. So what I'm going to talk about today is Alzheimer's disease. And this disease, everybody knows it's named after the physician, but it was actually on November 26, 1901, when this patient, August Dieter, was seen by Alzheimer's. And he, through interviewing her, immediately realized it's a problem of cognition. It's a cognitive disease of cognitive decline. So immunologists were not really interested in this. This was thought to be in the realms of neuroscientists because there's obviously a problem with the brain. But in 2013, the SNP analysis, looking for polymorphisms in genomes that associate with disease, revealed a gene called TREM2, a particular SNP, R47H, in TREM2, very strongly associated with Alzheimer's disease. Now, what is really interesting is TREM2 is not present in neurons. In the brain, it's only expressed in microglial cells. So that suddenly put the spotlight on microglia, the immune cells in the brain. And that these cells might have something to do, some very close association with a disease which was previously thought to be exclusively of neuronal function. Now, in a pioneering work, really this is a fundamental landmark paper that came out in 2017 from Ido Amit, Mihail Schwartz, and Marco Colonna's group, where they used a mouse model of Alzheimer's disease called 5XFAD. It has 3 of the familial Alzheimer's mutations in APP and 2 of the gamma secretase PS1 presenilin mutations in transgenically expressed in this model. And this sequenced microglia. And these are early days of sequencing. So the technology that they used was called MARS-seq. And they compared wild type and disease and microglia from this 5XFAD model. And shown over here is a figure from their paper, where in red are the genes that are exclusively associated with microglia isolated from these 5XFAD mice. So these microglia have a very distinct gene expression signature, and thereby they are in a different state. And this state is called disease-associated microglia, or DAMs. That's how this came to be known. And what was really intriguing is that in the upregulated genes in the DAMs was TREM2. So given this SNP, everybody obviously got really interested in this TREM2, and Ido's group, Marco Colonna, Ido, and in this paper, they show that if they cross this 5XFAD mouse to a TREM2 knockout background, so now this mice with the Alzheimer's alleles, they lack TREM2, you do not get these dams or the disease-associated microglia. So what's the function of the dams? The current state of knowledge is still fixated. So one of the real, one more discovery that Alzheimer's, he drew out while doing autopsy in samples from Alzheimer's disease patients after they had passed away. This is one of his slides. He realized that there is something called A-beta plaques that are deposited in the brain. So the current sort of standing is that TREM2 activates microglia to recognize A-beta, and this microglia removes A-beta by phagocytosis. So disease-associated microglia or DAMs are protective, but they're insufficiently protective because these 5XFAD mice, they start showing disease symptoms even by 6 months of age. So it's there to protect, but not doing the job very well. And so if we can improve this function of TREM2, we will cure Alzheimer's. And there is indeed a drug has completed phase 2 trial. The results are not out yet from a company called Alector. So at this point, a lot of you in the room might think, that's it, it's done, right? The disease has been cured. So, or in Hindi, right? Khel khatam pai sahajam. But we come from a culture of asking questions, right? We even question what God himself understands where creation begins, right? So this is even in the Rig Veda. So some of you might be asking questions. Is it really that simple? Is it really phagocytosis? That's that's all. Why is there a gene expression module in entirety that's upregulated and not just one gene? Selection doesn't really work at the level of an individual molecule. And Vijay beautifully talked about it in the morning, about an entire module of checkpoint inhibitors in the context of cancer. Particularly with regards to phagocytosis, it's a very interesting conundrum, because if TREM2 was able to drive microglia for improved phagocytosis and removal of A-beta, why do you need other phagocytic molecules, one being AXL? And what is really more intriguing is that AXL is a paralog of another receptor tyrosine kinase called MERTK. They have about 70% amino acid identity in the tyrosine kinase domain. Functionally, they're very similar. MERTK plays a role in phagocytosis. So why do you need AXL in the microglia, especially when microglia already expresses MERTK? So here is some data from the lab, and these are all unpublished data that I'm going to show today. We isolated microglia, shown over here as CD11b positive, CX3CR1 positive cells. And then we examined its expression of AXL and MERTK, and this is from a wild-type mouse. We indeed detect MERTK very well, even from 1-month-old mouse, 6, 12, 15. But if we look at mice at various ages, we never see AXL. And MERTK, it's not just there, it's functional. It can function in phagocytosis. This is a collaborative work with Jaime Grut-Zendler, where they, in the live mouse brain, they can induce a single neuron to die. And then you can see that the microglia shown over in green, within 6 hours, it has recognized the dead cell, moved over, cleared the corpse. But if you do the same experiment in a MERTK knockout mouse, the microglia cannot detect the dead cell. So MERTK is clearly playing a role in phagocytosis in the brain. So, AXL, the RNA-seq data, it looks at RNA. And AXL, there is not a very good correlation between RNA and protein, because there is a number of microRNAs that can regulate AXL expression. So we wanted to make sure AXL indeed is expressed at a protein level in this disease-associated microglia. We took advantage of one more gene that's expressed in these microglia called ITGAX or CD11C. Immunologists love it. It's expressed in DCs. We can use it for flow. And we now looked at microglia, CD11B positive, CX3CR1 positive, but we also tried to look for CD11C expression in this microglia to identify what are potentially DAMs. We did this experiment again at various ages in mice, starting from 2 months all the way to 15. We did it in wild type, we did it in 5XFAD. And the CD11C positive microglias only show up in this Alzheimer's mouse model, not in wild type mice. And they show up from around 3 to 4 months, clearly visible by 6 months. So they're not there initially, then they're coming up. And these are the microglia that now, if you look for AXL expression, they do express AXL. They express MER, but they do express AXL. AXL is only shown in the CD11C positive microglia, not in the CD11C microglia. So AXL is there, and we have extended this, and I'm going to just show you one other model. There are many mouse models. Part of the problem of trying to cure diseases in mice are pretty bad models. So we try to do it in other models, and the results are more or less the same. So the summary is that AXL is not present at homeostasis. MER-TK is already present and can function in microglial phagocytosis. TREM2 can function in phagocytosis, yet AXL shows up only in the context of AD. But is there a functional role of AXL, right? Or is it a mere bystander? So to test this, we took advantage of a novel mouse model made by a colleague. So AXL, as many receptor tyrosine kinases, when it gets activated, a natural feedback mechanism as part of its downregulation is that it gets cleaved by proteases. Some of the same proteases that cleave TREM2, that cleave APP in the context of the brain. So Adam 1017 cleaves AXL, which leads to signal downregulation from AXL. It's no longer functional. So one of our colleagues at Thorpe, he mutated some amino acids in a transmembrane, juxtamembrane region of AXL and rendered AXL cleavage resistant. So now there is less of the soluble cleaved AXL, and there is more membrane-associated AXL. So there is more physiologically active AXL at the membrane. This is not ectopic expression. We are not force expressing. We are just sustaining physiological AXL activity. So we got this mouse from Ed, crossed it to 5xFAD, and examined what is happening in this context. First, we had to show that indeed there is more AXL in the cell surface of microglia. And then we put this mice through a series of behavioral tests. Alzheimer's being a disease of cognitive decline, we try to model this in mice following standard tests. One of the tests, for example, is called novel object recognition, where you train a mouse to get familiar with objects placed in its cage. Then you move the mouse away, change one of the object, put it back. Now, if the mouse can remember what it has seen before, it knows that, okay, I have seen this, I'm bored of it, I want to go see something new. So it spends much more time with the novel object. And that we can capture through heat maps in video images. So here is a wild type mouse spending most of the time with a novel object. Here is an Alzheimer's mouse which is spending equal time between both objects, presumably because it has lost its ability to remember what it has seen before. And the data can be quantitated over here. So what happens now if we give the mouse a non-cleavable AXL allele? These are germline. So a non-cleavable AXL, despite carrying the 5xFAD, does it confer any protection? Yes, indeed. If you look here at the 5xFAD AXL CR genotype in the mouse, they look just like wild type, despite carrying the 5xFAD, which under normal conditions would render this mouse susceptible to cognitive decline. We did this not just at 6 months, we tested this mice at 9 months, and even at 15 months. And even at 15 months, these mice remain impermeable to the 5xFAD-induced cognitive decline. They're still fine. We didn't settle for just one test. We did multiple other behavioral tests. Here is a test called Barnes maze, where the mice have to learn where there is an escape hole, and we can figure that out by testing them at a later date. And typically, wild-type mouse would spend most of the time in the quadrant where the escape hole is. But an Alzheimer's mouse, by contrast, is pretty much spending time randomly in any quadrant because they cannot remember presumably because they can't remember where the target escape hole is. But if you look at 5xFAD AXL CR, they again behave just like wild type. They seem to remember where the escape hole is. We also did actual electrophysiological analysis of circuit of the CA1, CA3 circuit that you can measure the synaptic plasticity by something called LTP, or long-term potentiation. And again, here you can see, by contrast to the wild type, the 5x FAD has reduced LTP, but if you have 5x FAD allele with the AXL CR allele, then they are just like wild type. So they have LTP. So we also looked at synapse, and I'm, in the interest of time, I'm skipping it. So in conclusion, by improving AXL function, we can actually confer resistance against Alzheimer's-induced cognitive decline. Now, this is a germline allele, right? So is AXL really functioning in the microglia? We had some circumstantial evidence because AXL is not really expressed in any other cells of the brain. There is some AXL expression in astrocytes, but in the context of Alzheimer's disease mouse model, it's actually reduced expression, and we can look at it by RNA and by protein level. But instead of circumstantial evidence trying to address this more directly, what we did was construct a mouse model where AXL was missing in the myeloid cells. We could do this by taking an AXL flox by flox mice, crossing it with a CSF1 receptor CRE, which will ablate AXL in myeloid cells, and also had the 5x FAD allele. So these are lots of mouse breeding and experiments. We could show that there is now reduced amount of AXL in the microglia. And now if we test these mice, and we tested these mice earlier at 4 months, when the 5x FAD still remembers, they still haven't suffered the cognitive decline. They start showing this at about 6 months of age. But in the absence of AXL in the microglia, now this 5x FAD mice shows loss of novel object recognition even at 4 months. So there is an even earlier onset of this cognitive decline in the mouse if they lose AXL. And this was also reflected in the LTP assay, which I will skip through in the interest of time. So by contrast to gain of function of AXL, which is protective, loss of AXL accelerates the disease. So adding this up, AXL seems to be indeed an important player. Now, so microglial AXL functions in conferring protections against AD-associated cognitive decline, and it indeed can be a novel therapeutic target. We didn't stop at CTLA4 after all. We went after PD-1 and PD-L1, and so in the context of Alzheimer's, these could be newer targets. Obviously, I have to recognize that we haven't specifically ruled out contribution of the little AXL that's there in astrocytes. We can do that by taking out AXL in astrocytes. Now, I would argue that this is expected. You know, this might be interesting and therapeutically important, but this is still expected. But the real surprise came from asking a very, very fundamental question. What is the mechanism? You know, is AXL doing the same thing as TREM2? So this is where we started, right? Why do we have all these different phagocytic receptors? And the published work suggested that AXL actually functions downstream of TREM2. So if you take out TREM2, you don't really get AXL upregulation. So again, everything is driven by TREM2. So we did single nuke RNA-seq for microglia, for all the cells in the brain, and then we subsetted the microglia from the various different genotypes, from the 5x FAD, from wild type, from mice where AXL was taken out from the microglia, as well as where AXL was stabilized, right? This gain of function AXL. And this is the 5x FAD, the TREM2-dependent microglia. And just by looking at the, so previously, as I mentioned, Ido Amit's group showed that in the absence of TREM2, you do not get the disease-associated microglia. Instead, you get a different state called DAM1. And similarly, in the absence of AXL, we also don't get the homeostatic microglia. We don't get this disease-associated microglia. Most of the microglia is stuck in this a different state. But we've also confirmed this by actually collaborating with Ido Amit and doing the same MAR-seq, and they report about a 50% decrease in the dams. So this is all good. But the real surprise came when we looked for what's this condition of the microglia, what's the transcriptional signature of the microglia in this gain of function AXL model. So if you see in the UMAP, this is where the TREM2 dependent microglia maps, but the AXL dependent microglia is not just more of the TREM2 dependent microglia. It maps at a very different space, suggesting that this microglia is quite different in nature. Again, one of the limitations is we don't have yet the access to the TREM2 cleavage-resistant mouse, and that would probably be the best head-to-head comparison. But we did the actual functional experiment. We made a mouse which is 5x FAD, which lacks TREM2, but has the gain of function of AXL. So if AXL is really functioning downstream of TREM2, the gain, the benefit of having stable AXL is going to go away in this system, right? What happens? So here is, again, the novel object recognition test. What I showed you, the AXL CR confers benefit in the context of a TREM2 wild type. Here is now a TREM2 knockout 5x FAD. Does AXL protect even in this condition? Indeed, it does. The AXL CR, even in a TREM2 knockout background, confers just as much protection as in a TREM2 wild type or TREM2 sufficient background. So AXL is functioning quite independent of TREM2. And I have to say, this is a very surprising result. And we obviously did other assays, supporting assays. This is a Barnes maze. Again, you can see the AXL CR dependent protection is the same in TREM2 knockout as well as in TREM2 wild type. And we also did LTP. So AXL seems to function through a mechanism distinct from TREM2. And here the things came to a full circle, right? So we started to ask why the AIMs appear better than DAMs. What is the real mechanism of microglial protection in neurodegeneration? Is it really just simply phagocytosis? That's the answer. And in the Ido Amit paper, the current Scholl paper in Cell, where they did this MAR-seq, everybody paid attention to the genes that goes up. And indeed, to be honest, many of the genes that goes up, they are phagocytic receptors. Right? And if we look at the score of the genes that goes up in 5x FAD compared to control, in the AXL CR mice, we have even a further increase in this disease-associated microglial score, in this phagocytic score. But what I think we should pay attention to is the flip side of the coin. When people see genes that goes up, we typically ignore the genes that goes down. But in that paper, if you look, there is a set of genes that goes down. And these are homeostatic markers of microglial function. Now, the function of these receptors are a little bit unclear, I would say, but these genes are used as markers for microglia. P2RY12, TMEM119, CX3CR1, all these genes actually goes down. So there is a trade-off for the 5x FAD, the DAMs in 5x FAD, the TREM2-induced DAMs. So they gain a disease-associated score while losing their homeostatic score. But unlike this, the 5x FAD AXL CR, not only can they gain the score, the disease-associated score, be it phagocytosis or whatever it is, but they still maintain their homeostatic score, their homeostatic gene expression. And this we have validated also by doing spectral flow. And here's just a couple of examples of some of those markers like MERTK and CX3CR1. You see that they go away in 5x FAD, but they're much stronger there. They're preserved in the context of AXL CR independent of the TREM2 status. So consistent with the microglia being homeostatic, if you look at the homeostasis of the whole tissue, if you look at the brain, you look at other cells, look at astrocytes, for example, they're known, so you can subcluster the astrocytes, and a couple of these clusters, cluster 3, cluster 4, they're only present in the Alzheimer's brain. So the astrocytes are also in a distinct state in the Alzheimer's brain. And this is shown over here. So if in gray are all the astrocytes, The black are the astrocytes from a homeostatic background genotype, from a wild type genotype. The 5X FAD are astrocytes from a 5X FAD or Alzheimer's background or Alzheimer's genotype. And you can see they map at different places.
Sort of.
Now, if you look,
you can
yes,
wrap up in 2 minutes,
just a couple of moments, a couple of minutes. It's 1 minute 46. I'll try to finish by then. So 5X FAD, AXL, CR, you can see the astrocytes, their transcriptional state resembles much more closely to the wild type homeostatic state than the 5X FAD. And we can also validate it. There's less GFAP positive astrocytes or activated astrocytes, and the same goes true for oligodendrocytes. So in the last minute, what I would do is make some speculation. I can be completely wrong. It could be wild. But typically, in the context of, and this is actually not unheard of for immunologists, right? So if there is an acute challenge that we face, our tissues or our system can indulge in a trade-off, where you trade off homeostatic function to acquire a new function to combat whatever is perturbing homeostasis. And after that has been resolved, the system can go back to homeostasis. This becomes a particular problem in the context of chronic disease, such as A-beta. The A-beta plaques are going nowhere, right? And now if the microglia focuses all its attention towards taking care of A-beta, protecting axons from A-beta-induced damage, they have lost or sacrificed some of their homeostatic function. And this, although it can confer transient protection, it's not going to confer lasting protection. So instead, if we can generate a state in microglia where they have acquired new function, without having to trade off their homeostatic function, I think we can generate what we are calling resilience. And we are obviously calling this immunoresilience because this is a particular state of the microglia that we are trying to confer by engineering, by first understanding the biology of microglia, and then by engineering the receptors to bring in this state, which is going to which we think is going to be better, not just against Alzheimer's, but in general in terms of preserving homeostasis in the brain as well as in tissues beyond. Now, again, this is an unfinished story. I would say there are a number of outstanding questions. We don't know what are the DAM-associated functions. Phagocytosis, I would assume, contributes in part. Don't know whether it's the whole story, but we also don't know what are the homeostatic functions. Now, in the field, there is idea about microglia playing a role in synaptic pruning, so we are examining it, but right now the questions are still not resolved. With this, I would acknowledge the people who have done the work. There's many people in the lab that contributed to it. I co-direct the lab with Carla. If you're interested in the work, you can email us with questions or of your interest. And I would also give a prop to the Global Immuno course that we are trying to teach in India for master's level students. Take a photo of the QR code, apply. We really encourage you. Thank you so much for your time. Happy to take questions.
Thanks, Saurav. Wonderful talk. So, questions?
Yes.
Saurav, wonderful talk. So, a couple of questions. Number one is that you are deleting the gene or perturbing the gene microglial cells, and you seeing changes in the function astrocytes and maybe in oligodendrocytes. So, what is it that the oligos and astrocytes are recognizing following the perturbation? That's number one question. Number two, what you said is absolutely correct, that the homeostatic functions people just trying to ignore. And DAM is a combination of so many factors, and it's basically activation of microglia. This is a processing, there's a presentation, there is phagocytosis, cytokine production. And it may not all be good for the clearing plaque, but it may be also in some cases adding fuel to the fire. So what does AXL do if you really go into fine comb the signatures and realize that the DAM is all encompassing, and what is AXL changing that score?
Right. let me try to answer your second question first. And I really appreciate that you agree with this, because it could be, the idea can be controversial in terms of losing the homeostatic function. And I entirely agree with you that there cannot be selective pressure on microglia to recognize A beta plaque. Microglia is there in in all these organisms, and Alzheimer's disease is evolved much later. So it's really a stressed state, like you said. It's a combination of phagocytosis, migration, cytokine production, and some of it probably helps in containing the neuronal damage to a certain extent, and then it ends up backfiring. What aspect exactly AXL contributes to? Again, AXL has pleiotropic function. It's involved in phagocytosis. It's anti-inflammatory. It helps in migration. So it could be any of those behaviors of microglia. Much like TREM2, it's involved in phagocytosis, anti-inflammatory signaling, like dampening inflammation. And there are other receptors. This is an entire program that's upregulated. What induces it? Like you showed so beautifully with IL-27, in terms of the checkpoints, we really don't know. And there were beautiful talks today in terms of looking at the transcription factor changes, and I think it would be wonderful to do this in this context, in how microglial states change and evolve. And your first question was about the astrocytes, and really, that's a great question. And again, I can only speculate because we really don't know. Astrocytes are very important for tripartite synapses. If microglia, let's assume, like one of the more popular functions of microglia in synaptic pruning, if that is, like, we see C1q go down, right? So if we are to assume that microglia is diverting their function away from synaptic pruning or taking care of synapses to focus on the A beta, the astrocytes are trying to compensate for the function and ending up getting activated. That could, but yeah, we have done cell chat and NICHNet and things like that, but I don't have a very clear idea of why oligos and astrocytes change states in the context of A beta, in the context of a 5X FAD. I think it's more symptomatic of a general loss of homeostasis, but what's driving it, we don't know. Yeah.
Okay. Wonderful, thought-provoking presentation. Sir, I want to ask you, there are a lot of retinal biomarkers or choroidal biomarkers that can be picked up in Alzheimer's disease in the early stage. Now, whether you have seen those things in the retinal level, or those TRAMP or AXL in the retinal tissue, because we can get access to the retinal tissue.
No, that's a great idea. I mean, there is microglia that infiltrates the retina, or that's there in the retina, right? So are they also changing states? And if we can somehow look into the retina as a surrogate for the microglial changes and use that as a diagnostic for Alzheimer's, that would be really interesting. We have been more interested in sort of the fundamental biology of microglia and homeostasis, so we haven't looked at that, but that's a good practical consideration.
Thank you,
for
sir.
diagnosis. Yeah.
There's a question from the back.
Yes, and don't call me sir. I have a name, it's Saurav. There are many sirs in the room.
Hi, Saurav.
Yes, thank you.
Yeah, I have a very basic question. I wanted to understand, why did you use a CSF1R CRE to study effects in microglia and not CX3CR1? Because CSF1R is also expressed on non-immune cells. And so the effects you are seeing in CSF1R CRE mice, how do you account for the fact that it does not have any side effects? Sorry if I've missed the point.
No. I mean, CX3R1 is also expressed in all the myeloid cells, but there are tricks that you can use with the CX3CR1 CRE-ERT2 to try and just ablate in microglia and replete the monocyte population. So on the concept, we don't really see many monocyte-derived macrophages, at least to the time point that we have looked. We look mainly at 6 months. There's good publications from Florent Genou's lab, for example, about monocyte-derived macrophages coming in. So in fact, looking at CSF1 CRE, it's serendipity. We didn't plan it this way, but it also can allow us to look at the monocyte-derived macrophages. So what, from as far as I know, CSF1 is more of a myeloid, not microglia specific. It doesn't distinguish. But those cells have been implicated in Alzheimer's, especially at late stages. So in our not necessarily by planning and design, but somehow we are lucky that we can even rule out any contribution from those cells, right? We have ablated axel from those cells, so we can test that. Yeah.
Thank you.
Okay.
Thank you.
Maybe if I can ask one quick question.
Oh,
Sorry.
sure, of course.
So, AXL is part of the TAM receptor family.
Correct.
So when you made CR-AXL, what would happen if you have CR multi-K or TAM?
Great question. So we are doing these experiments. The result, it's too preliminary, but we don't think MER does the same thing as AXL. We don't know why, but the experiments show that MER is not doing the same thing as
Certainly
AXL.
AXL doing more than just phagocytosis. It's because
multi-K
and TARO are also able to do the same job.
Yeah,
right?
so these receptors, they have a series of tyrosines in their kinase domain, and that's not identical between AXL and MER. So we are also even making point mutations to try to identify what's the difference. AXL has been is more commonly implicated in migration, which there's not so much literature about MER and migration, but whether that's true or not, we don't know. But both have been shown to be involved in phagocytosis. They're expressed in different cells, like AXL is more in dendritic cells, CD11C positive cells, and not. To elaborate just a little bit more on this point, now microglia has been even in pain, right? In spinal cord, in chronic pain, there's microglia that have been described to be CD11C positive, AXL positive, and knocking out AXL exacerbates chronic pain. So this goes to Vijay's point about this state of microglia is a stressed state, essentially. Why this program and what drives this, I don't know, and it would be really interesting to find out.
Great, thank you.
Yeah.
Thank you very much. Let's give a big hand to this wonderful.
Thank you.
Thank you to the speaker and the chair. I request the speaker and the chair to kindly come up on the dais for a group picture. We now move on to a panel discussion on immunology education and research in academia and industry. I would like to invite Dr. Jagdish Mittu to chair the session.
Both, yeah. All right. Thank you so much. Good evening, and thank you all for coming for this panel discussion. I'm not Taslim Arif Said, as has been mentioned in the booklet. He had to step out for some reason, and Dipankar was kind enough to put me in the slot. So thanks to him. The theme for today really fits into the line of science and society. The theme is called Immunology Education and Research in Academia and Industry. I read this theme as how do we go about educating people, not just a lay person, but even others. If we just walk through the posters, immunology has become so beautiful, so complex. It's not easy for even other scientists to understand exactly what's going on in immunology. So we need to discuss this. So I'm going to be inviting the panelists. One is CEO Bioengineering Therapeutics, Ms. Divya Chandradara. Divya, please come. And the second person is Dr. Vani Janaki Raman from IIT Chennai, Madras. And Mr. Jean Lei from San Diego, who's here for the first time in India. Thank you, Jean. Thank you. So the structure of this panel discussion is basically, I'll post 3 questions. Actually 2, depending on time. If Dipankar allows me, I'll have maybe a 3rd question, which is probably more controversial. So as a chair, I'm of the belief that a nation like India, to be healthier, to be tolerant, and to be peaceful, each of us, each of the citizens of these nations must understand 3 major subjects, 3 major topics. Microbiology and virology, to be aware of exactly what microbes can do, what viruses can do. Genetics, to know that there is not really too much of difference across races, which unfortunately being misused, as you see in the world, even in India. And immunology, to really know about what exactly your body is up against. What are the defenses, how these defenses can be destroyed and get into trouble. So, but immunology, like I said, has become so beautiful, so in-depth, so complicated. Like I said, just take a walk along the posters, you will know how complicated it has become, and even from some of the talks. So I would like to answer the first question, and what do the panelists think? So how do we take immunology to the society, to the young people, to the growing people, to the people in other areas who don't know much about science? So what would be your response to that? Let me start with Mr. Jean Lei. Please go ahead.
Yes, as everyone here, we just been through the COVID pandemics the last 4 years. I think during this period of time, a lot of awareness of how important of the immune system to defend ourselves, the body, to the infectious diseases. The immune system is not only the infectious diseases, inflammation, the chronic diseases, aging, Alzheimer's disease, cancer, all related to the immune systems. So the awareness of immunology becomes so important. For myself, while I was in the vet school last year, during the internship, I took advanced immunology where I learned hybridoma monoclonal antibody. And a lot of, especially the paper of the magic bullet, how can we mass production of monoclonal antibody conjugate with the drug? Today we call ADC, antibody drug conjugated to treat cancer. So that time is 40 some years ago. I'm so inspired by this new technology, hybridoma technology. So all in my career, the career for the last 40 years, all get into the antibody and immune system, immunology. So that's how I get into. Thank you.
Thank you, Jean. Divya, what do you think?
I think
First of all, thank you for organizers for inviting me for this talk. First of all, I think we all celebrate April Fool's Day, but we don't celebrate that much the World Immunology Day, which is the April 19th. I think the changes should come over there. And immunology is a sector which is deep driven. There is a lot of subcategories to learn: immunotoxicity, immunochemistry, immunodiagnostic, immunopathology. The students should get awareness of what are all these things, where this will create a curiosity even in the school days for them to go further, study, do some innovations in that. And also the social media campaign should happen. Vaccination. Now, polio is almost 100% eradicated from the country. But what about if you ask a common man, a layman, questions, what are the vaccinations you get in the first 5 years? Not everybody will be able to answer those things. I think that kind of awareness should come. And I would like to give one small example of what we have seen, experienced. We were doing one of the clinical studies. The study subjects were all the truck drivers, basically. And we had one of the initial testing as a HIV testing. And for surprising, among 40 subjects, we did a random HIV rapid kit testing, 3 were found positive. The awareness is so much less. And when we told we have to educate you about this, as soon as they got to know that this is positive, they just ran away. So these kind of awareness among laymen is also very important, what I feel. And there is, in the rural areas, there are Asha Karyakartes who are working on different government policies, government things. Why immunology, vaccination is all such a neglected aspect is very surprising to me. And immunology is not just vaccines, things, and also even about the probiotics, immunomodulator, natural microbials, all these things, awareness has to be created right from school. Education is what is missing, is I feel strongly. And even for the vulnerable populations like the HIV populations, HIV patients, there should be a kind of awareness which is missing, should be created, is what I feel.
Thank you, Divya. Janaki.
Thank you. Thank you for the organizers and Professor Nandi for inviting me. And yeah, about awareness in immunology, I think this is one of the closest field amongst all the biology field, closestly relatable by people because it's in our day-to-day life. It has become a kind of fancy word, immunology, because in all advertisements that you see, hand washes to health drinks comes with immune boosters. That's how it's being advertised. So probably it's important, that the right kind of immunology and the right spirit for immunology, is should be taken, and like how the other panelists pointed out, I think the better popular science lectures on immunology in school and college levels will definitely help. And we just have to tell them that what is the excitement about the subject, rather than flooding with the information on immunology. It's important to tell why it's fairly a young field, compared to all the other biology field. It's fairly a young field. As late as 1860s, we have been thinking malaria is caused by bad air, and cholera and malaria are caused by bad air. But so compared to that, it's a very, very fairly young field, and something as major as lymphatic vesicles planning the cranial region, we discovered it just few years ago, right? So we just need to kind of convey that it's a fairly young field and lot of things are there to be explored.
All right.
Yeah,
thank you. So basically, scientists need to spend a lot of time, a fair amount of time, to carry the society forward. Otherwise, we'll end up in situation where, for example, I come from the agri area, how GM crops is being shunned by people who don't necessarily understand the technology per se. So my next question is really relevant to what happened at the inaugural event yesterday when Dr. Amit Avasthi was inducted as the welcoming president-elect. He declared that during the next 2 years, that is during his term, he'll be really happy if he as the president during his term can carry out 2 goals. You may remember that. One is bring immunology to the colleges. Now, I think I would like to add, and I had a chat with him this afternoon, not just colleges, but high school students. And second is encourage and empower more women scientists in the domain of immunology. And I think I see that quite positive here. A lot of women are here not from just from India, but also from outside. I think there should be more and more of that. So Amit, I think we are right up that alley of what you're trying to achieve during your 2 years, and anything we can do, we should do that. So let me open that with, again, Jean. So what do you think, how is it happening where you are in terms of education at the level as early as possible, as opposed to leaving it to until the PhD and then postdoc and all of that? How do the society, people, common person would come to know about what's happening in the area of immunology?
Let's start from myself. Once I get into the immunology, and I provide the tools, immunological reagents available for all the scientists for further scientific discovery, or for the drug company for the new drug discovery. And I always thought, as the industry side, I like to promote immunology. And this part of the industry, the social responsibility. So since Bioengineering started year 2002, and 2008, we just barely make some money. I thought it's time to get into the sponsor. So I started with the American Association of Immunologists, the AI meeting, and gradually expand to ICI, International Congress of Immunology, or IUIS, now the new name, and the local, like European Congress of Immunology, FIMSA, and the countries. Gradually, we expanding for the sponsorship. Where area I see that the area we like to promote, I'd like to be the worldwide awareness of immunology, how important of the immune. Immunology, invite them to come to join, attend the workshop or the presentation as well.
Very good, thank you. Divya?
Yeah, thank you, Jagdish, for the question. And one of the points he mentioned about how we empower women for the to contribute more in immunology as scientists. So if you see the history in 1930s, 1940s, look at the contribution of women scientists. Francis, who is the person who was the co-founder of the HIV. The dendritic cells was found by a woman. Antibody, the concept of antibody came from one of the women, Gabriel, who invented dendritic cells. Lot of things have come out from the women. I think right now also, government of Karnataka, we say there's something called Elevate they're doing for the startups, for encouraging entrepreneurs. But why not start women entrepreneurs in AMR or something like that? Because there's a lot of scope in innovating things in immunology, immunodiagnostic things, where you can find out whether it is whether it is a bacterial infection or viral infection. This will help a lot of AMR resistance and superbugs, what you call. There's a lot of things to innovate. I think these kind of entrepreneurial opportunities, funding and fellowships, and also I think already IISc has a tech transfer cell with them, where the industry and the scientists working here can collaborate with each other, where you can develop a beautiful technology here and transfer to one of the MNC companies. Students who love science, what do you want to do? They're not clear. I want to be in science. I want to get into life science industry. Where in life science? What are the streams available in life science? What is their interest? It's still not explored. I think this kind of awareness is very much important.
Thank you.
Thanks. About taking immunology as a subject to schools or colleges, for that matter, it all depends on teaching. right? So basically, we knowing a subject is not equivalent to we knowing to teach a subject. Given that immunology is fairly a complex subject to teach, basically, okay, and it's partly disliked because this is something that I've heard from others, is because we speak in numbers. Our cytokines are numbered, our TLRs are. Presenting it as a look, it's a memory subject. Why not present it in a very different flavor that reaches a contemporary generation, right? For example, the world talks about robustness of algorithms, robustness of AI, ML, right? But do we ever tell them, convey them the robustness of biological systems, the natural systems out there, right? So the world has excitement about, robustness of artificial systems, but what about the natural systems out there? Do we ever tell them, your plasma cell can secrete, more than 2000 antibody molecules per second? What is that robustness all about, right? Or, so many PAMs out there, so many PRRs out there, how this PAM PRR decision making is done in the cell, right? So somewhere we kind of fail. It's on our part also, where
Yes.
who teach
So-
it, rather than presenting it as a memory based subject. I mean, convey them the excitement or the robustness of the natural systems,
It should
and
not be-
why it should
yeah.
be so interesting and zealous towards pursuing it as a science. And another thing that probably, in my opinion, could be interesting for school college kids to pursue immunology as a career would be telling the career opportunities out there. It's not just about bench science, right? It could also be a policy making, such as, in case of vaccine advocacy, and when we did pandemic and, the vaccine distribution, etc. So immunologists are required in policy making and many other fields outside the bench as well. So, or even, for example, even inside immunology, using immunology as a platform, one can kind of, use different domains and traverse into different domains and look at other facets of immunology, and which our experimental setups do not really address.
All
Like,
right.
for example, phagocytes are repelled by phagocytes repel capsulated bacteria due to the sheer electrostatic forces.
Okay, so that's fine. That's fine. Thank you.
So other domains, I mean, telling students about the other career opportunities
Sure,
of immunology.
that's important as well. I
matters.
think this is where we all need to start. I'm sure many of you are excellent communicators when it comes to society, interacting with students, not just PhD and graduate students, but I think to the students in other high schools as well. So in fact, as a matter of fact, I had the pleasure of taking one of the world experts on flow cytometry, Dr. William Telford from NIH, NCI in Maryland. He came to my village along with the scientists from MIRM, Manipal Institute of Regenerative Medicine, just last month, and he was very good. I mean, the language was not a barrier; it was easy to translate and all that. But children actually in a village high school were excited about what can a microscope do. So I think it's okay. You don't have to be memory-based syllabus. It has to be more experience-based discussion. So that's where I will end up. But one more question. I think there is something.
Excuse me. May I add on?
Sure. Yeah,
comment
yes,
on
sorry.
the,
Sure.
Because sounds like a very complicated, right? Immune system very complicated.
Yes.
All the signaling pathway very
Yes.
complicated.
Yes.
How to remember chemokine? What's the number? CD number. So biology try to do some educational function here. So we made a lot of poster, make it very simple. I have a Dr. Vijay Kuchu sitting here. He made the best poster. It's called Immunological Network. So go to BioLegend's booth. There's a beautiful poster. You can see whole immune system signaling pathway.
Great.
And you talk about chemokine, we have chemokine. Every single one, we have like a cancer immunoediting, all related to cancer, how that happen, all the communications as well. So
We'd love to get that poster. Thank you. So one other question, Dipankar, if you may, before we end this. This is really relevant to research in immunology, and one of the biggest debates that's happening out there in the world about the use of animals in drug discovery. And something like this is not really being discussed in general in society, apart from having debates with the animal activists and so on. So that's a session that's probably missing here. So people do ask. I mean, also we heard talks from yesterday and today how models are not really the best models because finally translatability is not right and all that. Yet we use them because to get some information apart from the biology of that animal, also what we find in those animals, could it be applicable to humans? So this is so why should people ask why should this practice be continued? That too when there are so many amazing technologies that are available, and can there be discussion on that in the future where how we are gradually moving towards replacing animals with other technologies? It may not be perfect, but can more investment be happening in those technologies so that we reach that state of perfection? I'm not saying you should eliminate animals right away. You cannot do that. A lot of very good work coming from mice and rats and so on. But how many of us are actually really thinking about it? So any thoughts on that, Jean, from you? How we are moving towards new technologies? I know this RRR mechanism is there. Are they being really used in all labs across the world, or only a few of them are using that?
Yeah, we have a reagent, animal model of mouse or human, but we provide what scientists they're asking for, support them. I think, Divya, you probably have you in a preclinical and clinical study research. So you should probably be the answer for this question, animal or human.
I think the RRR model, we have to take it more seriously, replaceable, refinement also. And also while designing a sample size, for example, this many number of mice has to be involved in the study. We can rework on this. Why have to stick on the same sample size? This many for the low dose, this many for the high dose, this many for the medium dose. So all this can be refinement can happen, and replacement of animal modules with in vitro modules. There are now 3D modules, cellular modules. I think we should rework on a lot of things of these aspects and even the CRISPR is coming up. So there is a lot of scope there involved in this, especially replacement of animal modules. This is for the young scientists to come out with newer innovations is what I feel.
Thank you.
Yeah, I think there are efforts on all that. I mean, I'm not an expert in this field, but I can still speak about some small things that I knew. The organ on chip models and, the 3D organoids are, I think people are there nearing that.
Okay.
But I don't know if it's fully replaceable, the animal
Not yet, but
experiments
hopefully
and-
working towards that, hopefully.
Right.
So I'm going to end here with opening up for a few questions from the audience. But one thing I do want to mention is that we're all aware of the storms that hit Florida, North Carolina, Helena, I think it's called, and maybe another one. But the biggest storm, I think, individually for a body is a cytokine storm. And I think there's going to be a talk on that tomorrow, and cytokines play a huge role. So anyway, this is where I think the discussion should be happening at all different levels in society. And hopefully, when we do get results, they will be easily acceptable. So with that, I would like to open it for maybe 1 or 2 or 3 questions. Anybody wish to make a comment or a question to any of the panelists? No? Okay, fine. Yeah, so, okay. Saurav, yes.
No, I mean, it was a great conversation, and many of the things that you mentioned, I mean, there's at least one aspect that we are trying to contribute to. So we are trying to have a global immuno course for master students, changing the way we teach, more about experiential asking questions, not so much memorization. It's about the fundamentals of immunology, and this time we are going to have a day for high school students to come and play with 3D printed antibodies to learn about antibody-antigen interactions, eventually up to herd immunity. So that's part of the program this year. We are trying to do this at Ashoka, and I showed a poster, so I would encourage to spread the word about that. We're trying to contribute.
Thank you. I encourage Dipankar to take it up also at IAC, when you have the time. Okay.
Yes, thanks. Clive Gray from South Africa. I'm also part of the IUIS, and I'm the chair of the Education Committee. In fact, that we have a colleague here is also from the Vaccine Committee. But talking from the Education Committee, we also hold courses throughout the low middle income countries, including India. And there's also online pre-course material through online Immunopedia. And I think it complements very well with the global talks. So this is not only about immunology facts and concepts, but it's also about deeper learning and about grant writing and about the soft skills that go with the career of being an immunologist.
Thank you so much.
Yeah.
Anyone else? There is one there at the back. Oh, sorry, ma'am, please.
May I?
Yeah.
Okay. Yeah, maybe just a reflection as an educator. I think many of us are specialized and are able to educate in a particular area. And I think that's the context of this discussion, how we can make more awareness and knowledge in immunology. But as educators, I think we can also inspire how to think about a problem, how you approach it, how you ask the right questions. And I think it's important to consider that many of the people that we interact might become immunologists or might not, but might still learn from this way of thinking. And I know that many of us here in the room share that philosophy. And so we apply it maybe in the efforts that we do. They're maybe minor, but I think if we have that approach to inspire how to think and ask the right questions, might be impactful in immunology and beyond.
Thank you. There was one question out there at the back and one more here. Okay, just these are the last two questions. Oh,
hello.
three.
How can we reduce the huge cost bottleneck in research, especially in low to middle income countries like India?
So maybe that's a topic for another hour. So if you excuse me, I'll leave that aside, but we can discuss more on an informal chat outside. Thank you. One question here.
Yeah, so before I could ask my question, I'll just give a context. So I am a BE biotechnology student. So taking that into context, I would say in the field of computer science, there's something like hackathons and a lot of workshops that are happening. I feel as a student, if I am introduced to something like Immunothon or something like that, I mean, I don't know if it's come up yet, but something like this, I think would really make the students to have problem solving abilities and try to arise at a problem, like have industry people come and give their problem statement and see a new approach for students, like anyone who's into engineering or Masters of Science or Bachelor of Science. I think this is an initiative, and also workshops. I think hands-on experience and workshops is the only thing that makes a subject interesting for people, and as a student as well, even for me. So yeah, I think this is something that is interesting, and
That's
if
great.
someone can take it forward.
Let's work together on that. Good idea. Just last question, please. One with the blue shirt. That's it? The same thing? Okay, with this, I think we are running against time. I think the Yakshagana artists are at the back trying to practice their steps. So please join me in thanking the panelists, Dr. Jean Lay, Divya Chandradhara, and Dr. Vani Janakiraman. Thank you so much. Thank you for your patience.
Dr. Divya Chandradhara from Bioagile Therapeutics, Dr. Vani Janakiraman from IIT Madras, and Dr. Jean Lay from BioLegend, along with the chair of this session, to kindly come up for a group picture.
Sorry, one.
And the Secretary, Dr. Kaustav Nayak, to present the IIS Industry Academia Award to Dr. Jean Lay for his support to the society. Followed by the award session, we will have Yakshagana, a traditional Indian art form, and the pamphlets for the same will be distributed soon. Congratulations to Dr. Jean Lay. As we conclude the scientific talks for the day, we now transition to a mesmerizing Yakshagana performance celebrating our rich heritage. I invite Professor Gopal, who himself practices Yakshagana, to give a brief introduction about the performance and the artist.
Form originated, evolved, and nurtured in coastal and western Ghats of Karnataka. It possesses a strong and unique classical connection. It has a history of 500 years. This group performing art, Yakshagana, is a group performing art. Literally means Yaksha, nature spirits, and the people are the Gana, Yakshagana, who brings to life the characters and stories drawn from Indian epics, Ramayana, Mahabharata, Bhagavata, and Purana. This typical Western Ghats art, it is performed through rhythmic combinations of classical singing of poetic songs, accompanied by instrument percussions like Maddale and Chande and Tala, dance, acting, improvisation of the performance, expressions, dialogues, scholarly discussions, comedy, and with the typical ornament, costumes, and makeups colors. But today, the performance, as Professor Dipankar told, 1 hour, and it will be performed by the kids, okay? It's a whole night performing art originally, and now because of faster lifestyles, it is reduced to shorter versions, okay? Whole night we cannot definitely do it, nobody will sit and watch. But it is capable, okay? Another important thing is language integrity is another speciality of this art. What is language integrity? Even though it is performed from the whole night, all discussions will go in Kannada only. There will not be no single English word, okay? But it is possible they can take Samskrita shlokas for the discussion. That is allowed, okay? I think sound started. This is our Ganapati Puja. Then only they will come here, okay? So it's a all-in-one form of Indian performing art. That means anything in the art form you can see all here on one stage here. And it is a mode of propagating stories of Indian epics, Mahabharata, Ramayana, and Purana to common people through singing, dance, expression, and scholarly discussions. It is not only it gives not only an entertainment, but also enhances mental ability, teaches moral values through stories. That is the heart of this art. And for more details, I think I have given one reference, because you are all researchers, your life starts with references, okay? Without reference, you don't see any. So it is there, you can have a look. And Today's Yakshagana story is on Kamsavada. As most of you, though Indians, I think you have heard. And we will display the story. I think the pamphlet has a story and the performing sequence is also there. And we will be parallelly displaying on either side the bigger screens. And it is organized by Yakshamandala Art Group of IISc, and it will be performed by Yaksheshwari Yakshagana Troupe, Bengaluru. I think that is about the art. Hope you will enjoy it. And sorry, it's already delayed by more than an hour, half an hour. Nothing to worry. We'll try to finish in 1 hour. I think you will have your patience to watch it, then you comment. Hope you will enjoy the performance. Thank you.
Check. Check. Hello. Check, 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 check. Mic check, mic check. Hello, hello. Check, check. Check, check. Hello, hello. Mic check, mic check. Check, check. Check, check. Check, check. Hello, hello. Check, check. Check, check. Check, check. Check, check. Hello, hello. Check, check. Check, check. Hello, hello. Check, check. Check, check. Hello, hello. Hello, hello. Check, hello, hello. विघ्नेजाय सरस्वती पार्वती गुरवे नमः दक्षिणामूर्ति जैव सकल कार्यार्थ सिद्धै ಅರರೆ ನಿನ್ನಯ ಮಹಿಮೆಯನ್ನು ಪೊಗಳಲಳವಲ್ಲ ಅರರೆ ನಿನ್ನಯ ಮಹಿಮೆಯನ್ನು ಪೊಗಳಲಳವಲ್ಲ ಧರಣಿಯನು ಪತ್ತೀರ್ಥ ಮಾಶೇಷ ಗರಿದೋ ಅರನ ಸುಕುಮಾರ ಸಕಲಾಗಮಕ್ಕೆ ಸಾಕಾರ ಧರೆಯೊಳು ವಿಶೇಷ ಮದವೋರ ವಿಘ್ನೇಶ ದೇವ ಜಗದೀಶ ಮುದದಿಂದ ನಿನ್ನ ಅರದ ಮಾಧುರ ಅರದ ಮಧುರ ಮೆರೆಯುತ್ತಿದ್ದನು ಮುದದಳೊಂದಿನ ಮೆರೆಯುತ್ತಿದ್ದನು ಮುದದಳೊಂದಿನ ಹರಿದವನಿಗೆ ಜೀವನದೂತ ಕರೆಯುತ್ತೆಂದೋ
ಮಧುರ ಮಹೀಂದ್ರ ಮಹಾಮಲ್ಲ ಕಂಸ ನಾನು ನನ್ನ ತಂಗಿಯ ಮದುವೆಯನ್ನ ಚಂಬಳೆಯಿಂದ ಮಾಡಿಸಿ ಸಂತೋಷದಿಂದ ಬೀಗಿದ್ದೆ ಆಗ ಕೇಳಿಸಿದ್ದು ನಭೋವಾಣಿ ತಂಗಿಯ 8ನೆಯ ಗರ್ಭ ಸಂಜಾತನಿಂದ ನನ್ನ ಮರಣ ಎಂದು ಮರುಕ್ಷಣವೇ ಅವಳ ಕತ್ತನ್ನ ಕಡಿದು ಚೆಲ್ಲಲು ಮುಂದಾದೆ ಆದರೆ ವಸುದೇವ ತಡೆದ ಹುಟ್ಟಿದ ಮಕ್ಕಳನ್ನೆಲ್ಲ ತಂದೊಪ್ಪಿಸುತ್ತೇನೆ ದೇವಕಿಯನ್ನ ಕೊಲ್ಲಬೇಡ ಎಂದು ಬೇಡಿಕೊಂಡ. ಅವರಿಬ್ಬರನ್ನ ಕಾರಾಗೃಹದಲ್ಲಿರಿಸಿದೆ. ವಸುದೇವ ನುಡಿದಂತೆ ನಡೆದ. ಹುಟ್ಟಿದ 6 ಮಕ್ಕಳನ್ನ ಕೊಂದು ಕಳೆದೆ. 7ನೆಯದು ಗರ್ಭಪಾತವಾಯಿತು. 8ನೆಯ ಮಗುವನ್ನ ಕೊಲ್ಲಬೇಕೆಂದು ಕತ್ತಿಯನ್ನ ಎತ್ತಿದಾಗ ಆ ಹೆಣ್ಣು ಮಗು ನಿನ್ನ ವೈರಿ ನಂದಗೋಕುಲದಲ್ಲಿ ಬೆಳೆಯುತ್ತಿದ್ದಾನೆ ಎಂದು ಹೇಳಿ ಮಾಯವಾಯಿತು. ನನ್ನ ಹೊಗೆಯನ್ನು ಕಾಣಬೇಕು. ಇದಕ್ಕೆ ನಾನು ಅನುಸರಿಸಿದ ಮಾರ್ಗ
ಬಲು ಬರಲಿಲ್ಲವರು ಅಳದಿ ಬರಲಿಲ್ಲವರು. ನಂದನ ನಿಲಯದಲ್ಲಿ ಲೀಲೋ ವಿನೋದದಿ ಬೆಳೆವರಂತೆ. ನಂದನ ನಿಲಯದಲ್ಲಿ ಲೀಲಾ ವಿನೋದದಿ ಬೆಳೆವರಂತೆ. ನಂದನ ನಿಲಯದಲ್ಲಿ ಲೀಲಾ ವಿನೋದದಿ ಬೆಳೆವರಂತೆ.
ನನ್ನ ವೈರಿಗಳನ್ನ ಕೊಲ್ಲಬೇಕು. ಇದಕ್ಕಾಗಿ ಪೂತನೆಯನ್ನ ಕಳುಹಿಸಿದೆ. ಆದರೆ ಅವಳನ್ನ ಕೊಂದು ಕಳೆದ. ಅಂತೆಯೇ ಬಕ, ಧೇನುಕ, ಪ್ರಲಂಬ, ಶಕಟ, ವಾತಾಸುರ ಇವರೆಲ್ಲರನ್ನೂ ಕಳೆದುಕೊಂಡೆ. ಸ್ವಕ್ಷೇತ್ರದಲ್ಲಿ ಬಲ ಹೆಚ್ಚು. ಆದ್ದರಿಂದ ಅವರಿಬ್ಬರನ್ನ ಇಲ್ಲಿಗೆ ಕರೆಸಿಕೊಳ್ಳಬೇಕು. ಇದೇ ಸರಿಯಾದ ಮಾರ್ಗ. ಯಾರಲ್ಲಿ ಅಸುರನನ್ನು ಬರ
ಕೇಳುವುದು ಗುಣ ನಿಧಿಯ ಕೇಳುವುದು ಗುಣ ನಿಧಿಯ ಕೇಳುವುದು ಗುಣ ನಿಧಿಯೇ ಕೇಳುವುದು ಗುಣ ನಿಧಿಯೇ ಯದು ಕುಲ ಶೀಲ ಶಿಖಾಮಣಿ ಯದು ಕುಲ ಶೀಲ ಶಿಖಾಮಣಿಯೇ ನಾಳೆಗೆ ನಡೆ ಗೋಪಾಲರ ಕರೆತಾಯ್ ನಾಳೆಗೆ ನಡೆ ಗೋಪಾಲರ ಕರೆತಾಯ್ ಬಾಲಕರ ದತ್ತನು ಕೊಡುಗೊಂಬೆ ನೂದಿಟಾಯ್ ಆ ಬಾಲಕರ ದತ್ತನು ಕೊಡುಗೊಂಬೆ ನೂದಿಟಾಯ್ ನಾಳೆಗೆ ನಡೆ ಗೋಪಾಲರ ಕರೆತಾಯ್ ಬಾಲಕರ ದತ್ತನು ಕೊಡುಗೊಂಬೆ ನೂದಿಟಾಯ್.
ಜಗತ್ತಿನಲ್ಲಿ ಹುಟ್ಟಿದ ಪ್ರತಿಯೊಂದು ಜೀವಿಯು ಕೊನೆಯುಸಿರು ಇರುವ ತನಕವೂ ಬದುಕುವುದಕ್ಕಾಗಿ ಹೋರಾಡುತ್ತದೆ. ಸತ್ಯ ತಾನೇ? ಅಂತೆಯೇ ನಾನು ಕೂಡ ಪ್ರಯತ್ನಿಸುವುದು ಅಪರಾಧ ಆಗುವುದಿಲ್ಲ. ಆ ನಿಮಿತ್ತ ನೀನು ನಾಳೆಯೇ ಹೋಗಬೇಕು. ನನ್ನ ಹಳ್ಳೆಯ ಮುಳ್ಳುಗಳಾದ ಆ ರಾಮಕೃಷ್ಣರನ್ನ ಮಧುರೆಗೆ ಕರೆತರುವ ಹೊಣೆ ನಿನ್ನದ್ದು. ನನ್ನಿಚ್ಛೆಯಂತೆ ಅವರು ಇಲ್ಲಿಗೆ ಬಂದರಾದರೆ ಅವರ ಕತೆಗೆ ಕೊನೆಗಾಣಿಸುವ ಹೊಣೆ ನನ್ನದ್ದು.
ಕಡೆದು ತರುವುದೇನೋ ಸರಿ, ಆದ
ಆದರೆ ಅದಕ್ಕೆ ಕಾರಣ ಬೇಕಲ್ಲ. ಕರೆಸುವ ಕಾರಣ ಏನು ಬಲ್ಲೆ?
ಬಿಲ್ಲವ ಬಗೆನು ಚತುರ್ದಶಿ ಇಲ್ಲಿ ಬರಲಿ ಮತ್ತ, ಚತುರ್ದಶಿ ಇಲ್ಲಿ ಬರಲಿ ಮತ್ತ. ಕುಲ್ಲರ ನೀತಂ ಒಪ್ಪಿದಲೆನಗಿರಲಿ, ಕುಲ್ಲರ ನೀತಂ ಒಪ್ಪಿದಲೆನಗಿರಲಿ, ಮಾಮಲ್ಲರ ಮುಖಜು ಒಲ್ಲಿಸುವೆನು ದಿಟ, ಮಾಮಲ್ಲರ ಮುಖಜು ಒಲ್ಲಿಸುವೆನು ದಿಟ. ಕುಲ್ಲರ ನೀತಂ ಒಪ್ಪಿದಲೆನಗೆ ಕುಲ್ಲರ ಮುಖಜು ಒಲ್ಲಿಸುವೆನು ದಿಟ.
ಸರಿಯಾದ ಪ್ರಶ್ನೆಯನ್ನೇ ಕೇಳಿದ್ದೀಯ. ಈ ವಿಚಾರವಾಗಿ ಯೋಚಿಸಿ ಈಗಾಗಲೇ ಒಂದು ತೀರ್ಮಾನಕ್ಕೆ ಬಂದಿದ್ದೇನೆ. ವರ್ಷಂಪ್ರತಿ ನಡೆಯುವ ಬಿಲ್ಲ ಹಬ್ಬ ಈಗಾಗಲೇ ನಡೆದಿದೆಯಾದರೂ ಬರುವ ಚತುರ್ದಶಿಯಂದು ವಿಶೇಷವಾದ ಬಿಲ್ಲ ಹಬ್ಬವನ್ನು ಆಯೋಜಿಸುತ್ತೇನೆ. ನಮ್ಮ ಕಡೆಗೆ ಹೋಗೊಟ್ಟು ಆ ರಾಮಕೃಷ್ಣರು ಬಂದೇ ಬರುತ್ತಾರೆ. ಆಗ ನನ್ನ ಕೆಲಸ ಸಾಧಿಸಲು ಸುಲಭವಾಗುತ್ತದೆ. ಇದ್ದಾರಲ್ಲ ಸಲ್ಲ ಜಂಟಿಗಳು, ಅವರಿಂದ ಆ ರಾಮಕೃಷ್ಣರನ್ನ ಕೊಲ್ಲಿಸುವ ಯೋಚನೆ ಮಾಡಿದ್ದೇನೆ. ಈಗ ನಿನ್ನ ಕೆಲಸ ಇಷ್ಟೇ, ನೀನು ಅಲ್ಲಿಗೆ ಹೋಗಿ ಅವರಿಬ್ಬರನ್ನ ಕರೆದುಕೊಂಡು ಬರಬೇಕು.
ಏಕೆ ಕಂಸ ಪ್ರತಾಮನೋವೇತೆ, ಏಕೆ ಕಂಸ ಪ್ರತಾಮನೋವೇತೆ, ಏಕೆ ಕಂಸ, ಏಕೆ ಕಂಸ ಪ್ರತಾಮನೋವೇತೆ. ನೂಕಲು ಸುಗೈ ಹರೆಯ ಬಾರವ, ನೂಕಲೋ ಸುಗೈ, ನೂಕಲೋ ಸುಗೈ ಹರೆಯ ಬಾರವ. ಗೋಕುಲದಿ ಅವತರಿಸಿ ಮೇರೆವನು ಲೋಕವಂದ್ಯ, ಗೋಕುಲದಿ ಅವತರಿಸಿ, ಗೋಕುಲದಿ ಅವತರಿಸಿ ಮೇರೆವನು ಲೋಕವಂದ್ಯ. ಗೋಕುಲದಿ ಅವತರಿಸಿ ಮೇರೆ ಬನು ಲೋಕವಂದ್ಯ.
ಅರಸನ ಮಾತನ್ನು ವಿರೋಧಿಸುವ ಶಕ್ತಿ ನನಗಿಲ್ಲವಾದರೂ ಅಭಿಪ್ರಾಯವನ್ನು ತಿಳಿಸುವುದು ಮಂತ್ರಿಯಾದ ನನ್ನ ಹೊಣೆ. ಯಾರನ್ನು ನಿನ್ನ ಹಗೆಗಳು ಎಂದು ಭಾವಿಸಿದ್ದೀಯೋ ಅವರು ಸಾಮಾನ್ಯರಲ್ಲ. ದುಷ್ಟರನ್ನು ವಧಿಸಿ ಶಿಷ್ಟರನ್ನು ರಕ್ಷಿಸಲು ಭೂಭಾರವನ್ನು ಇಳಿಸಿ ಧರ್ಮವನ್ನು ನೆಲೆಗೊಳ್ಳುವಂತೆ ಮಾಡಲು ಅವತರಿಸಿದ ಶ್ರೀಮನ್ನಾರಾಯಣ ಎಂದು ಲೋಕ ಒಪ್ಪಿದೆ. ಪರಮಾತ್ಮನಲ್ಲಿ ಹಗೆಯ ಸಾಧಿಸಿ ಬದುಕಲು ಸಾಧ್ಯವಿಲ್ಲ. ಅವನ ಕಾಲನ್ನು ಹಿಡಿದು ಸಮಯ ಬೇಡಿದರೆ ಆಯುಷ್ಯ ವೃದ್ಧಿಸುವುದರಲ್ಲಿ ಅನುಮಾನವೇ ಇಲ್ಲ. ಎಷ್ಟೆಂದರೂ ನಿನ್ನ ಅಳಿಯೋದಿರು, ದ್ವೇಷವನ್ನು ಬಿಟ್ಟು ಪ್ರೀತಿಸುವುದೇ ಬಳಿತು ಎಂದು ನನ್ನ ಅಭಿಪ್ರಾಯ. ನಿನ್ನ ಉಪದೇಶದ ಅವಶ್ಯಕತೆ ನನಗಿಲ್ಲ. ಪ್ರೀತಿಸುವುದೇ ಆಗಿದ್ದರೆ ನನ್ನವರನ್ನೆಲ್ಲ ಕಳೆದುಕೊಳ್ಳುವ ಪ್ರಮೇಯವೇ ಬರುತ್ತಿರಲಿಲ್ಲ. ನಾವೆಯನ್ನ ಏರಿಯಾಗಿದೆ, ದೂರದ ತೀರವನ್ನ ತಲುಪುವುದು ಅನಿವಾರ್ಯ. ಹಿಂದೆ ತಿರುಗಿ ನೋಡುವ ಜಾಯಮಾನ ಕಂಸನದ್ದಲ್ಲ, ಛಲವನ್ನ ಸಾಧಿಸದೆ ಬಿಡುವವನಲ್ಲ. ನೀನು ಹೋಗಬೇಕು, ಹೋಗತಕ್ಕದ್ದು. ಇದು ರಾಜಾಜ್ಞೆ. ಕೆಡುವ ಕಾಲಕ್ಕೆ ಬುದ್ಧಿ ಇಲ್ಲ, ಸಾಯುವ ಕಾಲಕ್ಕೆ ಮದ್ದಿಲ್ಲವಂತೆ. ನಾನೆಷ್ಟೇ ನೀತಿಯ ಮಾತನ್ನು ಹೇಳಿದರೂ ಬದ್ಧಿಯಾಗುವುದಿಲ್ಲ. ಈತನಿಗೆ ಕೊನೆಗಾಲ ಸಮೀಪಿಸಿದೆ ಎಂಬುದೇ ಸತ್ಯ. ಎಷ್ಟೆಂದರೂ ನನ್ನ ಸೌಭಾಗ್ಯ. ಪರಮಾತ್ಮನನ್ನು ಕಣ್ಣಾರೆ ಕಾಣುವ ಯೋಗ. ನಂದ ಗೋಕುಲ್ಗೆ ಹೊರಡುವ ಸಿದ್ಧತೆ ಮಾಡುತ್ತೇನೆ.
ಬರಲಿತ್ತ ಅಕ್ಕುರನು ಭವಾಬ್ದಿಯೇ ಅತ್ತರವಿಯುದಯ ಜಲಕ್ಕೆ ಬರಲಿತ್ತ ಅಕ್ಕುರನು ಭವಾಬ್ದಿಯೇ ಉತ್ತರಿಪಿ ಎಂದೆನು ತಾಯ್ ವ್ರತವಡರುತ್ತೆ ಬಂದಾಯ್ ಉತ್ತರಿಪಿ ಎಂದೆನು ತಾಯ್ ರಥವಡರುತ್ತ ಬಂದಾಯ್.
ಶ್ರೀಕೃಷ್ಣನನ್ನು ನೋಡುವ ತವಕದಲ್ಲಿ ರಾತ್ರಿ ಕಳೆಯುವುದೇ ಕಷ್ಟವಾಯಿತು. ಹರಿನಾಮ ಸ್ಮರಣೆಯಲ್ಲಿ ರಾತ್ರಿಯನ್ನು ಕಳೆದೆ. ಭಾಸ್ಕರನು ಪೂರ್ವ ದಿಕ್ಕಿನಲ್ಲಿ ಮೇಲೆ ಏರಿಪಡುತ್ತಾ ಇದ್ದಾನೆ. ಇದು ಅಕ್ರೂರನ ಭಾಗ್ಯೋದಯವೂ ಹೌದು. ಅದೆಷ್ಟೋ ಜನ್ಮದ ಪಾಪದ ಲೇಪವನ್ನು ಕಳೆಯುವ ಸಮಯ ಸನ್ನಿಹಿತವಾಗಿದೆ. ತಡ ಮಾಡುವುದಿಲ್ಲ, ರಥವೇರಿ ನಡೆಯುತ್ತೇನೆ.
ಬಂಧನ ಕ್ರೂರ ಗೋವಿಂದನ ಧರುಜನಕ್ಕೆಂದು ಯಶೋದೆ ನಂದನರ ಮಂದಿರವನು ಕೊಕ್ಕೊಳ್ಳೋ ಲಕ್ಷ್ಮೀ ಮನೋ ಅಕ್ರೂರನನ್ನು ಕಂಡು ಅಕ್ಷಣವೇ ಪೀಠದಿಂದಿಳಿದೋ ಸುಕ್ಷೇಮವೇ ಬಂಧು ಭಾರ್ಯನು ತಲಪಿ ಸುಕ್ಷೇಮವೇ ಕ್ಷೇಮವೇ 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 ಸುಕ್ಷೇಮವೇ ಬಂಧು ಭಾರ್ಯನು ತಲಪಿ ಭಾರನು ತಲಪಿ ಸುವಿಚಕ್ಷಣನ ಮೈದಡವು ತಿಂದಾಳು ಸುವಿಚಕ್ಷಣನ ಮೈದಡವು ತಿಂದಾಳು.
ಅಜ್ಜಯ್ಯ, ನೀವು ಹಿರಿಯರು, ನನಗೆ ನಮಸ್ಕಾರ ಮಾಡಬಾರದು. ದೇವ, ನಿನಗೆ ನಮಸ್ಕರಿಸುವ ಭಾಗ್ಯ ನನಗೆ ದೊರೆತಿದ್ದೆ ನನ್ನ ಪುಣ್ಯ. ಅಕ್ರೂರ, ಬಹು ದೂರದಿಂದ ಬಂದವರಿದ್ದೀರಿ. ಇಲ್ಲೇ ನಿಂತು ದೊರೆಯುವುದು ಸೂಕ್ತವಲ್ಲ. ಬನ್ನಿ, ಕುಳಿತುಕೊಳ್ಳಿ. ಮಾರ್ಗಾಯಾಸದ ಪರಿಹಾರಕ್ಕೆ ಹಣ್ಣು ಹಾಲು ಇಟ್ಟಿದ್ದೇನೆ, ಸ್ವೀಕರಿಸಬೇಕು. ನಿನ್ನ ದರ್ಶನದಿಂದ ಆದ ಆಯಾಸ ಎಲ್ಲವೂ ಮಾಯವಾಯಿತು, ಹಸಿವು ನೀಗಿತು ಕೃಷ್ಣ. ಈವರೆಗೆ ಮಧುರೆಯಿಂದ ಅನೇಕರು ಬಂದದ್ದುಂಟು. ಅವರೆಲ್ಲ ರೂಪ ಮರೆಸಿ ಬಂದವರು. ನೀವು ಮಧುರೆಯಿಂದ ಬಂದವರಲ್ಲವೇ? ಹಾಗಾಗಿ ಒಂದೆರಡು ಪ್ರಶ್ನೆ.
ಮಾತೆ ದೇವಕಿಯೋ ಮಾತೆ ದೇವಕಿಯೋ ಮಾತೆ ದೇವಕಿಯೋ ವಸುದೇವಾದಿ ಯದುವೀರ ಎಲ್ಲ ವಸುದೇವಾದಿ ಯದುವೀರ ಎಲ್ಲ ವಸುದೇವಾದಿ ಯದುವೀರರೆಲ್ಲ ಕೋವಿದರ ಬೆಸಜನಗೆ ಮಾನನಿಧಿ ನೀ ಬಂದುದೋ ಕೋವಿದರೆ ಬೆಸಜನಗೆ ಮಾನನಿಧಿ ನೀ ಬಂದುದೋ ಬಂದುದಾವ ಕಾರ್ಯವು ಕೇಳುದೆನಲು ಬಂದುದಾವ ಕಾರ್ಯವು ಕೇಳುದೆನಲು ಬೇಳುದೆನಲು ಬಂದುದಾವ ಕಾರ್ಯವು ಬೇಳುದೆನಲು ಬಂದುದಾವ ಕಾರ್ಯವು ಬೇಳುದೆನಲು
ಮೊದಲಿಗೆ ಸಮಸ್ತ ವಿಚಾರವನ್ನು ಕೇಳಿ ತಿಳಿದುಕೊಳ್ಳುವ ತವಕ ನನಗೆ. ನನ್ನ ಮಾತಾಪಿತರಾದ ವಸುದೇವ ದೇವಕಿಯರು ಸುಕ್ಷೇಮವಾಗಿರುವರೇ? ಮಧುರೆಯ ಸಮಸ್ತ ಪ್ರಜಾವರ್ಗದವರು ನೆಮ್ಮದಿಯ ಬದುಕನ್ನು ಸಾಗಿಸುತ್ತಾ ಇದ್ದಾರೆಲ್ಲವೇ? ಹೌದು. ದುಷ್ಟತನವೇ ನನ್ನ ಬದುಕಿನ ಧ್ಯೇಯ ಎಂದು ಬದುಕುತ್ತಿರುವ ನನ್ನ ನಾಗಪಂಚ ಹೇಗಿದ್ದಾನೆ? ಇದೆಲ್ಲದರ ಜೊತೆಗೆ ನೀವು ಬಂದ ವಿಚಾರ ತಿಳಿಯಲಿಲ್ಲ. ನೀವು ಹೇಳಿದರೆ ಕೇಳುವ ಮನಸ್ಸುಂಟು. ಹೇಳುತ್ತೀರೋ ಕೃಷ್ಣ?
ಶ್ರೀಪತಿ ಅರಿಯದಲಪೇಕ್ಷಿಪೆಯನ್ನ ಶ್ರೀಪತಿ ಅರಿಯದಲಪೇಕ್ಷಿಪೆಯನ್ನ ದೋಸವಕೆಂದು ಮಾವ ದೋಸವಕೆಂದು ಮಾವ ಗೋಪಾಲ ಬರಲಿ ಮಧುರಾಪುರಕ್ಕೆ ಎಂದೆಂಬ ಗೋಪಾಲ ಬರಲಿ ಮಧುರಾಪುರಕ್ಕೆ ಎಂದೆಂಬ ನಿರೂಪವಿತ್ ಅಟ್ಟಿದನು ದೇವಾ ನಿರೂಪವಿತ್ ಅಟ್ಟಿದನು ದೇವಾ ಗೋಪಾಲ ಬರಲಿ ಮಧುರಾಪುರಕ್ಕೆ ಎಂದೆಂಬ ನಿರೂಪವಿತ್ ಅಟ್ಟಿದನು ದೇವಾ
ಶ್ರೀಹರಿ, ಅಕ್ರೂರ, ಈ ಪ್ರಪಂಚದಲ್ಲಿ ಚರಾಚರ ವಸ್ತು ಅಲಗಾಡಬೇಕಾದರೆ ನಿನ್ನ ದಯೆ ಅಗತ್ಯ. ಸತ್ಯ. ಹೀಗಿರುವಾಗ ಏನೂ ಅರಿಯದವನಂತೆ ನಟಿಸುತ್ತಿರುವೆಯಾ ಭಗವಂತ? ಆಗಲಿ, ನಾನೇ ಹೇಳುತ್ತೇನೆ. ನಿನ್ನ ತಂದೆ ತಾಯಿರು ಇನ್ನೂ ಬಂಧನದಲ್ಲೇ ಇದ್ದಾರೆ. ಮಧುರೆಗೆ ಎಂದು ಬಂದಾನೋ ಕೃಷ್ಣ, ನಮ್ಮ ಸೆರೆಮನೆ ವಾಸ ಎಂದು ಕೊನೆಯಾದಿತು ಎಂದು ಚಿಂತಿಸುತ್ತಾ ಇದ್ದಾರೆ. ಮಕ್ಕಳೇ ಇಲ್ಲವದ್ದಿದ್ರೂ ಚಿಂತೆ ಇರಲಿಲ್ಲ, ಆದರೆ ಇಂಥ ದುಷ್ಟ ಮಗನಿಗೆ ಜನ್ಮವಿತ್ತೇವಲ್ಲ ಎಂದು ಕೊಡಗುತ್ತಿರುವ ಉಗ್ರಸೇನ ಮತ್ತು ರುಚಿಮತಿಯರು ಒಂದು ಕಡೆಯಾದರೆ, ಕಂಸನ ದುರಾಡಳಿತದಿಂದ ಬೇಸತ್ತ ಪ್ರಜಾವರ್ಗ ಇನ್ನೊಂದು ಕಡೆ. ಬದುಕು ಬೇಡ ಎನಿಸಿದಳು ಸಾಯಲಕ್ಕೆ ಆಗದೆ ತೊಳಲಾಡುತ್ತಿರುವ ನನ್ನಂತ ಅದೆಷ್ಟೋ ಜನ. ದಿನದಿಂದ ದಿನಕ್ಕೆ ಹೆಚ್ಚುತ್ತಿರುವ ಕಂಸನ ದೌಷ್ಟ್ಯ. ಒಟ್ಟಿನಲ್ಲಿ ಎಲ್ಲವೂ ಬೇಸರದ ಸ್ಥಿತಿ. ಮತ್ತೆ ನಾನು ನಾನಾಗೆ ಬಂದವನಲ್ಲ, ಮತ್ತೆ ಕಂಸನೇ ಕಡಿದು ಹೇಳಿದ ಮಾತು, ಬಿಲ್ಲ ಹಬ್ಬಕ್ಕಾಗಿ ರಾಮಕೃಷ್ಣರನ್ನು ಕಡೆತರಬೇಕೆಂದು. ಹಾಗಾಗಿ ಇಲ್ಲಿಗೆ ಬಂದವ ನಾನು. ಬಿಲ್ಲ ಹಬ್ಬ ಕಳೆಯಲು ಬಂದವನೋ ನೀನು?
ಎಂದ ಮಾತನೋ ಎಂದ ಮಾತನೋ ಎಂದ ಮಾತನೋ ಚಂದ ಮಾತನೋ ಕೇಳಿಗರು ಜತೆ ಮಾತ ಕೇಳಿಗರು ಜತೆ ಮಂದರೋದರ ನುಡಿದ ನಾಕ್ಷಣ ಮಂದರೋದರ ಮಂದರೋದರ ನುಡಿದ ನಾಕ್ಷಣ ಎಂದ ಮಾತವ ಕೇಳಿಯರು ಜನಿ ಮಂದರೋದರ ನುಡಿದ ನಾಕ್ಷಣ ಮಂದರೋದರ ನುಡಿದ ನಾಕ್ಷಣ ಮಲ್ಲ ಮಾವನ ಅಂತ್ಯ ಬರುವವು ಬಿಲ್ಲ ಹಬ್ಬವೇ ಲೇದು ಬೈವೆಯೇ ಮಲ್ಲ ಮಾವನ ಅಂತ್ಯ ಬರುವವು ಮಲ್ಲ ಮಾವನ ಅಂತ್ಯ ಬರುವವು ವಯ್ಯ ಮಲ್ಲಮಾಯನ ಅಂತ್ಯ ಪರ್ವವ ಮಲ್ಲಮಾವನ ಅಂತ್ಯ ಪರ್ವವೋ
ಅಕ್ರೂರ, ನನ್ನ ಸಾವಿನ ನಿರೀಕ್ಷೆಯಲ್ಲಿ ಇರುವ ನನ್ನ ಮಾವ ಕಂಸ ಇಂದು ಪ್ರೀತಿಯಿಂದ ಬಿಲ್ಲ ಹಬ್ಬಕ್ಕೆ ಕರೆಯಿದ್ದಾನೆ. ಸಂತೋಷ ಅಲ್ಲದೆ ಬೇರೇನು ಹೇಳು? ನಾವು ಬರುತ್ತೇವೆ, ಆದ್ರೆ ಇಂದಲ್ಲ ನಾಳೆ ಹೊರಡೋಣ. ಅಲ್ಲಿಯ ತನಕ ನೀವು ಆ ಕಡೆ ವಿಶ್ರಾಂತಿ ಪಡೆಯಿರಿ. ಈ ವಿಷಯವನ್ನು ಅಣ್ಣನಲ್ಲಿ ತಿಳಿಸಬೇಕು. ಯಾರಲ್ಲಿ? ಅಣ್ಣನನ್ನು ಬರ ಹೇಳಿ. ಅಲ್ಲಿಗೆ ಹೋದ ನಮ್ಮನ್ನು ಕೊಲ್ಲುವ ಸಂಚು ಹೂಡಿದ್ದಾನೆ ಕಂಸ. ಹೋಗುವ ಸಿದ್ಧತೆ ಆಗಬೇಕಲ್ಲ ಅಣ್ಣ.
ತಮ್ಮ, ಅಕ್ರೂರ ಬಂದಿದ್ದರಿಂದ ನಮ್ಮವರೆಲ್ಲ ಗೊಂದಲಕ್ಕೀಡಾಗಿದ್ದಾರೆ. ಆದ್ದರಿಂದ ಸ್ವಲ್ಪ ಹಾಸ್ಯವಾಗಿ ಈ ಬಗ್ಗೆ ವಿಚಾರವನ್ನು ತಿಳಿಸೋಣ.
ನೋಡ ಬನ್ನಿರಿ ಉಗ್ರಸೇನನೋ. ನೋಡ ಬನ್ನಿರಿ, ಬನ್ನಿ ಬನ್ನಿ, ನೋಡ ಬನ್ನಿರಿ, ನೋಡ ಬನ್ನಿರಿ, ನೋಡ ಬನ್ನಿರಿ ಉಗ್ರಸೇನನು. ನೋಡ ಬನ್ನಿರಿ, ನೋಡ ಬನ್ನಿರಿ, ನೋಡ ಬನ್ನಿರಿ ಉಗ್ರಸೇನನು. ನೋಡ ಬನ್ನಿರಿ, ನೋಡ ಬನ್ನಿರಿ ಉಗ್ರಸೇನನು. ನಾಡಿ ಅಧಿಪತಿ ಅವನು ಮುಂದಿನ್ನು ಈ ನಾಡಿ ಅಧಿಪತಿ ಈ ನಾಡಿ ಅಧಿಪತಿ ಈ ನಾಡಿ ಅಧಿಪತಿ ಅವನು ಮುಂದಿನ್ನು ನಾಡಿಗಧಿಪತಿ 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 ಅವನು ಮುಂದಿನ್ನು ನಾಡಿಗಧಿಪತಿ ನಾಡಿ ಅಧಿಪತಿ ನಾಡಿ ಅಧಿಪತಿ ಅವನು ಮುಂದಿನ್ನು ನೋಡಬನ್ನಿರಿ ನಾಡಿಗಧಿಪತಿ ಅವನು ಮುಂದಿನ್ನು ಈ ನಾಡಿಗಧಿಪತಿ ಅವನು ಮುಂದಿನು ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಯಮ್ಮ ಜನನಿ ಜನಕರ ಯಮ್ಮ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಯಮ್ಮ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಮೋಕ್ಷವು ತನುಜಾನಂದಯ ದೀಕ್ಷೆಯೋ ತನುಜ ಕೊಂಸಗೆ ತನುಜ ಕೊಂಸಗೆ ಜನನಿ ಜನಕರ ಬಂದ ಮೋಕ್ಷವು ತನುಜೋ ಕಂಸಗೆ ಮೃತ್ಯು ದೀಕ್ಷೆಯು ಜನನಿ ಜನಕರ ಬಂದ ಮೋಕ್ಷವು ಮೃತ್ಯು ದೀಕ್ಷೆಯು ತನುಜ ಕಂಸಗೆ ಮೃತ್ಯು ದೀಕ್ಷೆಯೋ ಜನನಿ ಜನಕರ ಯಮ್ಮ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಯಮ್ಮ ಜನನಿ ಜನಕರ ಜನನಿ ಜನಕರ ಬನ್ನಿ 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 ನೋಡ ಬನ್ನಿರೇ ಬನ್ನಿ ನೋಡ ಬನ್ನಿರೇ ಬನ್ನಿರಿ ಬನ್ನಿ 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 ನೋಡ ಬನ್ನಿರಿ
ಯಾರು ಕೂಡ ಗೊಂದಲಕ್ಕೀಡಾಗಬೇಕಾದ ಅವಶ್ಯಕತೆ ಇಲ್ಲ. ಯಾಕೆಂದರೆ ನಾವು ಹೊರಟದ್ದು ಬಿಲ್ಲ ಹಬ್ಬಕ್ಕೆ, ಆದ್ರೆ ಇಲ್ಲಿ ನಡೆಯುವುದೇ ಬೇರೆ. ಉಗ್ರಸೇನನ ಪಟ್ಟಬಂಧ ಮಹೋತ್ಸವ ಹಾಗೂ ತಂದೆ ತಾಯಿಯರ ಬಂಧ ಮುಕ್ತಿ. ತಮ್ಮ ಇವರಿಗೆಲ್ಲ ಸಂತೈಸಿದ್ದಾಯ್ತು. ನಾವು ಹೋಗಬೇಕಾದ ಸಿದ್ಧತೆಯನ್ನು ಗೊತ್ತೇ?
ನದಿ ಉದಯ ಕಾಲದು ತುಂಬಿದೆ, ಉದಯ ಕಾಲದು ಉದಯಗಾಲದು ಎತ್ತೋ ತುಂಬಿದೆ, ಉದಯ ಕಾಲದು ಉದಯ ಕಾಲದು ಉದಯ ಕಾಲದು ಉದಯ ಕಾಲದು ತುಂಬಿದೆ. ಮಧುರೆಗೆಂದು ಮುದದಿ ಗೆಳೆಯರ ಮುದದಿ ಗೆಳೆಯರ ಮುದದಿ ಗೆಳೆಯರ ಹೊರಟರು, ಮಧುರೆಗೆಂದು ಹೊರಟರು, ಮಧುರೆಗೆಂದು ಹೊರಟರು, ಮಧುರೆಗೆಂದು
ಕೃಷ್ಣ, ವೈರಮನೆಯನ್ನು ವಿಷಾನ್ನಕ್ಕೆ ಸಮಾನ ಅಂತೆ. ಆದ್ದರಿಂದ ನಾವು ಏರಿ ಹೋಗುವ ರಥದಲ್ಲಿ ಹಾಲು, ಮೊಸರು, ತುಪ್ಪ, ಬೆಣ್ಣೆ ಎಲ್ಲವನ್ನು ಸಿದ್ಧಪಡಿಸಿದ್ದೇನೆ. ನಾಳೆ ಬೆಳಿಗ್ಗೆ ಸೂರ್ಯೋದಯಕ್ಕೆ ಸರಿಯಾಗಿ ಮದುವೆಗೆ ಸಾಗೋಣ.
ಬಂದನು ಬೇದರಿಲ್ಲದೆ ಬಂದನು ಬೇದರಿಲ್ಲದೆ ಕಳರಜಕ ಅರಸನ ಬಟ್ಟೆಯ ಮಡಿ ಮಾಡುತ್ತಲಿ ತಂದನು ಕಳರಜಕಾಯ. ಇಲ್ಲ
ನಾವು ರಾಜರಜಕ
ಮಡಿವಾಳ್ರು
ಹೌದು. ಅರಸರಿಗೂ ನಮಗೂ ಅಷ್ಟು ವ್ಯತ್ಯಾಸವಿಲ್ಲ.
ಇಲ್ವಾ?
ಹೌದಪ್ಪ.
ಏನು?
ಅರಸ ಎಷ್ಟಕ್ಷರ?
3 ಅಕ್ಷರ.
ಅಗಸ ಎಷ್ಟಕ್ಷರ?
ಸುಳ್ಳಲ್ಲ, ಇದು 3 ಅಲ್ಲ.
ಹಾಗಾದ್ರೆ ನಾವು ಸಮಾನ ಅಲ್ವಾ?
ಇದೊಂದು ವಿಷಯದಲ್ಲಿ ಹೌದು ನೋಡಿ.
ಅರಸರು ಇರುವ ಸ್ಥಳದ ಹೆಸರೇನು?
ಅರಮನೆಯಲ್ಲಿರ್ತಾರೆ.
ನಮ್ಮದು ಒಂದು ಮನೆ ಉಂಟು ಸ್ವಾಮಿ.
ಹೌದಾ?
ಹೌದು. ಅರೆಮನೆ
ಅಂದ್ರೆ
ಸೂರ್ಯ ಚಂದ್ರರನ್ನು ಒಳಗೆ ಕುಳಿತುಕೊಂಡೇ ನೋಡಿ
ಅಂದ್ರೆ ಮೇಲ್ಭಾಗ ಏನಿಲ್ಲ,
ಚಾವಣಿ ಇಲ್ಲ.
ಛೇ,
ಅರಸರಿಗೆ ಬಲ ಯಾವುದು?
ಕತ್ತಿಯ ಬಲ.
ನಮಗೂ ಒಂದು ಬಲ ಉಂಟು.
ಯಾವುದದು?
ಕತ್ತೆಯ ಬಲ.
ಕತ್ತೆಯ?
ಹಾ. ಇನ್ನು ಅರಸರು ಮಲಗಿಕೊಳ್ಳುವ ಕೋಣೆಯ ಹೆಸರೇನು?
ಶಯ್ಯಾಗಾರ ಅಂತ ಹೇಳ್ತಾರೆ.
ನಮ್ಮದು ಒಂದು ಗ್ರಹ ಉಂಟು.
ಏನದು? ಅದೆಂತ? ಅದೇನದು?
ಸೊಳ್ಳೆ ಗ್ರಹ.
ಓ ಹಾಗೆ.
ಇನ್ನು ಹೆಚ್ಚೊತ್ತು ಮಾತಾಡುವುದಲ್ಲ, ಅರಸರಿಗೆ ಬಿಟ್ಟು ಕೊಡುತ್ತೇನೆ.
ನಿನಗೆ ಕಣ್ಣುಂಟ ಕಾಣ್ತದೆ ನಿನಗೆ?
ಅಧಿಕ ಪ್ರಸಂಗದ ಮಾತಾಡಿದ್ರೆ ಮೂಗಿನ ಮುಖ ಬಾರಿಸ್ಬಿಡ್ತಾರೆ. ಎಚ್ಚರ.
ಹೌದಾ?
ನೀನು ಯಾರು ಅಂತ ಗೊತ್ತಾಗಲಿಲ್ಲ. ನಾನು ರಾಜರಜಕ.
ಯಾರಯ್ಯ ರಜಗಾನಿ ನಿಲ್ಲು ನೋಡಿದ್ದಾಳೆ, ಯಾರಯ್ಯ ರಜಗಾನಿ ನಿಲ್ಲು ನೋಡಿದ್ದ, ನೀನಿಲ್ಲು ನೋಡಿದ್ದಾಯ್. ಕೋರುವಿ ಮಡಿವಸ್ತ್ರ ಇವುದು ಮತ್ತಾಯ್, ಕೋರುವಿ ಮಡಿವಸ್ತ್ರ ಇವುದು ಮತ್ತಾಯ್. ಸೇರು ನಿನ್ನಾಲಯ ಬೇಗನೆಗವುದು, ಸೇರು ನಿನ್ನಾಲಯ ಬೇಗನೆಗವುದು. ಮೀರಿದರಿ ಮಾತು ಕೇಳು ಬಂದ ಪುದೈ, ಮೀರಿದರಿ ಮಾತು ಕೇಳು ಬಂದ ಪುದೋ.
ಓ ಮಡಿವಾಳ!
ಏ ಮಗು, ನೀನು ಮನೆಯಲ್ಲಿ ತಂದೆಗೆ ಹೀಗೆ ಏಕವಚನದಲ್ಲಿ ಮಾತಾಡ್ಸುದಾ?
ಇಲ್ಲಪ್ಪ, ತಂದೆ ತಾಯಿಗೆ ದ್ವಿವಚನವೇ.
ಮತ್ತೆ ನನ್ನನ್ನು ಓ ಮಡಿವಾಳ ಅಂತ ಅಂತಿದ್ದೆ. ನೀನು ನನ್ನ ತಂದೆ ತಾಯಿ ಅಲ್ಲವಲ್ಲ, ಹಾಗಾಗಿ ಏಕವಚನದಲ್ಲಿ ಕರೆದಿದ್ದು. ವಯಸ್ಸಿನಲ್ಲಿ ಹೆಚ್ಚು ಕಡಿಮೆ ನಾನು ನಿಮ್ಮ ತಂದೆ ಸಮಾನರೇ, ಹಾಗಾಗಿ ರಜಕರೇ ಮಡಿವಾಳರೇ ಅಂತ ಕರೆಯಬೇಕು.
ರಜಕರೇ ಮಡಿವಾಳರೇ.
ಉದ್ದ ಬೇಡ, ರಜಕರೇ ಅಷ್ಟೇ ಸಾಕು ಸಾಕು.
ಹಾ,
ಇನ್ನು ಏನು ವಿಷಯ? ನೋಡು, ಬಟ್ಟೆ ಬಟ್ಟೆ ದೂರ ದೂರ ದೂರ. ಏ ಮಗು, ನಿಮಗ್ ದಾರಿಯಲ್ಲಿ ಬರ್ಬೇಕಾದ್ರೆ ಒಂದು ಕೆರೆ ಬಾವಿ ಏನು ಸಿಗ್ಲಿಲ್ವಾ?
ಅದೆಲ್ಲ ಸಿಕ್ಕಿತ್ತು, ನಿನ್ನಂತವ್ರು ಸಿಗ್ಲಿಲ್ಲ ತೊಳಿಲಿಕ್ಕೆ.
ಹೆಚ್ಚು ಮಾತಾಡಿದ್ರೆ ಮುಖ ಹೋಗಿ ಮುಸುಳಿ ಹಾಕ್ಬೇಕು. ಸಿಟ್ಟಿ ಯಾಕೆ ಮಾಡುವುದು? ಇಗೋ ನಮ್ಮ ಬಟ್ಟೆಯನ್ನು ತಗೊಂಡು ದೂರ ದೂರ ಮಡಿ ಮಾಡಿಕೊಂಡು ಬಾ. ಇದಕ್ಕೆ ನಾನು ಒಪ್ಪದಿದ್ದರೆ
ನಿನ್ನ ಹಲ್ಲು ಮುರಿತಾನೆ.
ಸಾಕು ಸಾಕು.
ಏನೋ ಮಕ್ಕಳು ಅಂತ ಸುಮ್ಮನೆ ಬಿಟ್ರೆ ಹೆಚ್ಚು ಮಾತಾಡ್ತಾ ಇದ್ದೀರಾ? ಏ, ನಾನು ನಿಮ್ಮಂತ ಸಣ್ಣ ಜನರಿಗೆ ಬಟ್ಟೆ ತೊಳೆಯುವ ಅಂತ ಮಾಡಿದ್ರ
ಅಲ್ವಾ?
ಅಲ್ಲ, ನಾನು ರಜಕ. ಈ ಬಟ್ಟೆ ಯಾರದು ಅಂತ ಮಾಡಿದ್ರಿ? ಯಾರಿದ್ದು? ಮಧುರ ಮಹೇಂದ್ರ ಕಂಸನ ಬಟ್ಟೆ ಇದು.
ತಮ್ಮ, ಇವರಿಗೆ ನಾವಿಬ್ರು ಯಾರು ಅಂತ ಗೊತ್ತಾಗ್ಲಿಲ್ಲ.
ನಾವು ನಿನ್ನ ಅರಸನ ಅಳಿಯಂದಿರು. ಅವ ಹಿರಿಯವ ಬಲರಾಮ. ವಾ, ಹಾಗಾದ್ರೆ ನೀನು ಕಿರಿಯವ ಎಡರಾಮ.
ಛೇ, ಅವ ಕೃಷ್ಣ, ನಾವಿಬ್ರು ರಾಮಕೃಷ್ಣ.
ಓ,
ಜಾಸ್ತಿ ಮಾತಾಡಿದ್ರೆ ಮುಖಕ್ಕೆ ಹೊಡಿತೇನೆ, ಜಾಗ್ರತೆ.
ನೀವೇ ಆ ಪಾಪಿ ಮಕ್ಕಳು?
ಹೌದು, ನಾವೇ.
ಹೆಚ್ಚು ಮಾತಾಡಿದ್ರೆ ಬಟ್ಟೆ ಹೀಗೆ ಹೀಗೆ ಎಂಡಿ ಬಿಸಾಕ್ತಾನೆ, ಜಾಗ್ರತೆ. ಬಟ್ಟೆ ಕೊಡುವುದಿಲ್ವಾ? ಕೊಡುವುದಿಲ್ಲ. ಬೇಡ, ಹೋಗ ಹೋಗ ಹೋಗ. ನೋಡಿದ್ರ ಸ್ವಾಮಿ, ಅದಕ್ಕೆ ಹೇಳುದು ಒಂದು ಬಾಯಿ ಬಲ ಇರ್ಬೇಕು, ಇಲ್ಲಾಂದ್ರೆ ಕೈ ಬಲ ಇರ್ಬೇಕು ಅಂತ.
ಹೋಗ ಹೋಗ.
ಅವ ಮುಸಿ ಮುಸಿ ನಗಿಯಾಡ್ತ.
ಇದ್ದವ್ರು 3 ಜನ ಕದ್ದವ್ರು ಯಾರು? ಕೈ ತೋರ್ಸು. ಆ ಕೈ 2 ಕೈ ತೋರ್ಸ. ಇಲ್ವಾ? ಓ ಬಲರಾಮ, ಕೈ ತೋರ್ಸು. ಈ ವರ್ಷ ಮದ್ವೆ ಆಗ್ಬಹುದು.
ನೀನ್ ಇದು ಮಾಡ್ಲಿಕ್ಕೆ ಬಂದದ ಬಟ್ಟೆ ತಗೊಂಡೋಗ್ಲಿಕ್ಕೆ?
ಬಟ್ಟೆ ಕೊಡ. ಬಟ್ಟೆ
ರಜಕನ ಕೊಂದದ್ದಾಯ್ತು, ಮದ್ದಾನೆಯನ್ನು ಮರ್ದಿಸಿದ್ದಾಯ್ತು,
ಕುಬ್ಜೆಯನ್ನು ಉದ್ಧರಿಸಿದ್ದಾಯ್ತು, ಶಸ್ತ್ರಾಗಾರದಲ್ಲಿ ಪೂಜಿಸಿದ್ದ ಧನಸ್ಸನ್ನು ಮುರಿದಿದ್ದಾಯ್ತು. ಈಗ ಏನಿದ್ರು ಅವನ ಆಸನಕ್ಕೆ ತೆರಳೋಣ. ಯಾರಲ್ಲಿ? ರಾಮಕೃಷ್ಣರನ್ನ ಬರ ಹೇಳಿ. ಅನಾವಶ್ಯಕವಾಗಿ ಸಿಟ್ಟು ಬಂದದ್ದಲ್ಲ. ಮತ್ತೆ ನೀವಿಬ್ಬರು ಸಂಸ್ಕಾರವಿಲ್ಲದ ಬಾಲಕರಂತೆ ವರ್ತಿಸುತ್ತಿದ್ದೀರಿ. ಸಭಾ ಮರ್ಯಾದೆ ಇಲ್ಲದ ವಿವೇಕಿಗಳಂತೆ ಬಂದು ನನ್ನ ತೊಡೆ ಏರಿದಿರಲ್ಲ. ಹೌದು, ನನ್ನಲ್ಲಿ ಅಷ್ಟು ಸಲಿಗೆ ಏನು? ಸಲಿಗೆ ಉಂಟು ಮಾವ.
ಮಲೆತು ನಿಲುವೆ ಏಕೆ ಮಾವ ಅಳಿಯರಲ್ಲವೇ? ಮಲೆತು ನಿಲುವೆ ಮಲೆತು ನಿಲುವೆ ಮಲೆತು ನಿಲುವೆ ಮಾವ ಅಳಿಯರಲ್ಲವೇ? ಮಲೆತು ನಿಲುವೆ ಮಲೆತು ನಿಲುವೆ ಮಲೆತು ನಿಲುವೆ ಹೇಗೆ ಮಾವ ಅಳಿಯರಲ್ಲವೇ? ಮುತ್ತಿನ ಅಳಿಯ ಅಲ್ಲವೇ? ಮುತ್ತಿನ ಅಳಿಯರಲ್ಲವೇ? ಒಲಿದು ಕರೆಸಿ ಏನು? ಒಲಿದು ಕರೆಸಿ ಒಲಿದು ಕರೆಸಿ ಒಲಿದು ಕರೆಸಿ ಒಲಿದು ಕರೆಸಿ ಏನು ತಿಳಿಯಬೇಡವೆ? ಒಲಿದು ಕರೆಸಿ ಒಲಿದು ಕರೆಸಿ ಒಲಿದು ಕರೆಸಿ ಒಲುಗೆ ಏನು ತಿಳಿಯಬೇಡವೆ? ನಿನ್ನ ಅಳಿಯರಲ್ಲವೇ? ನಿನಗೆ ತಿಳಿಯಬೇಡವೇ?
ಸೋದರ ಮಾವ ತಂದೆಗೆ ಸಮಾನ. ಓಹೋ, ನಮ್ಮ ಜನ್ಮಕ್ಕೆ ತಂದೆ ತಾಯಿಯರ ಪ್ರೀತಿ ಕಂಡವರಲ್ಲ ನಾವು. ನಿಮ್ಮನ್ನ ನೋಡಿ ಸಂತೋಷದಿಂದ ನಿಮ್ಮ ತೊಡೆಯರಿದೆವು ಮಾವ. ಕಡೆ
ಮಾವ, ಓ ಬಿಲ್ಲು ಹಬ್ಬಕ್ಕೆ ಕರೆದವರು ನೀವು. ಈಗ ನೋಡಿದ್ರೆ ಮುಸುರು ಆ ಕಡೆ ಮಣ್ಣಿನ ಎತ್ಕೊಂಡಿದ್ದೀರಿ. ಯಾಕೆ, ಏನಾಯ್ತು ನಿಮ್ಗೆ?
ಮಾತು ಆಡುವಂತಿದ್ದರೆ ಮಾತಾಗುತ್ತದೆ, ಇಲ್ಲವಾದರೆ ಬಗುಳ ಆಗುತ್ತದೆ. ಕೃಷ್ಣ
ಬಗುಳ ಬೇಡ ನೀನು ಬಲ್ಲೆನೋ ಬಲ್ಲೆನೋ.
ರಾಮಕೃಷ್ಣರೇ, ಅನಾವಶ್ಯಕವಾಗಿ ಸಿಟ್ಟು ಬಂದದ್ದಲ್ಲ. ನೀವಿಬ್ಬರು ಮಾಡಿದ ಅಪರಾಧ ಒಂದೇ ಎರಡೇ. ಪಾಪ ನನ್ನ ಅಳಿಯಂದಿರು ಆಟವಾಡಿಕೊಂಡಿರಲಿ ಎಂದು ಬಕನನ್ನು ಕಳುಹಿಸಿದೆ. ಅವನನ್ನ ಯಾಕೆ ಕೊಂದಿರಿ? ಮಾವ, ಬಕ ಪಕ್ಷಿಯಾಗಿ ಬಂದ. ಅವನ ಜೊತೆ ಆಡಲು ಹೋಗಿ ಅವನ ಕೊಕ್ಕನ್ನು ಹಿಡಿದೆ. ಅವ ಸತ್ತು ಬಿದ್ದ. ನಾವು ಕೊಲ್ಲಲಿಲ್ಲ ಮಾವ. ಶಕಟನ ಕಥೆ ಏನಾಯ್ತು?
ನನಗೆ ಗೊತ್ತಿಲ್ಲ, ಅವನ ಹತ್ರ ಕೇಳಿ.
ಹೇಳ, ನೀ ಹೇಳ.
ನನಗೆ ಗೊತ್ತಿಲ್ಲಪ್ಪ.
ಅಯ್ಯ, ನೀವೇ ಕಂಡವರು ಹೇಳ.
ನಾನಲ್ಲ ಮಾವ.
ಹೇಳ ಹೇಳ. ದೇವಕನ ಕಥೆ ಏನಾಯ್ತು?
ಮಾವ ಬಂಡಿ, ಮಾವ ಬಂಡಿ.
ಮಾವ ಬಂಡಿ ಅಲ್ಲ, ನಿಮ್ಮಿಬ್ಬರನ್ನ ಯಮನ ಬಂಡಿಯಲ್ಲಿ ಕಳುಹಿಸುತ್ತೇನೆ.
ಮಾವ, ನಾವು ಬಂಡಿ ಮೇಲೆ ಏರಿದೆವು, ಅದು ಮುರಿದು ಮುರಿದು ಬಿತ್ತು ಮಾವ. ನಾನೇನು ಮಾಡ್ಲಿಲ್ಲ.
ಏನು ಮಾಡಲಿಲ್ಲ. ಹೇಳಿದಾಗ ಕೇಳಿದಾಗ ಹೇಳುವುದಿಲ್ಲ, ಅವನನ್ನ ಕೇಳಿದಾಗ ನೀನು ಹೇಳುತ್ತೀಯ. ಹೌದೌದು. ನನ್ನ ಕೇಳಿದಾಗ ಅವನು ಹೇಳುತ್ತಾನೆ. ದೇವಕನ ಕಥೆ ಏನಾಯ್ತು? ಒಂದು ದೇನುವನ್ನು ನೋಡಿದೆವು, ಅವರ ಹತ್ತಿರ ಹೋದೆವು, ಅದು ಭಯ ಭಯ ಭಯದಿಂದ ಸತ್ತು ಬಿತ್ತು. ನಾವೇನು ಮಾಡಲಿಲ್ಲ ಮಾವ. ಎಂತ ನಾಟಕ ಆಡುತ್ತೀರಿ? ಇವರೆಲ್ಲ ಸತ್ತು ಹಾಳಾಗಿ ಹೋಗಲಿ. ಪಾಪ ಮಹಾಮಾತೆ ಪೂತನಿ ಎಷ್ಟು ಒಳ್ಳೆಯವಳವಳು. ಹೌದಾ? ನಿನಗೆ ಹಾಲುಣಿಸಿದ ಆಕೆ ತಾಯಿಯ ಸಮಾನ. ಅವಳನ್ನ ಯಾಕೆ ಕೊಂದೆ? ನನ್ನ ಅಮ್ಮ ನನಗೆ ಹೇಳಿದ್ದು, ಪೂತನಿ ಬಂದು ಇತ್ತ ರಂಗನ ಎತ್ತಿಕೊಂಬೆನು ಅಂತ ಹೇಳಿದಳಂತೆ. ಹೌದೇ? ನನ್ನ ಅಮ್ಮ ನನ್ನನ್ನು ಅವಳ ಕೈಯಲ್ಲಿ ಇತ್ತಳಂತೆ. ಸ್ವಲ್ಪ ಹೊತ್ತು ಆಟವಾಡಿಸಿ ಹಾಲುಣಿಸಲು ಪ್ರಾರಂಭಿಸಿದಳಂತೆ. ನನಗೆ ಕಣ್ಣು ಮುಚ್ಚಿ ಹಾಲು ಕುಡಿಯುವುದು ಅಭ್ಯಾಸ. ಅವಳು ಕಣ್ಣು ತೆರೆದೇ ಇದ್ದಳು. ನಾನು ಕಣ್ಣು ಬಿಟ್ಟೆ, ಅವಳ ಸತ್ತು ಬಿದ್ದಳು. ಕೃಷ್ಣ ಶಹಬ್ಬಾಸ್. ನಿನ್ನನ್ನು ಹೊಗಳಿತಲ್ವಾ? ಏನು ನಾಟಕವಾಡುತ್ತೀರಿ? ಹೌದಾ? ನೀವಿಬ್ಬರು ಏನು ಮಾಡಲೇ ಇಲ್ಲ. ಇಲ್ಲಪ್ಪ, ಎಲ್ಲರೂ ಅವರಿಂದ ಅವರೇ ಸತ್ತು ಹೋದ. ಮುಚ್ಚಿಬಾಯ್. ನಿಮ್ಮನ್ನ ಹಬ್ಬದ ಊಟಕ್ಕೆ ಕರೆಸಿದ್ದಲ್ಲ, ನಿಮ್ಮಿಬ್ಬರ ಸಂತಾನವನ್ನೇ ಮುಗಿಸಲು ಕರೆಸಿದ್ದು. ನಾವು ಬಂದಿದ್ದು ನಿಮ್ಮ ಆದಿತ್ಯ ಸೀರಿಯಲ್ಸ್ ಅಲ್ಲ, ಅದರ అన్న బాగుండే
ಕಳಗಳಿದ ನೆನ್ನೆನುತ ಸುರರು ಹೂಮಳೆಗೆರೆಯೇ ಭುವಿಯೊಳಗೆ ಕೊಂಡಾಡಿದರು ಶ್ರೀಹರಿಯ ಜಯ ಜಯವೆನುತ
ಕೃಷ್ಣ ಕಂಸನನ್ನ ಕೊಂದದ್ದಾಯ್ತು. ಈಗ ಒಂದು ಬಾಕಿ ಉಂಟು, ತಂದೆ ತಾಯಿಯರ ಬಂಧ ಮುಕ್ತಿ.
ಎಲ್ಲವನ್ನು ಮುಗಿಸಿ ಸಾಂದೀಪನಿಯವರಲ್ಲಿ ವಿದ್ಯಾಭ್ಯಾಸಕ್ಕೆ ತೆರಳೋಣ. ಎಲ್ಲರಿಗೂ ಮಂಗಳಾಗ್ಲಿ.
ಆದಿದೇವ ಸಕಲಾಗಮ ಪೂಜಿತ ಯಾದವ ಕುಲ ಮೋಹನ ರೂಪ ವೇದೋದ್ಧಾರಕ ತಿರುಪತಿ ವೆಂಕಟ ವೇಣುನಾಥ ಪ್ರೇಮ ನಾರಾಯಣ ವೇ ನಮೋ ನಮೋ
Hello, hope you enjoyed the performance. Thank you. Thank you. Sorry if delayed by 5-10 minutes. I hope you will. You don't worry. Okay, this Kamsavada, and this is a typical Yakshagana from Uttara Kannada. Okay, that means Western Ghats area of Karnataka. It's the last session. We'll introduce okay, let me introduce the artist. So today's performance is performed by Yaksheshwari Yakshagana troupe of Bangalore, organized by Yakshamandala Arts Group of IISc, which I started. And Yakshagana has 4 sections. That is okay, Chowki and all over now. Now I only introduce these two, Himmela and Mummela. Himmela is the one, the percussion and the singing. They are the main controllers of the entire performance. The Sutradhara, the controller is the Bhagavata. Let me introduce Bhagavata. Today's Bhagavata is Subrai Hebbar. And the entire direction is by Bhagavata Subrai Hebbar and